All right, welcome everyone. Thank you all for being here, both in person and virtually. My name is Heather Varaich. I am a cardiologist, and I have the honor of working as a senior clinical advisor for chronic disease to Commissioner Califf. And, believe it or not, smoking cessation is something that I've been passionate about ever since I was a kid. In fact, I remember speaking in public about it. My dad, he's a military guy who'd smoke all the time. I had childhood asthma, so it's something that I grew up sort of being really thoughtful about. So it is really amazing to sort of come full circle in a way and be part of this really amazing collaboration between our two premier public health agencies, the FDA and NIH, to really take on this challenge in a wholly different light. A few housekeeping items for those who are here both virtually and in person. First, we will only be discussing cigarette smoking cessation and not other tobacco products. Secondly, while we have public comments at the end, there won't be a Q&A with the audience here, but I do appreciate you adding your comments and questions to the online public docket. If you do have any questions about this space, please go ask the security desk or see anyone who works here for directions. Bathrooms are just around the corner. Follow the signs. There's bottled water available. There's a kiosk for snacks and coffee. It does close at 2 p.m., so there won't be any coffee available after that. And now I have the distinct pleasure of introducing Dr. Robert Califf, Commissioner of Food and Drugs at the U.S. FDA. As someone who has had the pleasure of working closely with Dr. Califf, I can share that there's no one I've ever met who is more committed to the well-being of this nation and its people. No one who has greater intellect, empathy, and humility. No one who has been more committed to serve the public health than him. Smoking-related disease in particular is something he has worked tirelessly to eradicate, and there's no greater champion for this cause than him. Delighted that he'll be kicking off this public meeting. Over to you, Dr. Califf. Thank you.
Thanks, Heather, and good morning, everyone. It is my pleasure to welcome you to FDA's White Oak campus for today's very important public meeting on smoking cessation treatments. I want to thank all of you for participating in today's meeting, both those of you who are here in person, as well as those who are joining us virtually, which includes me. I'm really sorry I can't be with you in person due to an unfortunate meeting conflict. So I especially appreciate the chance to introduce this meeting and reinforce how important I believe it to be. When I returned to the FDA 3 years ago, I knew that the work we do on tobacco products would be critical to the public health. As a cardiologist who has spent much of my career studying and working to help prevent and treat the enormous array of harms caused by tobacco products, I have special appreciation for the danger of these products and the impact that our work to reduce tobacco-related disease and death can have. As a native South Carolinian who has lived in the Carolinas most of my life and practiced at a large hospital plagued with tobacco-related illness, I'm very attuned to the complex interplay of history and culture that's in play as we grapple with this problem. It's frustrating, to say the least, that tobacco remains neck and neck with hypertension as the leading cause of preventable disease and death in the United States. Cigarette smoking kills nearly 500,000 U.S. residents each year, and millions more experience smoking-related illnesses. The good news is we've made real progress in reducing cigarette smoking in the U.S., which is a notable public health achievement. Cigarette smoking among adults has been cut in half since 2009, when the FDA gained the authority to regulate these products. And the most recent annual National Youth Tobacco Survey, just released, showed that current tobacco product use among U.S. middle and high school students has dropped to the lowest recorded level in 25 years. Within the past year alone, at least 500,000 fewer students are using tobacco products, which is largely due to the significant drop in the number of students who reported current e-cigarette use. And yet, while this progress is encouraging, we still have much more work to do, including addressing disparities in the types of products that are being used and who is using them. We also need to get much more creative and encourage thinking beyond the, quote, usual, unquote, treatment options, specifically encouraging imaginative and innovative approaches precisely what we're here today to discuss. The goal of this meeting is to stimulate ideas for developing novel products that will reduce rates of cigarette smoking. Due to the immense health and economic burdens associated with cigarette smoking, this meeting will focus on cigarette cessation and not ways to stop using other tobacco products. The topic of cessation is a critical focus of FDA's mission, and today's session builds on our longstanding work in this area, including calls for greater innovation in the space. Last year, Dr. King and I published a commentary in the Journal of the American Medical Association proposing a comprehensive care package focused on strategies at the individual, health system, and population levels. In that article, and another article just published in the Annals of Internal Medicine by the FDA NIH Working Group that organized today's meeting, we noted that this approach should be complemented by research and innovation to identify novel strategies for encouraging and treating smoking cessation. Today's discussion is also an important complement to the Department of Health and Human Services Smoking Cessation Framework released earlier this year. The framework provides a vision and a set of goals to help drive progress towards smoking cessation and to address disparities in tobacco use and cessation uptake, including by race, geography, and mental and behavioral health. One of the goals of that framework, specifically Goal 6, is to promote ongoing and innovative research to support and accelerate smoking cessation. And that's the focus of our meeting today. I want to express an opinion about how difficult today's task will be. While 30 million Americans continue to use combustible tobacco, a large number by any measure, those who have not already quit often have extraordinarily high rates of characteristics that make behavioral therapeutic intervention difficult. High rates of depression, anxiety, serious mental illness, living alone, lack of insurance, and poverty. On top of all this, almost all of these people have tried to quit and likely are deeply addicted to nicotine. So this meeting is not for the weak of heart or the uncommitted. A combination of biological and behavioral interventions, and I hope a broader array of device-based and digital health technologies are clearly needed. Discussion. I'm mindful that we're not going to solve everything today, but it's a good start, and I encourage you to actively engage throughout today's session and beyond. To that end, I know some of you will be contributing during our public comment period. We encourage you to submit additional comments or information to the docket linked to the Federal Register notice through November 21st. There's one more thing I want to mention as we begin today's conversation. While it's essential that the FDA and our family of federal agencies work together, we can't do this alone. It's critical that we also bring to the table representatives of the industry that manufactures and sells cessation products, public health, academia, clinical practitioners, and other sectors to meaningfully engage on this critically important topic. This is another essential aspect of our comprehensive approach, and it's why I'm particularly pleased that we're partnering with our NIH colleagues for today's event. So thanks in advance for your commitment to public health. I look forward to working with all of you, and I hope you have a productive meeting. It's now my great pleasure to introduce our next speaker, Dr. Larry Tabak, Principal Deputy Director of the NIH, who will provide additional introductory remarks virtually. I've depended on Larry for advice about how to work in the federal system in both of my stints at FDA, and I'm sure he'll have some important things to say today. Larry, thanks for joining us today.
Well, thanks very much, Rob, and I'd like to thank the organizers for inviting me. This is a very important meeting, and I think that as Rob detailed, while we've made good progress, we still have far ways to go. NIH supports a robust and diverse portfolio of research in tobacco science in areas ranging from basic science to the epidemiology of tobacco use, prevention interventions, and of course, work on health disparities. We've made significant progress in reducing rates of cigarette smoking, But as everybody here knows, smoking still causes unacceptably high rates of morbidity and mortality from cardiovascular disease and cancer. Smoking also contributes to persistent health disparities among certain groups. And so to adequately reach the millions of Americans who would like to quit smoking, we urgently need to support research that can identify new evidence-based and effective smoking cessation medications, devices, and digital agents. And as we do this, we need to also focus on addressing health disparities and reaching the most vulnerable populations that are in need. We are very, very pleased to be working in partnership with the FDA on this effort to bring the expertise of both of our agencies together to find, develop, and safely bring to the public the medications, devices, and digital agents that can assist in smoking cessation. Along with presentations from FDA and NIH to set the regulatory and research stage, we also look forward to hearing today from the experts, the clinicians, community-based organizations, and researchers on the front lines on the new medications, repurposed medications, such as the GLP-1 receptor agonists, and digital health and device-based therapies that might help identify the novel approaches that are needed for the high impact that we hope to achieve in this area. Again, I want to thank you all for being here and for your willingness to partner with this effort. And with that, I will turn it back to the organizers of the meeting. Thank you.
Thank you to both Drs. Califf and Tabak for those comments. And now I have the pleasure of introducing Dr. Wilson Compton. Dr. Wilson Compton is the Deputy Director of the National Institute on Drug Abuse. He has co-led our tobacco cessation working group. He has been a real champion for this cause, and it is a real pleasure to be able to hear him speak today. Thank you.
Well, thank you very much, Haider, and welcome everyone to our meeting today. I thought I'd take a moment before I go into what will be a rapid-fire brief review of a large portfolio of research from the National Institutes of Health. But before I do that, I wanted to give a quick snapshot of what's in store for you today. We're starting out with our introductions from our leadership to remind you that this is a joint effort of two of the major public health agencies within the federal government, FDA and NIH. I'm providing a brief overview of NIH-supported research, which in about 20 minutes, or before I get the hook off the stage, we'll review a broad portfolio across multiple NIH institutes that support cessation research. We'll have an update from FDA from the various centers. We'll focus on clinical and community perspectives. After that, we'll begin to focus on what are some of the promising targets for cessation. We'll move into regulatory paths forward, and that's really how we bring together the research and the regulatory environment. And that actually overall is the theme for this meeting, is the importance of working together so that we can be as efficient and effective as possible in bringing new products, new techniques, new devices to market to really have the impact on the population that's so sorely needed. Okay, now into my part of the talk. I'm going to give you, in the next few minutes, a brief overview of the NIH portfolio in smoking cessation, but of course I want to begin with an overview of what does NIH do in the tobacco area. Overall, we have a robust portfolio, but I want you to notice two things about this financial slide. It includes something like $300 million a year in research directly supported with appropriated funds to the NIH, but we also have had the pleasure of partnering with the Center for Tobacco Products now for the last 15 years to support research that is essential to move the regulatory field forward that Dr. King and colleagues at the Center for Tobacco Products manage. So that's been a major part of the NIH program as well, and it's been an important collaboration that I'll highlight for you very briefly in a couple of minutes. Of course, there are very broad themes of research for the NIH program. spanning the spectrum from basic science through prevention, through epidemiology, through understanding health disparities in vulnerable populations. As Dr. Caleb pointed out, that may be some of our most important work. As we move forward with cessation, we're recognizing that the populations impacted by tobacco are unique and have special needs that go beyond simply helping them quit smoking. And so paying attention to the broader needs is essential in this space. Just to highlight one or two examples that begin to relate to the cessation, I've been very excited by our work in genetics that have helped us understand the specific genetics at each stage of the smoking transitions, whether that's the onset of smoking, whether that's the transition from first use of tobacco products to regular use, to developing full-blown dependence, and then helping us understand the genetic basis for who's able to quit and who isn't. Within this, there's some specific work beginning to show a potential role for genetics in terms of the pharmacology and the treatment. So might there in the future be a role for understanding who will respond to treatment and who won't based on genetic background? This remains an important area for research at NIH. Okay, that's my one foray into sort of a broad-based, very large portfolio at NIH. I want to highlight for a couple of minutes a little bit about our tobacco regulatory science program, which is our joint program with the Food and Drug Administration. This is an interagency partnership. I want to highlight first the Tobacco Centers of Regulatory Science. They just had their meeting last week, and these are very robust, large-scale centers that address major targets, whether that's things like what goes on with flavors in tobacco products, what goes on with levels of nicotine in tobacco products. These are just a couple of the main themes studied in these centers across the United States. I'll just highlight for you one finding that a study coming out of the Tobacco Regulatory Science Program suggests, based on modeling, that a menthol ban might have a major impact on tobacco cessation. So while we're focusing today on the issue of new treatments to help people quit smoking, there may be policy and regulatory approaches that can also lead to major saving of lives in the United States. Now, I couldn't miss a major program called the Population Assessment of Tobacco and Health, and I've personally had the pleasure of helping to launch and spearhead this program now for the last more than a dozen years since we started this landmark longitudinal tobacco study. Now, I'm not going to go into much about this, but I had one finding that I wanted to highlight for you that focuses on how research showing that vaping products, the electronic nicotine delivery systems, may help people who smoke cigarettes daily completely stop smoking, that transitioning on fewer days may be a role. So this is a suggestion that can we look at ENDS or vaping products as part of our range of efforts in terms of tobacco cessation. The last area I want to focus on, also from PATH, focuses on a very high-risk population. This is a study that took advantage of the fact that this large-scale population study included a single measure of psychosis. So it asked people whether they had ever experienced schizophrenia or other psychotic disorders. And not unexpectedly, we show that persons with this background were much more likely to use tobacco products. All different kinds of tobacco products were more common in this high-risk population. But what really was startling to me, and I think a very important message for the field as we're thinking about developing new treatments, is that persons who reported that they'd had a psychotic illness were very interested in quitting. They had made multiple attempts. They were more likely even than those without the disorder to make an attempt to quit smoking in the last year, and they used multiple different products to help them quit. So this is a hopeful message that flies in the face of what many of my colleagues, I'm a psychiatrist, think, that our patients aren't interested. This is a very important message for our field. Okay, I've highlighted very briefly some of our major research themes across tobacco research at the NIH, but now I'm going to switch and focus specifically on the meeting topic of today. What are we doing in the cessation space, and how can we help people quit smoking based on the science that the NIH supports. This is a robust portfolio spread across multiple NIH institutes, and I'm so pleased to have partners from the National Cancer Institute, from the National Institute of Minority Health and Health Disparities, and multiple other NIH institutes to participate in this. Some of those colleagues are here today, and you'll be hearing from them in the next few hours, but this is a broad program. I'm going to go through briefly a snapshot of some of the research conducted by each of the institutes. Starting, of course, with the National Cancer Institute. It's a broad portfolio related to cessation, developing new treatment approaches, particularly behavioral approaches, focusing on implementation science. How can we take interventions that we know can be effective and bring them to the largest number of people possible? How do we work with the cancer centers across the country to bring much-needed cessation products to cancer patients, but also to the broad population, both as a way to reduce the impact of smoking and other tobacco products, but also in terms of preventing the onset of these conditions as well. A couple of specific examples would be the testing the feasibility of a smoking cessation intervention through rural community pharmacies. So this is taking advantage of a part of the healthcare system that many of us sometimes overlook, despite the fact that I bet every single one of us has interacted with a pharmacy in recent days, and so this is a very important place where interventions could be delivered more effectively. I want to shift gears and focus on the National Institute of Minority Health and Health Disparities. NIMHD conducts a number of studies related to tobacco cessation. I want to highlight one for you that I'm very pleased has been led by the person you'll be meeting a little later this morning, Dr. Monica Webb-Hooper, who conducted a very important study looking at ways to enhance tobacco quit line outcomes. So by adding a specific behavioral intervention to the quit lines, Dr. Webb-Hooper was able to show that there were improved outcomes. This is so important because it is a way to bring culturally specific and modified treatments to high-risk populations that otherwise might be overlooked, and represents an example that you'll hear a little bit more about, I think, in a future panel. Turning to the National Institute of Alcohol Abuse and Alcoholism, of course, for NIAAA, the focus will be the overlap of tobacco and alcohol. A very well-known overlap. Both persons who drink are, of course, much more likely to smoke cigarettes, and those who smoke cigarettes are much more likely to drink alcohol and have alcohol-related problems. So the question has been, do medications that help people quit smoking, medications like varenicline that you see here, or does that have an impact on alcohol consumption? How about a major treatment for alcohol consumption, naltrexone? Does that have an impact on smoking behavior? The answer is probably not. We need to be thinking about the specific treatments for each of these. So in the combination, clinicians may want to consider the combination and not just focus on one or the other, but be thinking about both simultaneously. That's at least what I take away from some of the research looking at how we can understand this overlap and the available treatments for smoking cessation and heavy alcohol consumption. I'm going to shift and focus on my own institute, the National Institute on Drug Abuse, and we have a broad portfolio here, so we'll take a little more time on this one. We're certainly focusing on both basic science. I highlighted for you the genetics work that, for the most part, was supported by the National Institute on Drug Abuse. We have multiple targets here, focusing on such novel compounds as CBD. What does CBD do to tobacco use rates? That's the kind of descriptive studies, but let's focus now on some of our clinical trials and work. We have a reasonably robust portfolio for medication development in smoking cessation. So everything from preclinical through phase 1, 1B, and phase 2 studies, highlighting for you one example of a combination of a medication with virtual reality exposure. So virtual reality exposure, in terms of cue exposure, may be one way to use behavioral interventions to address tobacco use. By combining it with a medication, it appears that at least craving may be reduced somewhat. This is obviously a fairly early finding and will need some follow-up, but is the kind of example of how our portfolio can shed light on how to combine medications with behavioral approaches or other approaches. The devices pipeline is quite significant. I think particularly about the brain stimulation devices, whether this is TMS, direct brain stimulation, or more recently, we're seeing movements into using focused ultrasound as a way to manipulate the brain pathways that we've begun to understand are so essential in what causes and the progression of all substance use disorders, including smoking addiction. I have one particular study that I wanted to highlight for you because it relates to persons with serious mental illness. This is a very interesting study looking at persons with schizophrenia, and they started out, if you look on the left, this is, well, as best you can see on these slides. On the left, what we have are, what are the brain pathways that are involved in smoking behavior and in cues and in high risk and in craving? It turns out that the same pathways are involved in persons with schizophrenia and those who don't have schizophrenia. So there's nothing unique about persons with schizophrenia in terms of the aspects of their brain pathways that are involved in smoking behaviors. They followed up with the second part of this study to use TMS, to use magnetic stimulation to address some of these targets that were identified in the first phase of their study. And at least in a small, I call this a pilot study at this stage, but at least in the pilot study, they're showing target engagement and some changes in their smoking behavior. To my mind, as somebody who has spent his career working with persons with mental illness, this is a very important observation that we may have specific treatments that are helpful for persons with schizophrenia, although I will say since it's the same targets as the rest of the population, it may be useful for many others as well. Highlight for you a little bit about our digital therapeutics pipeline, our behavior therapies pipeline. In particular, I want to focus you on dual users as an important trial. We know that there are many people in this country who not only smoke cigarettes, but they also use other tobacco products, particularly vaping products. That doesn't do enough to reduce the lung risks or the cardiovascular risks. And in fact, the combinations suggest that the studies of those that use the combination show no health benefits from the combination. You need to switch completely to see at least a reduction in exposure to toxicants from these products. So this was a study that's beginning to look at, can we focus on these dual using populations and at least get them to shift to the electronic nicotine delivery as a pathway towards abstinence and cessation. Our small business program, which you may or may not have thought about, is another pathway we have for smoking cessation research. There's, for instance, a very interesting study looking at cytosine or cytisinicline for vaping cessation. This isn't the topic for today's discussion, but is a related area of research that, of course, the NIH is supporting and is of great importance for public health. Okay. That was my rapid-fire review of a $300 million NIH portfolio, or about a $450 million overall tobacco portfolio at the NIH that includes the tobacco regulatory science work we do. There are, of course, multiple challenges and areas of opportunity. That's what today's meeting is all about, is to inspire you with areas of opportunity and to make it very clear that we are, as I like to put it, open for business. This is a very important target for us, and we are using the appropriated funds from Congress to address this major public health challenge, which is how do we help the now tens of millions of smokers change that lethal behavior and extend their lives in meaningful and important ways. When we think about some of the challenges, of course, there are issues around what are some of the new treatment regimens? Can we do things early to prepare people for a quit attempt? A lot of people that are sort of half interested in quitting, but not very, and aren't willing to take that plunge. So what can we do to help them move towards quitting? Can we have a more gradual reduction as a meaningful way to help them? Certainly clinicians have been doing that for decades. Well, can that be thought through in terms of a more specific approach? Health disparities in vulnerable populations, as I've highlighted for you, both in terms of psychiatric background and in terms of socio-demographic backgrounds, are essential as targets if we're going to have an impact overall. I have mentioned the impact of other products. I've already talked to you a little bit about dual use and the difficulties in studying that and in the importance of addressing it. Well, there are some questions about whether some of these novel tobacco products might be examples of harm reduction or might be examples of ways to promote abstinence from cigarettes. This remains an important area for investigation and study. In particular, I'm personally a little disappointed that I don't know of any e-cigarette products that are being studied in the U.S. for cessation, or at least being put forward to be approved as potential cessation aids. That's a gap. We see that e-cigarettes, for example, have been useful according to the Cochrane reviews for tobacco cessation, for smoking cessation. So what can we do to make sure that these and other products are available in the U.S. and to determine how helpful or not so helpful they end up being? Now, the last challenge, I think, is in terms of youth cessation. And as Dr. Caleb pointed out, the National Youth Tobacco Survey really gives us some wonderful news. But one of the key findings is that at least the young teenagers aren't smoking cigarettes these days. Very few are. So cigarette cessation would not be the major target for youth. And while that isn't the topic of the study today, topic of today's discussion, vaping and e-cigarette use in youth remains a major topic for research, and certainly one that the NIH, in partnership often with FDA, is very interested in promoting. But we'll save that for another time and another day. Just want to highlight for you that there are multiple funding opportunities at the NIH. This is, in some ways, a broad list, but you don't even need one of these. We have broad-based initiatives just called our research projects that scientists can apply for, and we are open to have studies across the range of topics that I hope I've inspired you to think about today, to be supported by the National Institutes of Health, with an eye for how this can link to the regulatory framework that FDA manages, so that we can make sure that we bring these new medications, new devices, new digital therapeutics out into the public as efficiently and effectively as possible. I really want to thank you for your attention this morning and just remind you that NIH is interested in novel approaches, and we really actively encourage your high-impact research proposals. Feel free to reach out to me or any of the other people that will be speaking today. If I'm not the right person to give you a suggestion about your study, I certainly know who to send it to. Thanks very much.
Thank you, Wilson, for that excellent overview, and it's so great to see NIH investing so much work really across a wide spectrum focused on tobacco cessation. Our next session is going to provide an overview of the FDA regulatory framework around tobacco cessation, and I am delighted that we have an excellent series of speakers from FDA. We'll be starting off with Dr. Mary Tan Hai from the Center for Drug Evaluation and Research. So will be followed by David McMullen from the Center for Devices and Radiological Health. And lastly, in this session, we'll be hearing from Dr. Brian King, Director for the Center for Tobacco Products.
Good morning, everybody. I'm Mary Tan Hai. I'm the Deputy Director in the Office of New Drugs in Cedar. And I have a lot of ground to cover today as I provide you the overview of FDA's regulatory framework for drug approval. And I'm first going to start off with a little history here. In 1937, the Federal Food, Drug, and Cosmetic, this is actually moving forward, okay. In 1937, the Federal Food, Drug, and Cosmetic Act was passed as a result of the sulfanilamide elixir tragedy, where there were over 100 deaths due to the preparation containing diethylene glycol. And this legislation required drug manufacturers to actually demonstrate that their drugs were safe before it got to market. In 1962, after over 1,000 babies were born with defects to mothers who were prescribed thalidomide, can you hear me? Can you hear me now? Okay, sorry, I need to get a little closer. So in 1962, after over 1,000 babies were born with defects to mothers who were prescribed thalidomide for morning sickness, Most of these cases occurred outside the United States, but there were some in the United States, and this unfortunate tragedy resulted in the Kefauver-Harris amendments being passed, and this also amended the Food, Drug, and Cosmetic Act to include a provision requiring manufacturers of drug products to establish a drug's effectiveness by substantial evidence before being marketed. Substantial evidence of effectiveness, as defined in the statute, is provided here on the slide, and I underline adequate and well-controlled investigations because I want to elaborate a little bit more on that. The characteristics of an adequate and well-controlled study are further described in our regulations, and it's really an important characteristic that all pivotal trials must have because it helps distinguish the effect of an investigational drug from other factors that may influence the findings, factors such as spontaneous change in the disease course, placebo effect, or even biased observations. FDA has generally required at least 2 adequate and well-controlled studies, each convincing on its own to establish effectiveness. However, in 1997, FDAMA was passed, which amended the Food and Drug Administration, the FD&C Act, to state that under certain circumstances, the agency can also consider 1 adequate and well-controlled clinical investigation plus confirmatory evidence. And this amendment here was really signaling that the FDA will apply some regulatory flexibility under certain circumstances. And those flexibility include the type of evidence and the quantity of evidence to establish substantial evidence of effectiveness. And recently, FDA issued 2 guidances discussing the circumstances where this flexibility may be applied and the types of confirmatory evidence that may be considered, particularly for life-threatening or severely debilitating conditions with an unmet medical need, particularly that of tobacco dependence is one. Now, very important to establishing substantial evidence of effectiveness is the efficacy endpoint that is selected for the pivotal trial. Efficacy can be defined in the following ways. It can be a measure of how a patient functions, feels, or survives. This is considered a clinical benefit. It can be a validated surrogate endpoint shown to predict a specific clinical benefit. And these 2 types of efficacy measures are relied upon to support traditional approval of marketing applications. A third type of efficacy measure is a surrogate endpoint or an intermediate clinical endpoint that is reasonably likely to predict a clinical benefit. And under this circumstance, the marketing application is approved under accelerated approval, and the applicant is required to conduct a post-approval study to confirm the clinical benefit observed with the change in the surrogate endpoint. Important here is that the finding of substantial evidence of effectiveness is necessary, but alone is not sufficient for FDA approval. The approval decision still requires, among other things, a determination that the drug is safe for the intended use. So with that, I'm now going to move from the history of drug approval to talk a little bit more about drugs approved for smoking cessation. Currently, we have products that are approved in the prescription setting. These include the nicotine replacement products that are available as a nasal spray or an inhaler. You have your non-nicotine containing smoking cessation products, bupropion or varenicline. And then in the non-prescription setting or over-the-counter setting, you have nicotine replacement therapies that are available as patches, gums, or lozenges. All these products are approved as smoking cessation treatments based on the demonstration of abstinence. That is, the demonstration of the ability to quit smoking over a period of time compared to a control. So why is abstinence the primary endpoint for products seeking a claim for smoking cessation? Demonstrating abstinence from smoking is a valid surrogate for clinical benefit. As I mentioned in a prior slide, there are 3 different types of efficacy measures. And if you're able to have a valid surrogate that is actually established to demonstrate clinical benefit, these products can be approved under traditional approval. And the basis for this is because of the strong evidence that's been established demonstrating that quitting smoking reduces the risk of cardiovascular disease and death, reduces the risk of developing COPD, reduces the risk of developing multiple types of cancer, and even in those individuals with already established heart disease or lung disease, there is evidence of a reduced risk of disease progression and even improvement on organ function. Now, while reducing the number of cigarettes smoked daily or the reduction of number of days smoked is a desirable goal, there is conflicting data that this alone in itself would translate into clinical benefit. It's not to say that a lot of individuals who are trying to smoke, as Dr. Compton had mentioned in his presentation, is that they will have to go through multiple attempts to ultimately get to the goal of abstinence. And so you'll be hearing later in session 5 about a reduce to quit treatment paradigm. The endpoint of abstinence is not a complicated endpoint to capture in clinical trials, so those who are actually involved in drug development know what is required to actually measure abstinence. In contrast, what amount of reduction in cigarettes smoked or the number of days smoked that might confer a clinical benefit has not been established. Furthermore, capturing this type of information in a clinical trial does introduce complexity that may be prone to uncertainty in data collection, reliability in data collection, and challenging interpretability of the data in a marketing application. And finally, we have approved effective therapies based on this primary endpoint, such that there really isn't evidence that it's not an achievable goal in a drug development program, provided the drug actually has a treatment effect. And I'm going to share some of the information in the next couple of slides with therapies that are approved for smoking cessation. On this slide here, this is for Nicotrol nasal spray. This is a prescription nicotine replacement therapy. It got approved based on 3 placebo-controlled double-blind trials with a minimum 3-month treatment duration. 2 of those trials allowed the product to be used out to 1 year. Quit rates are the percentage of all persons initially enrolled who continuously abstained after week 2 or 4. And this table comes from the FDA-approved label. So what you see is that Nicotrol nasal spray was more effective than placebo at 6 weeks, 3 months, 6 months, and 1 year. This next slide is for varenicline. This is the non-nicotine containing smoking cessation product available as prescription product. Its approval was based on 6 randomized controlled trials that had different objectives. Participants in 4 of these trials had a minimum 12-week of treatment, and then they were followed up for 40 weeks post-treatment. And this slide here summarized the efficacy from those 4 studies. On the left side, that graph there is actually showing the abstinence, cumulative continuous abstinence over a course of 4 weeks, measured between weeks 9 through 12. The graph on the right is looking at continuous abstinence out to a year, so from weeks 9 through 52. The 4 different studies, the first study, study 2, is actually looking at varenicline at 2 different doses compared to placebo. The second one is on a flexible dosing regimen to placebo. And the other 2 studies, studies 4 and 5, are actually using 3 treatment arms comparing varenicline to bupropion and placebo. And really the take-home message from this slide here is that across all 4 studies, regardless of dose of varenicline studied, regimen of varenicline studied, there was increased abstinence rate compared to placebo or bupropion that continued out to 1 year. So in summary, all the products that have been approved for smoking cessation have demonstrated greater quit rates over placebo, maintained for at least 4 weeks, starting as early as 6 weeks. Superiority over placebo is maintained out to 6 months for the nicotine replacement therapy products, and superiority over placebo was maintained out to 1 year with varenicline. Quit rates are higher when the therapeutic intervention, so that's the drug product, is combined with counseling or behavior intervention. Unfortunately, despite availability of effective therapies, current cessation products and interventions are underutilized, and only approximately 31% of smokers who are interested in quitting receive counseling and/or medication. There's clearly an unmet medical need here because cigarette smoking kills more than 480,000 Americans each year and remains the leading cause of preventable disease and death in the U.S. In 2022, approximately 68% of 28.8 million U.S. adults who smoke wanted to quit, and a little bit over half of them made a quit attempt, with only about 8.8% able to quit. And among those who tried to quit, only about 38% used treatment, that is counseling or medication. The other point here is that there are disparities that exist among several demographic factors, including race, ethnicity, and socioeconomic status, that negatively impact health outcomes of certain patient groups who smoke. FDA considers tobacco dependence to be a serious life-threatening condition for which a medical product intended for smoking cessation may be eligible for review under FDA's expedited programs, for which there are 4. So regardless of the reasons why there's been a challenge with respect to improving quit rates, and regardless of whether or not we have effective therapies, we also know that not all therapies work for everybody. And so this is really an opportunity for us to address the unmet need, address treatment gaps. And we can do so, as we heard earlier from Dr. Compton, by identifying factors contributing to unsuccessful quit attempts and why individuals are actually relapsing even after successfully quitting. Identifying those factors, we may also be able to identify targets, new targets for intervention. We need to increase investment for research, discovery, and development of novel therapies. And we need to consider innovative trial designs and conduct, including elements that will enable better recruitment, enrollment, and retention of underrepresented populations. At CEDAR, we have several initiatives to advance innovation in drug development. The first 3 here, the CIDD program, the MIDD program, and advancing real-world evidence. These programs were established under the user fee negotiations, and they've been fairly established in practice now. Another one is actually the Drug Development Tool Qualification Program. This was actually established under the 21st Century Cures Act, and there are 3 qualification programs. These are actually to help sponsors who are interested in developing novel endpoints and biomarkers, clinical outcome assessment tools, and also an iSTAND program, which was recently established for measures that are not in the category of either a biomarker or a clinical outcome assessment measure. And also, CEDAR has recently established 2 centers, the Center for Clinical Trial Innovation, or C3TI, and the Quantitative Medicine Center of Excellence. And these 2 centers are serving as central hubs so that there can be engagement between FDA and sponsors who are interested in actually applying innovative trial designs in their drug development program or applying quantitative medicine throughout the drug development phases. I want to emphasize a little bit on the Center for Clinical Trial Innovation, C3TI. We have 3 demonstration projects, and 2 of them, I think, are really quite applicable to smoking cessation drug development programs. The first one is the point of care or pragmatic trial demonstration project. You're going to hear a little bit more about this today in session 5. And this is important when you think about conducting clinical trials that can actually get more involved with the community setting to be able to capture and enroll patients that are historically underrepresented. The second demonstration project is the selective safety data collection. This one would be really applicable to products where they've actually been approved, studied and approved in other indications, where there's actually really a good understanding about the safety characteristics of those drugs considered repurposed drugs. You're going to hear a little bit more about that in session 4. This particular program, demonstration project, really should be considered by many companies. I think about the GLP-1 receptor agonist as a little bit near and dear to my heart. I'm actually an endocrinologist, and the first GLP-1 receptor agonist was approved some 20 years ago in my division. So it's great to actually see that there's interest in developing this for a smoking cessation program. And certainly these products here, selective safety data collection, really should be considered because it will improve efficiency in drug development. So I want to end on some encouraging news. The slide on the left, there has been a notable decline in percentage of adult smokers in the U.S. spanning several decades. But we really cannot forget that there remains 30 million U.S. adults who smoke cigarettes. So this really means that our work is not done. This is a call for multiple interested parties, both in the public setting, the private setting. We need to continue to address this unmet need and continue the trend of decreasing cigarette smoking. And on that note, I thank you for your attention. I look forward to today's meeting.
Good morning, everyone. My name is David McMullen. I'm the Director for our Office of Neurological and Physical Medicine Devices. So we've heard today already about two medical devices, types of medical devices that might play an important role in smoking cessation. In general, a medical device is a device or an instrument that is important for the cure, mitigation, prevention of diseases and doesn't achieve its intended purpose through chemical means, or else that would be for our drug colleagues. In FDA, the Center for Devices and Radiological Health, or CDRH, regulates medical devices. So we take a risk-based approach to regulating medical devices. Just so everyone hears, we have Class 1, 2, and 3. Class 1 is generally our low-risk medical devices. Class 2 for more of our moderate-risk devices, and Class 3 for our highest-risk medical devices that require pre-market approval. Some examples of these medical devices, to put a picture to the classes. For Class 1 or low risk, these can be devices such as reflex hammers, so something that you're all probably familiar with and relatively low risk. Class 2, where many of the devices you'll hear about today will likely reside, are for moderate risk devices. As I said, these can include EEG or computerized behavioral assessments. Digital therapeutics are often in Class 2. In Class 3 are more for the high-risk devices. These can include deep brain stimulators, spinal cord stimulators, and the like. Within CDRH, my office, the Office of Neurological and Physical Medicine Devices, or OHC5, regulates the majority of smoking cessation related devices. We have two teams that are focused on this mission. One is our neuromodulation psychiatry team, and the other our neurodiagnostic team. So if you're looking to develop medical devices that might include brain stimulation, such as transcranial magnetic stimulation, which we've already heard about today, or computerized behavioral therapies or other digital technologies, these might reside in our neuromodulation psychiatry team. Whereas if you're focused primarily on the diagnostic aspect, that would fall under our neurodiagnostic team. Some example of smoking cessation relevant devices, and again, I encourage you to think about these as general categories, but we've already heard today about brain stimulation through transcranial magnetic stimulation, and we do have one device that has been authorized through the FDA for those purposes. But there's many other types of devices, and it covers a wide range. So another type of device is a carbon monoxide gas analyzer, which could be used to help patients that are enrolled in smoking cessation programs. And we've heard already about digital health today and digital therapeutics. So I just wanted to highlight computerized behavioral therapy devices for psychiatric disorders that are currently regulated in the office. And none of these devices are focused specifically on smoking cessation. But again, you can imagine that many of the devices we hear about today would fall in there. Speaking of the focus on digital health, device software is applied using a risk-based approach like everything else we do in CDRH. And so I just wanted to put these up on the slide, encourage you to take a picture, and to just highlight the fact that not all device software functions, not all digital health devices are going to need to be regulated by the FDA. And so there are many things that are the focus of FDA oversight as a device, and these are particularly devices that utilize behavioral therapies for the very specific treatment of specified psychiatric or behavioral disorders. But there's also many things that are not the focus of FDA oversight, and these can include devices that provide reminders or encouragement that patients trying to quit smoking can utilize. CDRH has many programs, as was highlighted previously, to support device developers. The most common one that you've maybe heard of is our Breakthrough Devices program, and this is for devices that have the potential to provide a more effective treatment for the diagnosis of life-threatening or irreversibly debilitating diseases or conditions. This is a voluntary program, and one that provides access to more frequent and timely interactions with FDA staff. The Total Product Lifecycle Advisory, or TAP program, builds on the Breakthrough program by providing not even beyond the more frequent interactions with FDA staff, but it also brings other stakeholder interactions earlier into the process so that you can ensure that the devices you're developing really answer the questions that will be important for beyond regulators, but patients, payers, and providers. And the goal there is to bring those stakeholder inputs earlier into the process to enable better evidence development plans and ultimately lead to increased patient access at an earlier time point. So patients are at the heart of what we do here at CDRH and FDA, and so please come and interact with us, reach out via pre-sub, have an informational meeting. We'd love to talk with you and develop plans on how you can generate that evidence to demonstrate safety and effectiveness. Thank you.
Hey, good morning, folks. I am Brian King. I am the director of the Center for Tobacco Products. Thrilled to be here. I'll say there are very few things that will get me in the great room at Monday morning in full glam, and this is one of them. Very important meeting as one of the co-chairs of the NIH FDA Working Group on Cessation, along with Dr. Compton. I'm really thrilled to see today's session and to see the diverse array of folks from public and private sector. That said, there's several of you in the room, but we had about 1,000 people register for today's public meeting, and I think that's testament to the importance of this. And so look forward to the fruitful dialogue that will ensue throughout the day. That said, I'm at the tail end of the federal bureaucracy portion of today's morning, so I'm going to round out the FDA trifecta in terms of the work that we do at the Center for Tobacco Product, and then we'll get on to the rest of today's proceedings. So I'll just start with giving just an overview of tobacco regulation in the United States and how we fit in the context of some of the previous talks you've heard from FDA colleagues in the broader context of cessation in the United States. So we got authority at FDA to regulate these products back in 2009, and so we're relatively young. We're in our adolescence compared to other centers who have been around significantly longer. And as part of that, we also got the authority for certain products at that time, and then we had to deem subsequent products, and so that includes e-cigarettes as well in 2016, which I've already mentioned several times this morning. But importantly, our initial authorities at the Center for Tobacco were products containing nicotine that was derived from the tobacco plant. And so, of course, for those of us who have been in the tobacco control and prevention field for many decades know that whenever you implement a policy, people try to find loopholes. And that's exactly what happened. We started to see a shift in people shifting towards synthetic nicotine since it was not initially covered under the Act. And so fortunately, in 2022, Congress clarified our authority to also include nicotine from any source, and then that includes synthetic, also tobacco-derived, as well as others. And so that is the foundation for our authorities at CTP. In terms of regulated entity, we have authority over manufacturing, marketing, and distribution. That is the triumvirate of areas where we do have authority. And in terms of what our regulatory scope is, it is tobacco products. And so, of course, you do have some lower-risk alternative products. We know tobacco products exist on a continuum of risk, with smoke products being the most deadly, but we do know that there are lower-risk alternatives as well. But in order for those products to be marketed for cessation purposes and therapeutic claims, they would have to go through the CDER side of the house. Otherwise, if there are no therapeutic claims, they go through the Center for Tobacco Products. And so, of course, you can see there are a variety of different tobacco products under our jurisdiction. We also have authority over components and parts. And so that is apropos for, of course, conventional cigarettes, as you see here, but also e-cigarettes and others. And so that's the scope of our authority. In terms of what we can do, there's a variety of things we can and a variety that we can't. I always get a good chuckle when I see things on social media of the various things I and my colleagues should be doing. We're remiss of the fact that we do not have one iota of authority to do several of those things. That said, we know from the tobacco field, local, state, and federal national action has been the hallmark of tobacco prevention and control for many years. That said, states and localities are not preempted from taking action, many in complement to our federal authorities, but importantly, there's many things that we can't do. And so you see some of those things like taxation and smoke-free, which are things being done at state and local levels, but you also see they're providing cessation services and regulating therapeutic products. And so you may be thinking to yourself, well, Dr. King, why the hell are you up here right now if you can't do anything related to cessation? Well, it's important to consider the context of the policies and activities that our center implements. And so this is a schematic of our broad programmatic portfolio. These are the 4 key areas in terms of issuing rules and guidances, reviewing tobacco product applications, enforcement and compliance, and also education. And so if you see these various programmatic activities, cessation stems from all of them. And so think for a rule and guidance, a product standard, something that we would implement at the population level would drive people to quit smoking. And so that's why it's a critical component of the work we do. Same thing with review of tobacco product applications. We're weighing the inherent risks and benefits of these products, again, to make sure, particularly for products such as an e-cigarette, that if the benefits do outweigh the risks, that the product actually shows that it's going to transfer people from a combustible product fully. Same with enforcement, compliance, and also education, where we're doing our part to make sure that we are driving people to the resources they need to successfully quit smoking. That said, there are some important distinctions, and so one is that the Center for Tobacco Products, we authorize tobacco products for sale. We do not approve tobacco products. There is no safe tobacco product. We know they exist on a continuum of risk, but we do not approve any tobacco product. That said, our colleagues in other centers, including CEDAR, they approve, of course, with that important safe and effective standard versus the standard that we have, which is what Congress gave us in 2009 when they implemented the Family Smoking Tobacco Prevention and Control Act. And so we got appropriate for the protection of public health, which I think is correct. They couldn't give us a safe and effective standard because we know there's no safe tobacco product. That said, appropriate for the protection of public health weighs the benefits of a product versus the risks, and part of weighing that involves the benefits. And so in cases of adult smokers, that would be transitioning from a combustible product to a lower risk alternative, but also importantly, the concerns, the risks there are youth initiation as well as initiation among other vulnerable groups. That said, the work we do is part of a comprehensive approach, which again has been the hallmark of tobacco prevention and control for many decades. And so this is a schematic from a commentary that Dr. Califf, the FDA Commissioner, and I wrote and published last year in JAMA, and it really highlights that comprehensive approach. And you have this trifecta of areas where we're seeing complementary and coordinated action. One is that population-based intervention, and so that's where CTP fits in terms of our broad population-level activities, which again can drive people to quit tobacco product use, particularly smoking. But you also have activity at the health system level as well, and also at that individual level, which you've heard from my colleagues in terms of those interventions that are going to help people quit. That said, that circle in the middle is the most critical: research and innovation. And so, of course, we've continued to have activity in this space, but it's important that we continue to learn from the past, but also seek new opportunities. And that said, for the Center for Tobacco Products, we remain wholly committed of making sure we're continuing to move all 3 of these key levers simultaneously, but also that we're also continuing to look for new opportunities. And I think that's why today is so important, that we continue to look at the research, be driven by the science, but also look forward to opportunities and innovation. And so here's the summary if I put you to sleep. Very quick. Bottom line is we have authorities through the Tobacco Control Act. We've got tobacco products. As long as they're not marketed for therapeutic purposes, they're under our purview. And we do evaluate using that public health standard, which is inherently distinct from the safe and effective standard of our other regulatory colleagues at FDA. And we also have a variety of programmatic activities, again, with the goal of preventing initiation, but also importantly, promoting cessation. And so we have a key role in terms of making sure that when we do implement policies, that we have that broader infrastructure, that people can optimize that opportunity to quit smoking. And so that said, I'm really thrilled about the rest of the day. And if you care to learn anything further about the Center for Tobacco Products, we have a variety of resources, including our education campaigns and others, and we're on all the social media platforms. My understanding is the comment sections of our social media handle are wildly interesting, but just remember, folks, what happens on social media stays on Google forever, so be kind. Thank you, and have a productive day. Bye.
Thank you, everyone. And now we are going to take a quick break. We are all going to be back at about 10:20, so we have about a 15-minute break for everyone. And we have a really exciting bunch of sessions afterwards, too. So thank you.
Comprehensive smoking treatment programs. Then we will have our second presenter, who is Mr. Delmonte Jefferson, Executive Director at the Center for Black Health and Equity. He's an integral part of the national movement to ban mentholated tobacco products and is nationally recognized as an expert in the field. And then, last but not least, we will hear from Dr. Lonnie Nelson, a distinguished descendant of the Eastern Band of Cherokee Indians and cancer prevention researcher, who recently joined the faculty at Fred Hutch Cancer Center and previously served as associate professor and the inaugural chair of the Department of Nursing and System Science in the Washington State University College of Nursing. So first, please join me in welcoming Dr. Leone.
Thank you so much, Monica, and thank you all for being here, and thanks to the organizers of today's event for giving me the opportunity to free associate a little bit about what it's like to be in the clinic taking care of this problem. I am a pulmonologist, but I've been thinking about the nature of tobacco use disorder for almost 30 years now, and have evolved my point of view over time. I'd like to share some of those ideas with you today. So let's see, how do we make this go forward? How do we make this go forward? Who's in charge of the slides here? Me? Yeah, I'm pressing it. Well, I could do this without slides if that's okay with you guys. Why not? What the heck? So it turns out that oh, yeah, thanks. Thanks for that. I do move around a little bit, so if all of a sudden you can't hear me, I need the signal from you guys. So I've been actually managing this problem for about 30 years, and that perspective has really shifted for me. And I feel like if we're going to pursue innovative and imaginative, if we're going to pursue innovative and imaginative solutions to this problem, we have to think a little bit differently. We really have to align the way we think about solutions with the nature of the problem as it presents in the clinic. Now, I'm going to say a few things that might be a little bit provocative and might run counter to the way we've traditionally thought about things, so I want you to know I have no conflicts of interest. I do get some grant and some public health contract support for the work that we do, but I don't have any commercial relationships. So let's see. Hey, it worked. 3 objectives in the next couple of minutes. I want to briefly review the rationale for the chronic disease moniker that we all use. We all talk about tobacco use disorder as a chronic disease, but don't really treat it that way systematically. I want to talk a little bit about that misalignment. I want to talk a little bit about those areas of misalignment that then sort of flow downstream from there, the kind of research priorities that we have, the outcomes that we choose, et cetera. And then finally, identify some key areas for collaboration and systematic improvement. Here's the first problem. We all talk about this as though it's smoking cessation. It ain't. That's the clinical goal. I am not saying that we should not all be working towards achieving abstinence of this behavior, but the underlying problem, the smoking behavior, is really the clinical manifestation of a distortion in functional properties in the brain. What are those functions in the brain? It's not thinking. It's not rationality. It's not decision-making. The functions of the brain that are distorted after exposure to nicotine are impulsivity, compulsivity, automaticity, or a sort of automatic responsiveness, the way our brain learns. And so functionally, we really need to be thinking about this problem not in terms of what we see, but in terms of the behavioral components that drive the smoking behavior: impulsivity, compulsivity, automaticity. Why is that important? Well, it turns out there's a lot of effects, downstream sort of molecular biological effects of nicotine exposure in the brain that drive those functional changes. There's second messenger systems that we haven't really paid a whole lot of attention to. There's sort of promotion of the so-called addiction switch, or delta phos-B, inside these cells. There's the changes in certain connections and different network modeling that actually happens in the brain. These are all potential outcomes that can help us understand not just what the therapeutic targets are, but really where our levers for change really exist in the clinic. We're not going to talk about these devices in terms of their ability to help people stop using combustible cigarettes today. But I am going to say that as much as these products, the evolution of the environment is really a challenge for tobacco control, they also represent some level of opportunity for us, because all of these devices use a whole new slew of nicotine analogs. Right? There's synthetic nicotine, differences in the racemic mixture of nicotine, and how those differences actually affect brain function in the long run might be important to us. The different salts that different manufacturers use in their proprietary formulas have different pharmacokinetic absorption characteristics. They get moved from the outside compartment to the inside compartment in the brain at different rates. These are not just challenges to tobacco control. They're insights into mechanistic points of view that can help us in the clinic actually drill down and devise new, novel, imaginative solutions to managing those functional deficits that we talked about. And so when I started thinking a little bit about what I wanted to say today, I started by thinking about the environment when I was a third-year medical student. Fresh-faced, seeing patients for the first time, terrified, staying up all night, taking care of asthmatics who would get admitted to the hospital wheezing. My job, of course, was to make the wheeze go away. But what I was really doing was managing the underlying inflammation in the airways. In 1985, when I was walking around with my short white coat, I had 3 things that I could use. I had bronchodilators, I had steroids, and I had phosphodiesterase inhibitors. I would check levels all night long to make sure that I wasn't hurting people while I was trying to help them. 40 years later, I have 15 different classes of medications. Why? It's not just that I have more medicines to deal with asthma. It's because we've come to understand the underlying functional mechanisms. By understanding those functional mechanisms for what we see as wheeze, I can identify where my levers of change are, what my therapeutic targets might be. and then slowly begin to understand how different people, all presenting with some level of wheeze, might actually behave very differently. Suddenly, asthma is not one thing. Asthma is a multitude of things with a variety of different interventions. Similarly, ladies and gentlemen, in the clinic, smoking is not one thing. Smoking is not monolithic. Smoking is a variety of behaviors characterized by impulsivity, compulsivity, automaticity, characterized by the social, emotional consequences, the context within which this disorder represents itself. Why, on God's green earth, do we assume that nicotinic agonists or antidepressants are going to work for everybody the same way? We can't. We have to understand the role and the role that they play and how to tailor our interventions to meet the needs. We need different phenotypes. We need to understand the sort of biological phenotypes. We need to understand the behavioral phenotypes. And to get there, I would suggest that in our minds, we begin the process of teasing apart the goal of cessation from the problem of tobacco dependence. We all want cessation. There's no denying that. That is our goal. That's the Holy Grail. But the problem of tobacco dependence is multifaceted, it is complex, it is solvable if our eyes are focused on the problem and not exclusively on the Holy Grail goal. And so just a couple of examples that I could come up with for ways that we might proceed, innovate, imagine our way forward. I already mentioned phenotypic characterization. I know we saw some great work already going on about genotyping people. That's a great start. There's other biological phenotypes that are being reported, but there's behavioral phenotypes that we need to actually be able to identify. We need more complex behavioral outcomes, not just whether or not people put the cigarettes down for a month, but really impulsivity, compulsivity, automaticity. We need animal models that help us identify what our therapeutic targets are for those fundamental functional deficits. And then post-market analysis, kudos to the FDA and NIH for creating these post-marketing surveillance mechanisms, but we need that data out in the field for confirmation, for analysis, for modeling. That stuff's going to be important to understand root cause. We need patient advocacy groups. There's a patient advocacy group for virtually every disorder we know about, but not for tobacco use disorder. Why? Because we've taught people to feel like it's their fault that they're still smoking. That's not true. The problem that drives the behavior, not the behavior that drives the problem. We need emergency authorizations. There's one product out there that patients love and they respond to, and it's known to be effective. The nicotine inhaler was so good and so many people loved using it. It stopped being produced for unknown reasons. The manufacturer simply decided it's no longer available. We need an emergency authorization for nicotine inhaler to come from a different manufacturer, because every minute that that product is not available, people are dying. And then we need systems change. You heard about the action plan that Dr. King recommended. One of the issues was sort of systematic responsibility from the healthcare system side. Why doesn't every healthcare system in the world have a well-developed tobacco use disorder intervention team? Why? I guarantee every health system in the country has an asthma intervention team. There are 60 pulmonologists in my group, and all of them know how to take care of asthma. Thank you very much for allowing me to rant. This is my team. Without them, I'd be nowhere. I appreciate it, and I look forward to your questions. Thanks. Oh, I guess with that, yeah, thanks very much. I do appreciate it. I guess with that, I'm going to leave it up to Del Monte. Right button. Right button. Right button. Right arrow.
Right arrow. Okay. Thank you, Frank, and thank you, Monica, for having me here. to talk about this. You had the clinical perspective. Now I'm going to talk from a community perspective, the right button. Equity and cessation is what we're going to talk about. And we're going to talk about first some of the root causes of addiction. I am the executive director of the Center for Black Health and Equity. We do work in tobacco prevention. We do AIDS prevention. We do food insecurity. We do a lot of different types of things. Our organization started off as the National African American Tobacco Prevention Network. So we started in tobacco control, and we started to get African Americans at the table. All these policies were being passed about 20 years ago, but no black people were at the table. They were passing smoke-free this, smoke-free that, and they were doing it without any input from black people, so that's how come our organization started. So if we want to talk about some of the root causes of tobacco addiction, I'm going to tell you guys a little story. It's a real short story. And I just found this out about 2 weeks ago. At the end of this month, I'm going down to Staggsville, North Carolina. There is a group of my family descendants, and we're going to we're going to follow to a plantation in Staggsville, North Carolina, where my descendants came from that migrated from Staggsville, North Carolina to some part in Alabama. I think it was right outside of Mobile or something like that, because the owner of our plantation had bought a bunch of land and he was going to grow tobacco. So it's so ironic that I work in tobacco prevention now. My descendants were part of this great group of people that migrated to Alabama to grow tobacco. So that's interesting, because some of our root causes of tobacco addiction deals with what our experience with tobacco. African Americans and tobacco, slavery and tobacco, that is a part of our genes. That is part of who we are and part of what we've done. A lot of the black people who came to this country as slaves worked on tobacco farms. They picked tobacco, they grew tobacco. And so we went from picking tobacco to the tobacco industry targeting black people and us smoking tobacco. So that's a strange and ironic. But we have to understand, when we talk about the root causes of tobacco addiction, yes, being slaves was bad. Remaining slaves was really bad. But the country that we're in, the United States of America, they confirmed, they allowed this to happen to African Americans. They allowed this systemic racism to exist and to continue to exist until this time. So when we talk about why black people are smoking at these incredible rates, I have not heard mentioned yet systemic racism and the impact of systemic racism on how come African Americans are smoking. How come other populations that are smoking, the LGBT population, the Native American population, all the priority populations that we know that are smoking at these higher levels, why are they smoking at these higher levels? We haven't talked about the isms. We haven't addressed the isms. I got to say something here. We're putting a lot of money, hundreds of millions of dollars, into the science, into studying this and the regulations, but no one is putting hundreds of millions of dollars into dealing with the isms, the reasons that we're smoking in the first place. No one's putting money there. There are few cessation resources going toward the isms and dealing with the why we're smoking in the first place and trying to address that. I think about Alcoholics Anonymous. Not that I'm an alcoholic or nothing, but one of the things that they try and do is they try and remove you from the situations. That's one of the strategies is you don't need to be in a situation that might compel you to drink. Well, if we've got these isms, if systemic racism is one of the factors why these populations are smoking to begin with, what are we doing to remove the systemic racism? What are we doing to address the systemic racism? Why aren't we even talking about the systemic racism? That's the thing that really kind of blows my mind completely. Misinformation and disinformation on the harms of tobacco use. It's interesting, as Dr. Hooper talked about, I was part of the group that helped to get the federal government to propose a rule to ban the sale of mentholated tobacco products. I was very proud of that, very pleased that we were able to get that to happen. Now, that was 2 or 3 years ago. That was 2 or 3 years ago. So the actual rule itself was supposed to go and be signed into law last year. We were waiting for that to happen. However, for reasons unbeknownst to us, it was delayed. There was misinformation and disinformation that the black population, would be really upset if we banned mentholated tobacco products. So let's not ban mentholated tobacco products. Misinformation and disinformation. But the unfortunate part was that our administration listened to that misinformation and disinformation, and they've postponed implementing that rule to ban mentholated tobacco products. And so mentholated tobacco products are still on the shelf, even though all of the evidence, all of the data, all the science, everything that they need to ban it is there. They have that. They've postponed it. They put it off the shelf for misinformation and disinformation. I want to just talk about some solutions. We talked about the isms, the systemic racism, the sexism, the classism, all of these different isms. We have to really build our community capacity. We got to work within our communities in order to address this issue of cessation. And that was my timer saying that I'm over, but I just want to say a couple of things. I was at the T-Course meeting last week, and we were talking about the Tobacco Centers of Regulatory Science, and they were talking about a lot of these things and the science and the studying of this and the studying of that, and that was amazing to me. And I just thought about if I was in a community and working with community groups, all that information that they shared then, and probably even today, would go right over the heads of these community folks, these folks impacted by the problem, these folks who we're trying to address, we're trying to work with, we're trying to solve their issues, and it would go right over their head. To what degree, to what extent do we increase their capacity so that they can sit in the room, so they can be a part of the solution, that they can give you some suggestions, that you don't just come in with, this is what we're going to do for you, but they can tell you, this is what we need. This is what we need. This is what we need to address cessation, to address whatever it is. This is what we need, and we listen to them. And how do we make sure it's culturally appropriate? Again, Black people were slaves. We worked on the tobacco fields. We experienced systemic racism. So my cessation needs might be different than a person who didn't go through those experiences. So our cessation needs, they have to be culturally appropriate and tailored to the populations we're trying to address. And finally, we have to address it in community infrastructure. We have to really build up and put our resources, not that we don't need to do research, but that hundreds of millions of dollars need to go towards building up our communities so that our communities can then be stronger and they can withstand cessation. That's all I have. Thank you.
Sure. My name is Lonnie Nelson. I'm a descendant of the East Band Cherokee Indians. I'm a clinical psychologist by training, and everyone in my family of origin smoked cigarettes. I was a pack-a-day smoker for 25 years. It was just part of life. Grew up as a Native person in this country, and there's a natural association with tobacco. There's a very complicated history concerning Indians and tobacco products. So originally, it was thought of as a sacred medicine, one of the 4 sacred medicines, and a very powerful spirit that one did not toy with. You had to show it the appropriate respect. And this culture, this society, is now dealing with the repercussions of disrespecting that spirit. The traditional interactions with that plant were outlawed as part of the outlaw of traditional spiritual practices of Native people of this continent. And then the commercialization of the tobacco products specifically targeted that population for consumption. This has resulted in the highest prevalence of commercial tobacco smoking in any racial or ethnic group in the country. 6 times higher rates of developing smoking-related cancers, and disproportionately higher rates of respiratory and heart disease. In addition to this, American Indian and Alaska Native smokers are half as likely to quit smoking as other races and ethnicities. Commercial cigarette smoking now accounts for half of all of the deaths among American Indian and Alaska Native people in the U.S. I had 2 older brothers. One died at the age of 49, the age I am now, of complications related to smoking and diabetes. The other died at the age of 59, 10 years older than I am now, from the same reasons, multiple strokes as a result of years and years of smoking. 2 primary reasons for these downstream effects are the lack of efficacious interventions for our population and the fact that less than 1% of all full-scale smoking cessation randomized clinical trials have focused on American Indian and Alaska Native populations. That other fellow up there, his name is Jonathan Bricker, and he was the principal investigator on a NCI-funded study that looked at the effectiveness of an app that was based on acceptance and commitment therapy for smoking cessation. He pitted it against NCI's their own CBT-based developed app. And the acceptance and commitment-based app actually outperformed NCI's app. I had met Jonathan for the first time back in 2016 when he was giving a talk on acceptance and commitment therapy. And I went up to him after the talk and I said, that approach is going to work better for Native people than CBT will. And he asked me why that was, and I explained how, we're raised to have particular values that govern our actions, and we don't really sway from those necessarily all that much as long as we're paying attention, so on and so forth. Anyways, we wrote an R1 together and submitted it to NCI. It got like a 31, but wasn't funded. That same year, I got 3 other grants funded, so I just put it on the back burner. He went on to do his thing. He completed this nationally representative RCT of the ACT-based app. And remembering our conversation, he did a subgroup analysis looking to see if, in fact, it was differentially effective for the people who were participating in the study that happened to be Native. And it turned out it was. So we dusted off that old R01 application and put some fresh preliminary data in there and submitted it back to NCI. It got a perfect score. And we are now in the process of culturally adapting or refining that app to be more fitting for Native populations. So this is the original home screen for the I Can Quit app, which was the one that outperformed NCI's CBT-based app. We've been working with the Community Advisory Board and going through lots of different design features and pros and cons and taking polls and votes of Native people that were concerned with the topic and were willing to spend time with us. And this is what the new landing screen looks like for IndigiQuit, which is the new name for the new app that will be based on acceptance and commitment therapy, but will be specifically focused for Native populations. Any of you who might have any passing familiarity with Native populations probably sees a medicine wheel in there somewhere, but it's a little bit disguised, but there's definitely a nod to it, right? And that was that took 3 meetings to decide on with the Community Advisory Board. And the fact that the avatar down there at the bottom, it's the character that's going to guide the person through the steps of the app. Multiple conversations about what this person needed to look like, and it was decided that she needed to look like a clan mother, because that's the one person in Native communities that everybody does what they say. So I guess what I'm saying is that if we want to have effective interventions for Native populations to quit smoking, we have to be as crafty as the tobacco industry was. We have to make it as targeted for that population as the advertising campaigns that got all of my family members to start smoking. Just to dispel a myth, the same subgroup analysis that Jonathan conducted on the nationally representative sample, he also looked to see, well, did natives use pharmacotherapy, FDA-approved pharmacotherapy at any different rate from the rest of the study sample? And no, there were no significant differences for any of the FDA-approved pharmacotherapy options. Natives used them just as much as anybody else. So in summary, American Indian and Alaska Native communities have the highest prevalence of commercial tobacco smoking of any racial or ethnic group in the U.S. We experience dramatic health disparities resulting from this fact. There's a need for efficacious and accessible interventions. By the way, the app's going to be free. It's just going to be posted on the Fred Hutch website, and anybody can download it for free. But they need to be designed for this population. We've already begun work on these. We're also going to teach an AI-powered quit bot to talk like a res kid. That's in the NIMHD-supported U19 that we recently received. So that's going to be a really fun project as well. And finally, initial data suggests no significant difference in pharmacotherapy use for smoking cessation between Native participants and those of any other racial or ethnic groups in nationally representative RCT. Thank you.
Thank you very much. That was exactly what I was hoping for in this panel. I think we have important perspectives represented here, and so I want to thank you for traveling here to be here in person with us for this meeting. So we're talking about patient perspectives, we're talking about community perspectives, and the purpose of this meeting is how do we advance smoking cessation treatment, thinking about opportunities, especially in the pharmacotherapy, but also in the behavioral space. So I have a few questions that I want to ask you and to think about your responses, I guess, in the context of this meeting. So we'll start with Frank. There have been challenges with integrating smoking cessation services into clinical practice, although we know that clinicians have that valuable opportunity one-on-one to assist their patients with quitting smoking. And as you mentioned, that we have to address the solution and not put all of the onus on this, people who smoke. So would you discuss some of the key challenges around consistent tobacco cessation services in clinical practice, and then what might be some solutions for this issue?
Oh, wow, what a great question. What a great question. So I would say when thinking about the clinician role in this problem, they have valuable opportunity. They also have the values consistent with treating this problem, but they don't. And so I think the reason for that exists on three levels. The most superficial level is that I think we're the victim of our own success. For whatever it's been, 50 years or so, we've been marginalizing the behavior, basically trying to motivate people to change the behavior. And as a consequence, that perspective has made its way into the clinic. For lots of other problems, the sort of public health, educational perspective is very different than what needs to happen in the clinic. But for this problem, they overlap too much. What the impact of that is that clinicians find themselves in a situation where whether or not they do a good job based on an outcome that's really outside their locus of control. So they do a bunch of stuff, and then they hope that the patient in front of them will go ahead and change the behavior. And when that doesn't happen, there's a lot of frustration. The sort of deeper level underneath there is related. We've been able to identify something that we're calling culpability bias. Culpability bias is a sense that the responsibility for the problem is not just about ownership. I have a problem like asthma, I own that problem, I need to deal with it. But really, blame, culpability for the problem. If I continue smoking, it's really on me. And that culpability bias is implicit. People don't know it's framing their decision-making, but it goes a long way towards minimizing the willingness to invest in ongoing therapeutic interventions that may take a really long time to actually come to fruition.
Thank you. I appreciate that. Let me ask one more question for you, and then I'll move to the other panelists. But just thinking about this, so in my experience working in clinical settings, clinicians have seemed more comfortable prescribing nicotine replacement therapies to their patients to help them with this, but some have been hesitant to prescribe, like varenicline. And what have you noticed that, and what would be some of the key considerations that we have to think about now to help bring clinicians along with us as we develop new therapies for smoking cessation, and so that once they're developed and available, we can have the clinicians on our side?
Yeah. This, I notice this problem. It's everywhere. You're exactly right. It drives me crazy. And I do think that it's sort of a derivative of a sort of 1980s mentality that this problem is really sort of an empty bucket problem, right? As nicotine levels in the body go down, the bucket empties, and when it gets to a certain level, the patient in front of us needs to replace that nicotine level in order not to withdraw, quote unquote. And so I think that that's a much more fundamental, long-standing model, whereas varenicline is generally viewed as messing with the brain in a lot of different ways. And the brain's a black box to a pulmonologist, to a cardiologist, to a surgeon, and so they would prefer not to mess with it if they don't have to. I think in the future, what we'll see is that we're going to recognize that all of these medications have a variety of different mechanisms of action, but what they're really there to do is to mess up the patterns that the brain is utilizing in order to drive this continued behavior. It's messing up the impulsivity, compulsivity. It's changing something that is sort of reflexive and automatic into something that is much more cognitive and controllable. And I think once people start to recognize that the issue is different, they'll be more likely to use more effective medications.
Thank you for that. So, Monty, let me move over to you with a question. You know, you made a lot of important points, I think, about the need for interventions to be culturally competent and other important considerations. What types of smoking cessation approaches or interventions tend to fare well in terms of acceptability and outcomes in the populations that you serve? And then on the other side of that, what approaches have been viewed as less favorable?
Well, in terms of approaches that have been favorable, I think about the Pathways to Freedom. There was the DVD that was done, and there was also a Pathways to Freedom pamphlet. They looked at the smoking experience in the Black community, and they set it up to where you could actually have a smaller group at a church or at a function or a community center, and you can go through the counseling with other members and other peers, and you can talk about shared experiences that you had that may have led to you smoking. So that was very important. It has been very effective because, again, this community, the smaller group, they had these shared experiences. And so they were able to quit and abstain from tobacco together. That's been very effective. What's not as effective, you know, Black people call the quit line. They call the quit line more than other folks. They call the quit line, but if you're calling the quit line and a person can't really relate to you from that quit line, they don't understand your experiences and what you went through and what you're experiencing even now, it's not going to be as effective. So Black people call the quit line, but they don't stay quit after calling the quit line. So we call it often, but we don't stay quit.
Got it. Thank you. So let me ask the same question to you, Lonnie, in terms of the kinds of smoking cessation approaches that you find to be acceptable, feasible, and lead to positive outcomes in the Native people that you work with, and then the types of approaches that have not been going so well.
Yeah, so cognitive behavioral therapy is a pretty Western and cerebral approach and is not really all that well accepted by most of my relatives. Things that deal much more with the direct experience, like acceptance and commitment-based therapies. You know, I don't know how familiar you are with that approach, but the acceptance part is an explicit willingness to endure the discomfort of withdrawal because of your commitment to a particular value that is your reason for making this decision, right? And so something that's very much more straightforward and head-on seems to resonate a lot more with the Native population than these more cerebral, theoretical, abstract approaches. Nicotine replacement is very popular. It's, like I said, the uptake of that pharmacotherapy was about the same as it is in all the other races in the study, about 34%. And of course, electronic nicotine delivery systems are rising in popularity in Native communities just as much as every other category. So those also are pretty popular for getting people to stop smoking commercial tobacco.
Thank you. Delmonte, I have one more question for you. When we think about, this meeting, what advice would you have for those in the room, those watching, investigators, clinicians, others who have interest in developing new treatments, particularly pharmacotherapies, things that might end up being like a drug therapy? What advice or what do you think we would need to really consider as we want to engage these communities that experience this disproportionately high burden in the case of both of the communities that you work with? What are key things that we need to keep in mind?
Engagement of a community in your studies does not simply mean giving $50 per participant to study them and to say this is what happened with them and this is what happened. That's not engagement. $50 a study is not engagement. Engagement is investing in those communities and seeing what works and providing resources to that community to be sustained even after you're gone. That's the investment part. And so I would say if you want to engage communities, then engage communities. But come with your purse strings. Come with some dollars to make these communities better. Don't just come with $50 to have a study participant.
I would say the key to engagement is authenticity, transparency, being present in your interactions with the community members that you intend to serve. It's being willing to have an honest conversation and to try and get genuine agreement on goals with the community that you mean to serve, and work together and honestly listen to what they tell you will work for them and then do it.
I have one more question, and any of you could answer this question. So one major challenge that we are facing in science and in medical, in healthcare, is the issue around distrust. And we know that distrust has been there for a long time, but has been elevated in the past few years. So if we're thinking about new therapies and something that might be something that you ingest or put in your body, and people are talking about, I need to know what's going in my body in terms of medication, what are, from the patient's perspective and community perspective, some of the challenges that you might anticipate around managing the distrust for a new drug therapy for smoking cessation, even with the high motivation to quit that most people have?
Yeah, it's a really sticky issue, particularly around tobacco use, because in the clinic, even when patients trust me 100%, as soon as they leave, they're sort of bathed in a lot of messages that can run counter to what we talked about in the clinic and can be, at minimum, confusing, but at maximum can actually kind of throw a monkey wrench into the relationship. And so what I always teach the trainees is basically 2 ingredients to engaging. Trust in this problem is of significant importance. And it's really about validation. So validating not the behavior, but the person who displays the behavior, why this behavior goes on, why it makes them feel the way they do, why they sometimes feel stuck. And then empathy, really engaging in open-ended conversation about the person's backstory, so to speak, exploring what's going on at home, what's going on in your community, what's going on at work, who are you, what have you experienced in the past. The more clinicians ask questions around this problem, the more likely they are to engender trust, as opposed to when you come right out of the box and say, you know what you need a patch. That just doesn't work.
I would also say that when you're considering the development of new pharmacotherapies, think about how you're going to explain how the drug works. I mean, you see a natural experiment in the stats that I put up there. 30 to 35% uptake for NRT, nicotine replacement therapy. It makes sense. They know they're addicted to nicotine. If you're going to give them an alternate source of nicotine, oh, well, yeah, okay. 8-9% uptake for varenicline? They don't understand how it works. Why is that supposed to help me when I'm addicted to nicotine, right? You can't explain the pathway. If you can explain the causal pathway of how the drug should help, the uptake will be much greater.
Yeah, and let me just say that some populations, Black populations in particular, they have a reason to distrust the government. The government has given them a reason not to trust them. And if I think about, a relationship, just a man and a woman or whatever, in a relationship, if you have given a person a reason not to trust you, in order to get them to trust you, you have to give them reasons to trust you. And so the reasons to trust you is what has to happen first. You have to think about, how are we going to get this community to trust us? How are we going to give them a reason? How are we going to engage with them? How are we going to listen to them? How are we going to be authentic in providing them with the resources that they need so they can trust us again? Because, again, they don't trust us, and we've given them that reason.
Well, thank you very much. We are at time. Please join me in thanking our wonderful panel for this great discussion, and I thank you personally for attending.
All right, I want to thank everyone for joining us for our NIH session on promising new targets and interventions. My name is Evan Herman. I'm a program officer in the Division of Therapeutics and Medical Consequences at NIDA, and I'm joined by my colleague, Dr. Neil Friedman, who's in the Tobacco Control Branch at National Cancer Institute. And today we have 4 invited speakers who are doing exemplar work in this area. I'd like to start by introducing our first speaker, Dr. Luba Yamin. Dr. Yamin is an associate professor of psychiatry and behavioral sciences at University of Texas Health Houston, and she'll be presenting on GLP-1 agonists and smoking cessation.
Hi, everyone. Thank you, Evan, for the introduction. I'm certainly excited to speak about my work today, which over the last number of years has been on the intersection of smoking cessation and post-cessation weight management. And I am affiliated with the University of Texas Health Science Center, and within this Health Science Center, I am with the Center for Neurobehavioral Research and Addiction, where we do a variety of clinical studies focused on various substances of abuse, and my personal interest, of course, is smoking cessation. I do have a couple of disclosures to report. I have, sorry about that. I have research funding from Novo Nordisk, and I have also served as a consultant for Eli Lilly. A brief outline here. So today we'll talk about the relationships between cigarette smoking, smoking cessation, and body weight. And from there, we'll dive into the potential utility of GLP-1 receptor agonists for facilitating smoking cessation and at the same time helping people manage weight as part of the journey. And then we'll talk about directions for future research. Smoking cessation is indeed associated with a variety of health benefits, reducing the risk of cardiovascular disease and pulmonary disease and multiple types of cancer. Unfortunately, the majority of people who quit smoking gain weight, and this weight gain can be substantial. It varies among people, but somewhere meta-analysis show us between 5 and 15 pounds at the end of 12 months. And now there is a non-trivial proportion that actually gains more than 20 pounds. And on the right-hand side, I have a figure here that comes from a smoking cessation study. And the top line, the green line, shows people that were continuously abstinent. And you can see that this weight gain post-cessation happens pretty rapidly, with the majority of it occurring during the first 6 months after quitting smoking. And then it continues to rise, but it tapers down somewhat. So to understand why people gain weight post-cessation, it's kind of easier to look the other way. So what does cigarette smoking do to body weight? So nicotine binds to nicotinic receptors in the brain, which leads to the release of a variety of peptides and neurotransmitters, which act both systemically and centrally. So systemically, we see the release of catecholamines that act directly on the adipose tissue, which leads to increased energy expenditure, increased metabolism. In the brain, there is a whole variety of neurotransmitters and peptides. We see dopamine here, of course, we all know associated with reward. But there is also a bunch of other neurotransmitters that collectively lead to the decrease in appetite and increase in satiety. And so this subsequently leads to decreased intake of food and a decreased body weight. And then, of course, we know that smoking also can serve as a behavioral alternative to eating. So now that we're taking nicotine away, we're taking away the appetite-suppressing effects of nicotine. So naturally, people feel hungrier. Now, that reward is also removed, the reward that people used to receive from smoking. So now they're starting to seek reward from food. And obviously, they're seeking food that is high in reward, so highly palatable kind of food, high sugar items, high fat items. And so that naturally leads to the increase in body weight. Now, there is also decrease in energy expenditure. Having said that, research shows us that the majority of the weight that occurs post-cessation is mediated by the increased intake of highly palatable foods. So post-cessation weight gain is not just a cosmetic issue. The problem with it is that it is a very common barrier for people to making a quit attempt. In fact, concern about post-cessation weight gain is practically endemic among women. However, men also express concern. Now, among people who do quit smoking, it is not uncommon to relax back into smoking once they begin accumulating weight post-cessation. And then, of course, there is an increased risk of developing weight-related metabolic diseases, including new onset type 2 diabetes. In fact, this meta-analysis shows us that the risk of developing new onset type 2 diabetes is 54% higher for individuals who quit in the past 5 years, and then it gradually tapers down. So this brings me to glucagon-like peptide 1. We currently don't have any treatments that address the problem of helping people with quitting smoking, and preventing this post-cessation weight gain. And so this is a good time to talk about GLP-1, which is an incretin hormone and satiation factor that is mostly known for its glucoregulatory and other metabolic effects. We know about increased secretion of insulin, decreased secretion of glucagon, and decrease in appetite. So GLP-1 is produced both peripherally and centrally. And importantly, to this discussion, centrally, GLP-1 receptors are expressed in the areas which are associated with hedonic and reward-related behaviors. This, unfortunately, is pretty small. However, I would like to say that my personal journey with GLP-1 agonists started about a decade ago in the context of a primary care setting where I saw a large proportion of individuals with type 2 diabetes. And GLP-1 agonists would be one of the medications that I would prescribe for the treatment of type 2 diabetes. And then I began hearing incidental reports of people saying that either their cravings for cigarettes have reduced or they quit smoking completely. And when I would ask them what happened, I would hear a variety of responses. Oftentimes people say, I just don't want to smoke anymore, or, when I smoke, cigarettes kind of taste funny at this point of time, kind of stale. And so this was intriguing, of course, as those reports were accumulating. And so after a while, I began looking for any kind of validation in the literature. And at the time, only the first study shown here was published. However, since then, a number of preclinical studies have published which collectively tell us that GLP-1 agonists decrease expression of conditioned place preference, decrease intake of nicotine in animal models, as well as decrease nicotine-induced dopamine release in the nucleus accumbens. And so when we combine this together, it appears that in the context of smoking cessation, GLP-1 agonists may decrease reward, potentially increase nicotine aversion. Having said that, there are probably other mechanisms involved. We are still trying to narrow down on those. For example, in other contexts, it has been shown that GLP-1 agonists may improve cognition as well as alleviate depressive-like symptoms. So taking that together, potentially GLP-1 agonists can help with improving motivation during nicotine withdrawal as well as decrease stress-induced nicotine intake. Again, probably there is several mechanisms implicated in this area, and they are not mutually exclusive mechanisms. So we know that GLP-1 agonists are very potent medications with respect to reducing body weight, as well as reducing hemoglobin A1C in patients with type 2 diabetes. This here is a STEP program of clinical trials in obesity with semaglutide. One study that is worth bringing up here is something that was published fairly recently. And this study is also a preclinical study, but that was conducted in the context of nicotine withdrawal. And so in that study, relaglutide decreased nicotine withdrawal-induced hyperphagia. So this is quite relevant to the current discussion. So going back to a number of years ago, given those incidental reports that I was seeing in my clinical practice, along with some initial preclinical work, let me design a first pilot trial of exenatide, which was the first extended release GLP-1 available at the time. So in that pilot trial, we enrolled patients who had prediabetes or body mass index of 25 or above. We had 2 recruitment sites. Patients were randomized in a 1-to-1 fashion to receive a clinically available dose of extended release exenatide, 2 milligrams once a week, subcutaneously for 6 weeks. Regardless of the randomization, everybody received a platform of nicotine patches and 21 milligrams in smoking cessation counseling. And our primary outcome was 7-day point prevalence abstinence, self-reported, biochemically verified by expired breath CO levels, and our secondary outcome was changes in post-cessation body weight. This is a little bit complicated, but I will mention briefly that we used a Bayesian approach for our data analysis, which was appropriate for this early phase study. What Bayesian does is it allows to ascertain probability of an alternative hypothesis. So in this instance, we are not betting on the null hypothesis that the treatment does not have an effect, but just the opposite. What is the probability of the treatment having an effect? And we a priori defined 75% or above as sufficient evidence that the effect existed. So our participants were 51 years of age on average. We had a predominance of males and black individuals, black males, I should say. On average, they smoked 18 cigarettes per day for 27 years. On average, nicotine dependence score was 6.2. So what did we see at the end of the treatment? With respect to abstinence, exenatide demonstrated greater odds of abstinence versus placebo, and posterior probability for that was 96.5%. Looking at the raw numbers, 46% in the exenatide group were abstinent as opposed to 26.8% in the placebo group. Moving to weight, exenatide demonstrated lower post-cessation body weight versus placebo with a posterior probability of 97%. When we look at raw values, people in the exenatide group actually lost a tiny little weight, whereas expectedly, people in the placebo group gained some weight at the end of treatment. Now, these results, we just started looking at the potential moderators of abstinence. These results have not been published yet, but we looked at demographic characteristics and smoking-related factors, and the factors that are shown here did not show moderation effect. However, we did see two factors that did show moderation. One of them was heaviness of smoking, specifically cigarettes per day. So to be clear, exenatide was effective regardless of how many cigarettes per day people smoked, but in people who smoked more, exenatide had stronger effects. And then we also saw some variability of nicotinic receptor that showed some moderation effect as well. With respect to safety, 4 participants in the exenatide group experienced transient injection site nodules, which is consistent with the side effects of exenatide. We did not have any reports of hypoglycemia. We did not have severe injection site reaction. We didn't have any other side effects. Importantly, we did not have any treatment discontinuations. So what next? Obviously, these results were very exciting, very promising, but of course very early. So I should say that the way we would interpret these Bayesian analysis would be that compared to NRT alone, exenatide plus NRT conferred more than 9 in 10 chances of producing abstinence and more than 9 in 10 chances of decreasing post-cessation weight gain. It was safe, it was tolerated, but as I said, it was early and preliminary and definitely needed replication and extension in larger trials. And this is exactly what we're doing right now at our Center for Neurobehavioral Research and Addiction. We have 2 large clinical trials going. One of the trials is a NIDA-supported R01 trial, which is largely similar to our pilot trial, except that it is done on a much bigger scale. So in this instance, it is a larger sample size. We're using longer treatment duration. It's 14-week treatment duration. We're also looking at the sustainability of effects. And so after the treatment is discontinued, 3 months later, we bring the participants back to reassess their smoking status, as well as body weight. And additionally, we're trying to tap into the potential mechanisms of how the medication works in this context by assessing neurobehavioral responses to nicotine-related and food-related cues. The second large trial that we're doing is a trial of semaglutide, and in this instance, we're looking at a variety of metabolic outcomes in the context of smoking cessation. So we're not only looking at body weight, but we're looking at changes in body composition, changes in ingestive behavior, satiety hormones, among other things. So what other clinical studies have been done in this field? There was a study conducted in Denmark in which they evaluated 12 weeks of dulaglutide treatment. In this instance, it was adjunctive varenicline and behavioral counseling. In that study, they did not find differences between dulaglutide and placebo. I should mention that abstinence rates were very high in both arms, 63% with dulaglutide, 65% with placebo. They did find significant differences in body weight, and that dulaglutide decreased body weight compared to placebo. A couple of other clinical studies can be found on clinicaltrials.gov. The results have not been published yet. I also would like to mention two relatively recently published analyses of electronic health records. In these instances, the researchers looked at individuals. In one case, it was people with type 2 diabetes. In the other case, it was people with type 2 diabetes and tobacco use disorder, documented tobacco use disorder. And so individuals were propensity matched, people who received semaglutide versus individuals that received other anti-diabetes medications. And the outcomes were more proximal outcomes because these were electronic health records, and so researchers looked at things like nicotine misuse, medical encounters for tobacco use disorder, smoking cessation medication prescription, as well as counseling. And so in all instances, semaglutide outperformed all other diabetes medications. I would like to conclude with future directions for this exciting line of research. It has been said today, probably in every presentation, about the importance of identifying patient subgroups that would most likely benefit from these treatments. We know very well that medications just don't work the same for everybody, and chances are GLP-1 agonists will not be an exception. Even when we look at GLP-1 agonists in the context of weight reduction, there are some people that lose a substantial amount of weight and some people that don't receive a whole lot of benefit from GLP-1 agonists. And chances are this will be the case also in terms of treating tobacco use disorder. So learning what those patient subgroups is going to be absolutely critical. Another issue is dosing. So if you take newer GLP-1 agonists, for example, I mentioned we're conducting the study of semaglutide. We're using the dose that is approved for the treatment of obesity, the 2.4 milligram dose. That dose requires 16 weeks of dose escalation. This is pretty lengthy and may cause some logistical challenges, especially in terms of somebody who is seeking treatment for smoking cessation. However, the question becomes, do we really need to go up to this high of a dose? Is it possible that we may see the effects at lower doses? And there are, in fact, some indications from preclinical literature that we may see behavioral effects of GLP-1 agonists at doses that are lower than those required to induce metabolic effects. The sustainability of effects, I mentioned something that we're currently looking in our study, both of our studies. Safety considerations. So we are enrolling individuals who are overweight or obese at baseline. What about people that are normal weight at baseline or people that are underweight? Are there any safety considerations involved there? And then we have been conducting research using GLP-1 agonists as adjunct to other smoking cessation medications. However, looking at GLP-1 agonists as monotherapy in the context of smoking cessation would be another direction to take. And then last but not least, we really need to understand how these medications work in terms of their mechanisms. That will allow us to potentially combine them with other medications with different mechanisms of action or behavioral therapies or both to ultimately develop that package wherein people receive the most benefit in terms of smoking abstinence as well as reduction in post-cessation body weight gain. I would like to acknowledge my collaborators. This has been a huge effort of researchers and clinicians from multiple disciplines and multiple institutions, including my institution, UT Health Houston, as well as MD Anderson Cancer Center, UT Del Med, and Baylor College of Medicine. The funding for my pilot study was provided by the Korea Development Award. The exenatide trial is funded by NIDA, and the semaglutide trial is funded by Novo Nordisk. And that is it. Thank you very much.
All right, thank you very much, Dr. Yamin, for that excellent presentation. Our next speaker is Dr. Mark George. Dr. George is a distinguished professor of psychiatry, radiology, neurology, and the director of the Brain Stimulation Division at Medical University of South Carolina. Dr. George will be presenting on brain stimulation methods for smoking cessation.
Thank you all. That was a wonderful talk. I'm honored to follow. It's a pleasure for me to be here. It's a homecoming of sorts for me. I spent 5 wonderful years over in Bethesda at the intramural NIMH campus, and my daughter was born in Silver Spring Hospital right around the corner here. And I'm really excited to talk with you about this topic. Been listening all morning, not to be insulted, but how many times we've talked about pharmacology and behavior, and very few times we've talked about brain stimulation. But it is increasingly going to be part of the future of many therapeutics, and especially, I think, in smoking cessation. I'm a psychiatrist and neurologist by training. I'm a brain imaging specialist, and I work at the VA hospital. I'm also editor-in-chief of a journal called Brain Stimulation, which has been running now for 17 years, and it allows me every day to look at the advances around the planet in this field and look into the future. I don't have any major disclosures. I'm going to talk about several products, one of which is by a company, BrainSway, and I've helped them out in the past. I don't invest in any of these devices because I enjoy being a scientist and a critic. Here is why the brain revolution, the brain stimulation revolution is going on. This is a classic cartoon that we see, a neuron and a synapse, and this is where, with the depolarization, you get release of compounds into the synaptic membrane, which travel across, and they do all of their magic. And I am an allopath. I take medicines. I write prescriptions. I think medications are wonderful. They're life-saving. They probably have saved my life at least a couple of times. So I'm not against medications, but I grew up as a neuropsychopharmacologist, and what I rediscovered was what I learned in medical school, and that is the brain is actually an electrochemical organ, and electricity is the currency of the brain. All of synaptic pharmacology just serves as a rheostat to decide whether electrical information flows from one neuron to the next. It's just drugs are switches at the membrane that really affect the most important thing, and that is the electricity in the brain. My thoughts, my actions right now with you are all electricity in different regions communicating. That's what we have to remember. And when we remember that, if we can put electricity straight into the brain, oh my gosh, the things that we can do, and we can do it focally and in ways we could never do with pharmacology. That's the sci-fi pitch of the future, but it's actually happening today. Let me show you how it's happening in other areas, and then we'll come back to smoking. So this is a list of the FDA-approved treatments, brain stimulation techniques. We have the grandmother of the field, electroconvulsive therapy, which is still a lifesaver. We have RTMS, transcranial magnetic stimulation, which I'll come back to. We can stick wires straight into the brain, and that is transforming the treatment of several diseases like epilepsy or Parkinson's disease. We can stimulate the vagus nerve in the neck or through the ear or other cranial nerves, and those have remarkable therapeutic properties as well. Now, and then there are several not yet FDA-approved approaches that are the hoofbeats on the horizon we can see, and one of them is this technology of transcranial pulsed ultrasound. Who would have thought that we could take ultrasound like we look at a baby in the womb, and if we pulse it, it will cause a human nerve cell to depolarize. And we've actually figured out ways of getting through the skull. The bone is usually a problem with ultrasound, but we've been able to figure that out. And so that's the new kid on the block. And we have less invasive things, transcranial direct current stimulation, which aren't yet FDA approved, but I think eventually will be. So, and this area is just exploding. These are a PubMed of the searches over the years. And with TMS, DBS, ECT, VNS, and then TFUS is the acronym for ultrasound. So let me go back to TMS, which is the one I started my career with over in Bethesda. We take an electrical current and put it through a coil, and the electricity stays in the coil, but the magnetic field that's produced can transmit unimpeded through space and time, and if we put it on the head, it can go through the skin and the skull, and it can induce a electrical current in the brain tissue directly underneath it. What a cool trick. We can stimulate the brain in an awake and alert person right here, no anesthesia, no nothing. It doesn't, right now with current coils, it just goes to the surface of the brain, but we know that the brain's massively connected, and we can stimulate a cortical region that is a cortical window, and wherever that region goes, we can then begin to get into the circuit. So this is an example of what it looks like applied on the head. So it's really electrode-less electrical stimulation. We're really electrically stimulating the brain. We're just using the magnetic field as this trick to get into the brain. So it's now used as a treatment. It's for depression, was the first FDA indication. It's non-invasive. It's largely side effect free. It can be applied focally in the brain. When we stimulate the brain and somebody leaves, they have nothing in them. There's no drug-drug interactions. There's nothing. We're just using brain electrophysiology. And very interestingly, if we stimulate at different pulse patterns, high frequency is excitatory, low frequency is inhibitory. We can actually push or pull different parts of the brain, inhibit or temporarily excite. And so in 2008, after a lot of work, it was FDA approved for acute depression. We did one treatment a day for 6 weeks, 30 weekday sessions. So the way we rolled it out was quite inefficient in terms of the amount of time that it takes for someone to come to the clinic. Now we have accelerated protocols. We figured out we can do the same thing in just a week with at least as good of effect, which is changing how we deliver TMS for depression. It's also cleared for obsessive compulsive disorder and smoking cessation. So where would we stimulate to get at smoking? And so this is a diagram. Really, there are two areas that you might go after. One would be kind of an executive control region, which would start with the dorsolateral prefrontal cortex, and you'd want to grow or improve executive control. No, I don't want to smoke. These are the negative consequences of smoking. I'm going to stop. I owe it to my kids. Or you could go after the craving. Boy, I really want to smoke. So you could try to inhibit that craving urge, or you could grow the executive. And so we then have applied TMS as a way to push or pull the circuit that way. That's a picture of Dr. Colleen Hanlon and doing TMS on me years ago. And so that's kind of a rough summary of all the discovery work that's been done. If you look at stimulating over the prefrontal cortex, which is where we largely stimulate for depression, people have tried TMS for almost everything you could think about. And here's a list of the effect sizes on craving, depression, pain, or by diagnosis. And then in this nice look at what the effect sizes are, you can see that in orange there, the effect on craving, the immediate effect on craving of dorsolateral prefrontal stimulation has the largest effect size of anything in depression. I mean, with TMS, and bigger than depression. So my colleague, Dr. Xingbao Li, and others in Charleston started by looking at what's the effect of a single session of prefrontal stimulation, high frequency, 10 hertz, to reduce cigarette craving in smokers. And you can see here in Dr. Lee's work very early, back in 2013, that if you looked at craving with either sham or TMS, both before and then after, that the TMS, just 20 minutes of TMS here, knocks down your craving for cigarettes. So a really interesting discovery. At the same time, my colleagues in Israel, Bumi Zangan, Abraham Zangan, who was a NIDA fellow just around the corner, and he got fascinated by building better TMS coils. And there's a picture of them here, and he designed these coils in his evenings and nights while he was a NIDA fellow. The patents for those actually come back to the NIH, but he went back to Israel and started a company called BrainSway, and they did this trial in 115 treatment-seeking smokers, and they wanted to know whether they should excite or inhibit the brain and whether they should pair it or not with craving. And so they, in a pretty complicated study, gave people either sham or 1 hertz stimulation or 10 hertz stimulation, with or without actually provoking craving immediately before the treatment. And what they found was that in terms of the number of cigarettes smoked per day, that 10 hertz, so excitatory stimulation, paired with behavioral activation of craving, really knocked down the number of cigarettes per day and also increased quit rates at some point in the future. So with that kind of discovery of the frequency needed and the pairing with behavior, they then ran that into a large international FDA pivotal study, and I paired with him to actually do it. And this was a labor of love at 12 sites in the U.S., 2 in Israel. It took almost a decade to do this study. There were lots of challenges along the way, but we did it, and we had self-reports and urinary cotinine samples, and there were 2 groups, 10 hertz sham, double-blind. And we did a smoking provocation right before each and every treatment. In Israel, in that early study, the smoking provocation was the PI, Dr. Danur Klein, who was a smoker, would sit down and smoke a cigarette right in front of the people. When Bumi came to me and says, we need to provoke it, I said, you cannot smoke anywhere near a U.S. health hospital. So we actually ended up having a paradigm where people looked at pictures of people smoking. They actually had a cigarette. They would touch it, smell it, they'd flick a lighter, but they weren't able to do anything else. So people would come in after, and they would have the symptom provocation of 5 minutes. They'd rate their craving. Then they'd get the TMS or sham for about 18 minutes, and then they'd get a motivational script to try to help them quit, CBT-like. The treatments were for 3 weeks, and then with one treatment a week for 3 weeks following, so about 15 acute treatments and then a few tapers. And they would come in, they would set a quit date at the early part of the 2nd week. And so they'd come in and try to quit. We looked at their consumption and craving, and we did a visual analog craving scale at the beginning, during, and the end of each session, which I think is important in terms of mechanism. So these are smokers like you saw in some of the earlier studies, 27 years, average smoking number of quit attempts were very high. These were chronic smokers. So did it work? This is a complicated slide, I apologize, because we have the intent to treat analysis and the completer analysis. Either way, the active is much more active in terms of the short-term follow-ups, just the 3 weeks, or the longer-term follow-ups out to 4 months. And you can see that the quit rates are pretty high, and the continuous abstinence quit rates are very high compared to sham. So a slam dunk in terms of efficacy, and the consumption went down, the craving went down. The thing that's very interesting is if we looked at the change during an individual session on the craving rating, that actually correlated or maybe even predicted whether people were going to be successful quitters or not. So I do think that we were, with this treatment, knocking down craving in an important way. So it was safe, there were very few adverse events, no seizures, and no change in weight for some reason I don't quite understand. TMS in general is not it doesn't have a weight effect. So when we look across at how this compares to all the things you've heard about this morning, and this is based on a study, Halpern in the New England Journal, where we got these effect sizes, it looked as if this approach was just as good as the other available things. So I was quite hopeful that now that we've gotten a good study, Class 1 evidence, FDA approval, that this was going to sweep through and change smoking in the country. And I've been very disappointed. So it was FDA cleared in 2020, but there has been limited clinical uptake to date. It's not reimbursed by insurance. The TMS manufacturer is not a big company. There's not a lot of marketing available. When I ask them what's going on, they say, well, who provides this? For depression, there's psychiatrists. Who actually is in charge of smokers? It's not a disease. It's not owned by a group of people who care for these patients. And in this study, we did it, and then we walked away. And there's we didn't have the money to actually do a maintenance or a relapse. If people go, do we need a tune-up or not? So I'm interested in this meeting because perhaps the best way to use this technology would be in special populations like cancer centers or COPD places or patients' cardiovascular MIs. And with some effects, we're actually starting to introduce the technology in those special areas where you have people who are still addicted, who are having negative consequences like a cancer. NIDA has helped out with a very important study by Dr. Lee and Dr. Herman, whose name I misspelled here. I apologize. I replaced these slides, but they didn't get replaced. Anyway, we went, Dr. Lee did a head-to-head of inhibitory craving versus excitatory dorsolateral, and in this study that's under review, he found that the dorsolateral prefrontal was a clear winner in terms of the reduction in cigarette smoking, and so he's now funded to do a UH3 study where he's comparing TMS here to sham, and our industry partner there is a company called MagVenture. That coil is a figure-8 coil, and there are about 6 manufacturers of those coils. My hope is if this is a positive trial and MagVenture gets FDA approval, there will likely be other TMS coil manufacturers who will apply for FDA approval, and we'd have at least broader availability of the technology in the population. So where else can brain stimulation help? We could go with deep brain stimulation of specific regions. However, that's awfully invasive. Ultrasound is promising, but it's still in early days. There's work where you use ultrasound or other ways to release a pharmacology, a compound in a specific spot, so it's not brain stimulation or medications. It's a combination of a delivery method. Those are exciting areas. But I think this work by Brainsway and Dr. Lee shows us that you can manipulate these circuits while people are craving, and you can have long-term durable effects on smoking. And so that's the key, I think, in terms of future. And if we had some form of a less invasive home-use device that could stimulate that circuit for smokers, then we might be able to have something that could really have a public health effect. I'm searching around for those. There are some devices that are working at home with depression, and so perhaps that is a way that will work forward. So with that, I want to thank you for letting me talk about this very important area.
Thank you, Dr. George, for that electrifying presentation on brain stimulation. Our third speaker is Dr. Roger Villadaga. Dr. Villadaga is an associate professor in implementation science at Wake Forest School of Medicine, and today he'll be talking about digital therapeutics for smoking cessation in adults with serious mental illness.
Right, it's such an honor to be here. I'm really glad to talk about our work. This work has been funded by NIDA over a course of over 10 years. So most of you know that tobacco use disorder among people with psychiatric illness remains a significant public health problem. We're talking about a very small portion of the U.S. population that consumes a very large amount of cigarettes. And this, of course, has significant health consequences, not only in the communities where these individuals live, their families and so on, but also has a significant economic cost. So in light of that, our goal was to develop a scalable solution to this problem. And so we envisioned the possibility of developing a digital therapeutic to address this health problem in a way that was more scalable and more accessible for a larger portion of this population. So this is a development timeline for the Learn to Quit digital therapeutic. It started with the K99 that funded the ideation and design of the intervention. We conducted early phase research through the R00 phase. Then we got funding for the pivotal trial. We received a supplement to turn that pivotal trial into a decentralized clinical trial, and then we finally received funding for an ancillary study to examine barriers to implementation of the digital therapeutic in this population. So, starting from the beginning, we conducted some user-centered design research, which we published in Nicotine Replacement Therapy and in Jamir Serious Games a few years ago. We started with doing formative user-centered design research of an existing digital therapeutic developed at the National Cancer Institute. It's called NCI QuitPal. I don't think it's any more available right now. But what we got from this initial formative work was a checklist of features that, based on our work with this patient population, seem to be critical to include in the design of this software digital therapeutic. With this work, sorry for all those little animations. This led to the next phase of development, which was ideating, sketching, prototyping the specific flows and routines that this digital device should entail, an overall vision of what this therapeutic should be doing. We sketched specific screens and wireframes, and then we came up with a number of personas that would guide the development process, both for software engineers, the team that we partner with, and also for us. And this led to working with an actual software company that we partnered with in Seattle while I was still working there. And that team took this initial concept or specs document into a concrete software system. And then this is some of the components of the Learn to Quit app in its initial version. So essentially, we used acceptance and commitment therapy for smoking cessation. I was part of the development of that intervention. My mentor was a developer of that treatment. But we integrated that with U.S. clinical practice guidelines. We used very simple layer structure for this device and using big buttons. And then ultimately what we wanted to was convey the experience of learn, practice, and play to quit smoking. And for that purpose, we used a number of animations, a lot of images, and a combination of storytelling combined and packed together with U.S. evidence-based treatment recommendations. Before we jumped into testing this digital therapeutic in a pilot trial, we actually conducted some formative research in the form of A/B phase single case designs. So I'm just going to very quickly go into what we learned from this. We learned that on average, participants that used during the Learn to Quit phase, they actually had very high levels of system usability. This is a metric that is used in the software development industry to kind of evaluate how easy and how usable it is, software device. And then we also found that on average, participants, when objectively measuring how often they were using the device, we found that participants with the Learn to Quit app were consistently using the app more often than with the comparator. In this case, we also used NCI Quit Guide, which, by the way, it was fantastic to be able to use NCI Quit Guide and then the smokefree.gov software as a comparator, because really that was a trigger for innovation and for allowing us to push forward our work. I'm just going to mention a few few of the qualitative feedback that we received early on. So one of the main themes that was more salient from this work was that we observed a very high retention and comprehension of smoking cessation content. So acceptance and commitment of therapy is not it's a psychotherapeutic approach to smoking cessation. Is not necessarily simple. It's a kind of highly abstract concepts. And what we found is actually the way we presented and we were able to communicate this content, participants were able to retain and remember the stories, remember the concept very, very well after the end of these case studies. And coupled with that, we found few usability barriers, positive user experience, and gamifying the intervention was acceptable. We then moved on to a pilot randomized controlled trial of this intervention. I'm just going to focus on the main results. This was a two-arm pilot randomized controlled trial. We used QuitGuide as a comparator. We measured using digital markers of app use continuously throughout the trial. It was a 4 months, 120 days. And what we found was, obviously the trial was not powered to detect abstinence, but we found a very strong signal. The nominal quit value was small, 4 hours 33, but this was enough of a signal for us to move forward to the next stage. And then also we found significant reductions in cigarettes per day between the two arms with a decent effect size. So once we complete this trial, we also wanted to examine a very important question, which we felt was important to do, and this was the first time that this type of mechanistic analysis was published in a digital device for smoking cessation. In the field, you may argue that maybe just using a device may have an effect just simply because you're using a software device that could be fancy, could be engaging, and this might be actually leading or mediating the effects of intervention. So we had the opportunity to actually test our question. Does it matter that you tailor the device to this patient population versus simply not having any device? And what we found was that engagement with the digital therapeutic that we developed did have a direct clinical association with clinical outcomes. So here you see the first chart. This is the Learn to Quit group. And what we find was that participants with higher smoking cessation reductions engaged more often with Learn to Quit. There was a clear separation between those that had higher rates of reductions, a trial endpoint, versus those that did not have a high reduction rate. And we found there was the engagement mattered. However, in the QuitGuide group, we found those lines were very much close to parallel to each other. So participants with higher smoking reductions in the QuitGuide group and did not clearly engage more often with QuitGuide. And when we tested this, what you saw visually in a statistical model, this is exactly what the model showed us. So being randomized to the Learn to Quit app was and having a positive outcome in terms of reductions of cigarettes per day was fully mediated by actual engagement with the digital therapeutic. So the next step in this process was conducting a fully powered randomized controlled trial. So in 2020, in March 2020, we got funding for this pivotal trial, which was run both at Duke University and at University of Buffalo. One of my site partners at Buffalo is Dr. Ashar, and we worked initially also with Dr. Ravi Jnol at UPenn. The trial had its main outcome. The primary outcome was quit rates. We aimed to look at economic analysis, a cost-effectiveness analysis, and we would also look at mediators of outcomes. And we would compare learn to quit with advice. Over time, the trial became a fully remote, decentralized pivotal trial after we developed procedures for that were after approval by the FDA of one device, which I'll show you in a second. So throughout this process, we engaged with the FDA to inform the Mind to Quit trial, and that started in 2018. The NIDA program, Kevin Walton and Will Ackland were heavily involved in this process with us and partnered with us in this process. In 2018, we had an initial consultation, and this was followed by 4 supplements that informed the clinical endpoints and the design of this pivotal trial. In 2022, this was the location of our trial participants, so 2 different sites. And after the FDA cleared the use of the ICO-Quit device for biochemical verification of smoking abstinence, we were able to convert this multi-site trial into a decentralized clinical trial, and this is currently location of study participants that we have right now. This is some of the information around the recruitment rates, and there's a big gap between enrollment and randomization, and that's because not all participants are eligible, because we have to determine psychiatric eligibility and so on. But the trial is moving forward. I want to highlight some feedback here that we've been collecting during the course of this trial. So we selected, this is not a rigorous qualitative analysis of all the feedback we've received, but I wanted to highlight some comments that are consistent with the user-centered design research that we conducted initially when we were designing the digital therapeutic. So for example, a nurse, highlighted how this was enlightening content that she had never seen before. A person that described themselves as being old and said that this is easy to use and navigate. Another person mentioned that this app was packing a powerful punch. It was not time-consuming, and then in a short amount of time, you walk away with good knowledge. And then this is another comment that came up. This is also another consistent theme. Another person mentioned that the approach that we were following to cope with cravings was unusual, and I had never seen it before. It was refreshing and something that they appreciated. And in addition, the emphasis on reflection and stepping back and focus on reflecting on their actions. And then some people mentioned that actually this app seemed to be very appropriate for people with cognitive difficulties, which was one of the key elements that we designed this intervention for, in addition to symptoms of psychiatric illness. So the goal is to eventually turn this digital therapeutic into a prescription digital therapeutic, in a way to close the gap between dissemination and implementation of digital therapeutic. And this is a big challenge. The field is still trying to make sense of how can we do that, but there's really good examples out there. And when it comes to health equity, I posted a couple of opinion pieces back in 2021, where we're making the case that regulation of digital therapeutics may be necessary but not sufficient. In the same way, the development of digital therapeutics that are rigorously developed, not only from a user-centered design standpoint, from a feasibility pilot testing standpoint, but also from a pivotal trial or RCT standpoint is critical. But that's not necessarily going to solve the issue of health inequities to access to different groups of individuals at a population level. So we looked into this question with our own target population. We work with people with serious mental illness, which this population is at the intersection of tobacco use disorder. And there's a third intersection, which is belonging to a racial ethnic minority population. And we argued that this was a high-risk intersection. We looked into the data then, and we used the U.S. National Survey on Drug Use and Health in 2000 that covers about 10 years. And when we looked at this question, the first thing we noticed is that among people without serious psychological distress, which is the best proxy we could identify for people with serious mental illness, we found that both these three groups, Anglo-Americans, Black African Americans, and Hispanic Americans, actually reduced their rates of smoking over that 10-year period. However, when we looked at the same groups, but with serious psychological distress, the picture was different. So essentially what we noticed is that Anglo-Americans, white individuals who smoke and had serious psychological distress had a significant reduction, but African Americans and Hispanic Americans had stagnant rates of smoking during that same period. And that motivated us to examine that problem in the context of the Parent R01, the Mind to Quit trial. So the goal that we set up ourselves with this ancillary study funded by NIDA was to conduct a mixed methods digital equity study that would allow us to measure digital divide factors within the participants in the clinical trial, how much they had appropriate devices to use the technology, how much they had knowledge or technological literacy to use the devices. We looked at experiences of racism during the participation in the study, quality of life as something like above and beyond cessation, a more holistic measure of health. And then we also interviewed qualitative former participants in the clinical trial to understand their experiences with the technology. And then part of that analysis will also include interviewing directors, administrators of mental health clinics to understand their views on administration of smoking cessation services in this patient population, and then program evaluation of these clinics as well. Essentially, we are going to be using this information to inform a pragmatic or hybrid type 3 trial that would help solve the gap or address the barriers to access to digital therapeutics in this patient population. So the goal is not necessarily to reach equitable health across all groups, but to actually provide equitable access to digital therapeutics, and this is the goal of this ancillary study, and this is one of the future directions that we're going towards. And I wanted to thank my team that day-to-day make all this work happen. So thank you so much for your attention. Appreciate it.
Thank you so much, Dr. Bodaga, for that excellent presentation. My name is Neil Friedman. I'm the branch chief of the Tobacco Control Research Branch at the National Cancer Institute. It's my pleasure to introduce our fourth speaker, Dr. Megan Piper. She is professor in the University of Wisconsin Department of Medicine, and she'll be giving a very complimentary talk to, I think, the three you've just heard in this panel, and that is about alternative cessation endpoints. Dr. Piper.
Thank you. Thank you for this opportunity to come and speak to you today. We've heard some fantastic just sort of life stories, research stories about different evolutions of things that brilliant people have been doing in their laboratories. And I'm actually going to just poach a lot of other people's ideas and sort of see how we can think about things a little bit differently. So we all agree that smoking cessation is the best thing someone who smokes can do to improve their health. I think we're all in agreement on that one. And the International Society for Research on Nicotine and Tobacco came out with an update in 2019 about how best to measure abstinence, how you would define it, what time period you would use, and sort of set as the gold standard this notion of long-term 6-month biochemically confirmed outcomes. There was another paper on how best to biochemically confirm abstinence. And so this has sort of been the gold standard that we've been searching for, right, in any kind of intervention that we do. However, what if we stepped back and sort of let go of the gold standard and thought about how much more we could learn, focus on, or address if we weren't just searching for, I believe Frank called it, the ultimate end goal? What if we looked at the problem in a different way? What if we looked at lots of different ways that people could be successful? So what kind of alternate outcomes are there? I came up with 4 sort of categories when I was thinking about this. One reflects cessation process. One focuses on treatment mechanisms, which a lot of folks are already looking at. One focuses on potential harm reduction outcomes. And then let's talk about outcomes for interventions for people who aren't ready to quit yet. So the first category is cessation milestone. So that was the term coined by Saul Schiffman and his colleagues. And this notion was that you don't just magically get to 26 weeks. We only look there, but it's kind of like that notion of, somebody drops their keys by their car and it's real dark and somebody else comes out and finds them and they're looking under the light, the street light that's 2 blocks away. Well, where'd you drop your keys? Over there. Well, why are you looking over here? Well, that's where the light is, right? We're looking where we've been told to look, but we're maybe not looking where the action is or where the focus needs to be. So what are the milestones? One is initial cessation. Can people actually establish 24 hours of abstinence? I don't know about anybody else's studies up here on the panel, but we have sometimes 25% of participants in our study that don't even get that far. They come, they're motivated, they show up, they come back, and they still can't quit even for 24 hours in those first 2 weeks. So what if we had an intervention that could really get people started? Would that be helpful? Would that be effective? Would that impact public health? What if we had an ability to delay a lapse, right? The lapse is that first cigarette. Once you've gotten to cessation, then you have the first cigarette. What if we could delay that lapse? Or what if we could do something so that that lapse was not as enjoyable? Because then might that ultimately delay relapse? Relapse being defined as 7 consecutive days of smoking. So if we could do something to stretch the time between lapse and relapse, or just ultimately delay whatever relapse might be coming. And so you can look at these milestones. So I'm going to talk about a lot of different self-report measures, and many of them, if they're available on the Phoenix website, you can look there. I've noted that. So cessation milestones are part of the Phoenix toolkit if you are interested in looking there. And one of the really great things about this is we can do it because of the advancement of technology, right? We now all carry please raise your hand if you have a small computer on your person or just under your seat. Exactly, right? They're ubiquitous. And we find that that's true even with people who have very low financial resources and high levels of social needs. And so that means we can ask people questions all the time, which I love to do, and my participants don't necessarily appreciate. But we can look at things over time. We can look at things in real time. We can use ecological momentary assessment to, again, not just look where the street light is, but look where the action's actually happening. And so we can do this via apps. We can do it via text prompts. There's lots of different ways to look at it. And by being able to collect this temporally precise data, again, we can look at these milestones much more easily. Treatment mechanisms, okay? If you go back to your stats class, if anybody took, structural equation modeling, right, we have the main effect of the intervention hitting and significantly influencing a long-term outcome like long-term cessation. And ideally, there are treatment mechanisms that mediate this effect. So one thing we know is that we understand a lot about what interventions cause long-term cessation, what interventions are directly linked. What we have more trouble with are the 2 different mediation paths. Is the intervention itself pushing the treatment mechanisms? Is it actually changing what we think it's going to change? And then the other big question, how much are those treatment mechanisms actually responsible for somebody's long-term cessation success? And so really understanding the full mechanistic pathway is another important thing that we can look at, again, enhanced by the fact that we can assess things using EMA. So understanding treatment mechanisms may allow for precision medicine based on 2 different factors, based on baseline measures. And you'll notice here that my baseline measures are not groups, right? They're not groups of people. They are things that might be related to how a treatment might be perceived, accepted, experienced. So limited distress tolerance. There's lots of different folks that have limited distress tolerance. It may or may not be related to specific psychopathology. Different levels of dependence. I could put in here differential experiences of systemic racism. I could put in here differential experiences with the healthcare system. but any sort of baseline measure that we might be able to use to inform precision medicine. Or we could do it based on real-time responses. So if somebody's having limited craving suppression, could there be some sort of adaptive treatment trial that this would be a situation where we would increase or augment or alter treatment? And by understanding which mechanisms different treatments operate through, we can make much more informed decisions about how to use sort of a personalized medicine approach. It also, like I just said, informs combinations. If you have an intervention that's doing a really great job at craving suppression, but the person is experiencing significant anhedonia, then you might want to think about how best you would combine interventions to address both of those phenomena that drive return to smoking. So different potential mechanisms I'm just going to review. These aren't going to be surprises to anybody, but we'll go through it. Craving, negative and positive affect, cigarette reward, anhedonia, cessation fatigue, and cessation self-efficacy. Withdrawal scales have they have craving items as well as negative affect items. These are not exclusive lists, but two very common ones in the field are the Wisconsin Smoking Withdrawal Scale. It's got 19 items with 6 subscales, but if you're really pressed for time, there's a 6-item version that covers all the same scales. And then there's the Minnesota Withdrawal Scale, which has 8 items, again, assessing the variety of different withdrawal symptoms that are frequently experienced. Then there's craving per se. Craving sometimes is due to abstinence-induced craving. Sometimes it's due to cue-induced craving. It can be related to, sometimes those cues are external, sometimes those cues are internal. And so this is the one mediator that we know consistently is what consistently drives pharmacotherapy effects, right? We find very few other mediators of pharmacotherapy effects other than craving, which we could spin it and say that means there's a lot of opportunity to hit any other mechanism. But if you want to look at craving, you can use, again, in the Phoenix, it's called Phoenix Self-Reported Craving, and it's the brief questionnaire of smoking urges. It's got 10 items with 2 different factors. One is focused on intention or desire to smoke, and the other one's focused on relief of negative affect and the urgent desire to smoke. Or you can use, if you're going to do it for EMA, like we've done in the past, you can just use brief stems. You know, on a scale of 1 to 7, how strong is your urge to smoke? How much are you bothered by a desire to smoke? Affect, again, you can use the withdrawal items, or you can use the positive and negative affect scale, the PANAS, right? It's an oldie, but a really goodie in terms of having positive affect items, negative affect items, highly activated items, as well as low activation items. And you can use it on various time scales. How are you feeling in the last 15 minutes? How have you felt in the last day, the last month? And again, you can use select stems for EMA. Cigarette reward. Now, this is something that folks have talked about as well. And this can be assessed using the Modified Cigarette Evaluation Questionnaire. It's basically, how did smoking the last cigarette make you feel? How satisfied were you? How much did it help you calm down or help you focus? And how much did it, how many aversive experiences did you have? And then there's 2 additional single items about respiratory tract, so throat hit, think about throat hit as that one, and craving reduction. But the goal here is, what if we go back to process, that first cigarette you have is, to quote my children, meh, right? It was fine, didn't taste as good, right? We know that there are some interventions that actually reduce the rewarding value of cigarettes. And so what if that were to happen? Then perhaps you'd have a longer latency between that first lapsed cigarette and that ultimate relapse. So thinking about how cigarette reward might be helpful in if we can reduce that rewarding value of those cigarettes, what would that do with respect to ability to maintain long-term cessation? Anhedonia. Anhedonia is the inability to experience pleasure. And so this has been something shown by my colleague, Jess Cook, truly to demonstrate all of the key symptoms, all the key dynamics and characteristics of a withdrawal symptom. And so you can measure that using the Snaith-Hamilton Pleasure Scale, which has got 14 different items. We also use 3 items in our EMA studies. Basically, we just ask how much pleasure they experienced in the last day with respect to social activities, recreation activities, and performance and accomplishment activities. And anhedonia, again, is one of those symptoms that you see particularly driving people who are dealing with serious mental illness, depression, schizophrenia, other things. When you don't smoke, everything doesn't feel as good, things don't taste as good, life is just not as good. Well, what if we could address that issue directly and then see what we could do with respect to how people experience life as non-smokers, as well as help them with their cravings? Cessation fatigue. So Tim Baker used to tell me this story about a person he worked with who was smoking and trying to quit, and they said, you know, the idea, I can stand guard duty for like a night or two, maybe a week. The idea of standing guard duty for the rest of my life is exhausting. And that is cessation fatigue. People who feel like they are constantly in a battle and they don't think they can do it any longer. And we know that it's positively associated with craving and negative affect. It's actually moderated by gender. Women have more fatigue when they're dealing with high levels of craving, whereas men have more fatigue when they're dealing with high levels of negative affect. And you can just a single item. I'm tired of trying to quit smoking. But there's also a cessation fatigue scale that's been validated. It's got 3 subscales, and you can just use the emotional exhaustion as a single 7-item scale if that's more to your interest. Self-efficacy. So one of the most consistent predictors of cessation success is whether or not you feel like you can do it. And it makes sense, right? If you're doing well, you feel like it's going well, if your cravings are really strong, you feel like you're starting to slip, then, you know, is this sort of becoming a self-fulfilling prophecy? So the smoking cessation self-efficacy questionnaire is also in Phoenix. It's got 12 items. And basically, it's how confident are you that you could resist smoking in the context of specific internal stimuli, like feeling depressed, or how confident are you that you could do it with respect to external stimuli, being around other people who smoke, being in stressful situations, that sort of thing. Or you can just straight up ask them a face valid question. How confident are you that you can quit smoking success? So again, we have these mechanisms, and we talk about them as though they exist on their own little island, right? I'm on craving island or negative affect island. But what happens is they're all ridiculously interrelated, right? And so how do we think about the interrelation between all these different mechanisms? And I'm not going to try and go through all of them, but if we just focus on craving, right? If craving is the key driver of smoking cessation pharmacotherapy effects, we could think that anhedonia, right, that inability to experience pleasure, makes me crave a cigarette. I feel, you know, food tastes better after I smoke. Things are better. I can socialize better after I smoke. And we know that negative affect and craving have a bidirectional relationship. The worse I feel, the more I want a cigarette because it's going to make me feel better. And then once I've had a cigarette, now I feel bad. Now I feel like I'm not succeeding, I'm not achieving my goals of cessation. If I'm having high levels of craving, I start to feel my self-efficacy slip. Geez, I don't know how long I can do this. My cessation fatigue starts to kick in. Gosh, I really don't think I can handle these cravings without going back to smoking. And if you know those cigarette rewards are particularly good, then really are you going to be able to resist those strong cravings, right? If you know it's going to feel really good if you finally give in and have a cigarette, then your cravings just get larger and larger. So that was mechanisms. Let's talk about harm reduction as another possible category for outcomes to think about. There's been a little discussion today already about reducing the number of cigarettes per day. Does that perhaps reduce dependence? Does it perhaps increase self-efficacy? Oh, I was able to cut down. Maybe I could actually go all the way to 0. But we also have to remember that you have to actually demonstrate harm reduction, right? So we know that just a fewer number of cigarettes per day, despite what some of our participants tell us, they're so happy they're down to 5 and we're just stay here. 5 is great. It's like 5 is better than you were, but you're not yet seeing the health effects of full cessation. So what if we switch to an entirely non-combusted or non-inhaled source of nicotine? Would that be harm reduction? Again, we'd have to think about outcomes that actually measured that reduction in harm. So reduced biomarkers of exposure, reduced combustion, so that's exhaled carbon monoxide, reduced total levels of nicotine, right, which would be relevant to increased levels of dependence, as well as some of the physiologic effects. You can assess cotinine. Specific tobacco-derived nicotine products, you could look at anabasine as a biomarker there. Cured tobacco, you can look at things like NNL. Don't ask me to say the long word, I can't do it. But you can also look at specific toxicants, polyaromatic hydrocarbons, volatile organics, and heavy metals, among others. So thinking about what kind of harm reductions could we actually demonstrate by getting people to either reduce, switch entirely, or completely quit. And finally, I want to talk a little bit about outcomes for people who are not ready to quit. Just because people say they're not ready to quit doesn't mean they're not ready for treatment. We've done a treatment trial where we said, are you interested in quitting? And people said, no. We said, are you interested in cutting down or changing your patterns? And people are like, yeah, sure, right? It's a soft start. They're willing to do that. Quitting can be really scary. What if there was a soft start? What kind of treatments could we give people that would address and improve cessation self-efficacy? Oh, I just have to put this patch on every day? I could do that. Let me try that. Yeah, that works. Okay. No, I could. Oh, maybe my cravings went down a little bit. Maybe I feel like I could tackle this. Cessation motivation. Could you increase cessation motivation with this intervention? And again, there's a Phoenix item for that. Quit attempts. Most quit attempts are not successful. And boy, I would love it if this partnership between FDA and NIH would result in some amazing new interventions that are like 80% effective. But even if they're not, every time people quit, they learn something, right? That's what we tell them all the time, and we have to remember that too. They've learned something about how to help them quit. So the more quit attempts we can have them make, the better and the closer they get to cessation every time. So that could be any quit attempt. It could be a quit attempt that lasts 24 hours. And then, ideally, these quit attempts are in using evidence-based treatment, right? I woke up in the morning and I had too many cigarettes the night before and I don't feel great, so I quit smoking, and then I just did it. As opposed to, hey, I woke up in the morning and I don't feel great, but boy, I have those patches that my doctor gave me if I was ever in the mood to quit. And maybe if I just slap one of those on, I can see how things go from here, right? So this engagement in actual evidence-based interventions, what if we had things that could do that? What if people worked on restricting smoking environments, reducing cued exposures? What if we had other harm reduction outcomes, reduce cigarettes per day or switching and trying different products? Again, all potential outcomes for interventions for people who aren't ready to quit. And the last thing I want to talk about are the timing of outcomes. 6 months post-quit day is the gold standard. And as we recommended in the SRNT paper, ideally, the notion would be you'd look at outcomes after treatment and you'd look 3 months after treatment has ended. And then I say to myself, now why do we look at treatment for a chronic disease after the treatment is over? How is it supposed to keep helping people if they don't have it anymore and it's a chronic relapsing disease? So I think this is something that the field needs to grapple with a little bit more. What standard are we holding ourselves to, and is it appropriate? We also need to understand the impact of treatment discontinuation. So are we seeing, people study, I did my study and I looked at 6 month outcome, but I didn't, because again, we didn't necessarily have the technology to look precisely at what happens around the time that treatment is discontinued. We don't spend a lot of time focusing our energies on what happens when people stop using whatever intervention it is. It could be a digital intervention that they just use a couple times. It could be a pharmacotherapy. It could be counseling. Who knows? And then recognizing that the impact of treatment changes over time, right? So if it's really important to address craving early on so that you can really help people feel like they've got more self-efficacy, thank you. That would be great. And if they, but what if something else comes into play later on? So it's not just craving early on, it's other things later on. Then becomes anhedonia, or it becomes cessation fatigue, then we have to actually add something that boosts at that later time period. So understanding how mechanisms change over time. So in conclusion, complete cessation of commercial tobacco and nicotine is an ideal public health goal. There are alternate outcomes that we could think about that might provide significant additional scientific and clinical insight into how interventions function. And use of longitudinal data collection allows us to look at a lot of different important constructs. So, thank you.
Thank you so much for that excellent presentation. We have a few minutes for discussion for the panel. And to start us off, I was interested for the first three speakers. Dr. Piper presented a discussion of different endpoints that could be chosen as part of these trials. But what are the factors that are driving your consideration and when you choose endpoints in the trials that you've presented on today? Maybe we could just go in order.
Well, admittedly, I've been mostly considering the conventional outcomes, but I'm also a clinician, and I work. I'm also a clinician, and I work with patients with tobacco use disorder as well as other substance use disorders. And what Megan specifically was talking about really hit very close to home. For example, things like 24-hour abstinence. Why? Okay. 24-hour abstinence. The first 24 hours is really a very, very, very vulnerable time. And I'll admit, 20 years ago, I used to smoke myself, and I can tell you that the first 24 hours, I was climbing the walls. And it does get easier as the time goes by. And so this kind of outcome is an important outcome. Anhedonia, again, can relate very much personally and in terms of patient experiences as well. So in fact, pretty much nothing that Megan mentioned today everything hit close to home. All of these things have a lot of validity. And we also, again, need to consider different patient subgroups because for somebody, anhedonia is a factor. For somebody, another outcome could be a more salient factor. But then again, some of those are very much interconnected as well. So I really enjoyed your presentation, Megan.
Yeah, I agree with what Megan discussed today, especially, I'm a psychologist as well, and the process of quitting takes many trials and errors. We have enough empirical evidence to show that, and it's really a great opportunity for someone who quit smoking to try multiple times and reinforce those skills that allow them to ultimately, meet their goal. And so I think that's an important factor for me to consider. The 7-day point prevalent abstinence, for example, this is one of the outcomes recommended in the guidelines. I think it makes a lot of sense clinically, and that's the endpoint I've chosen in my trials. The other thing that I've, in my own work, I think it's great to have a task force that, openly proposes, specific guidelines for, how to consider and how to pick endpoints. And I think the work that Megan has done, Dr. Piper and the SRNT has done over the years is really, really valuable because it's not an easy thing to pick the endpoint. And is a very important one. It's a critical surrogate of survival, so it's really important to. So, but I kind of want to emphasize the clinical factor that needs to be taken into account when we think about picking an outcome. And I think sometimes continuous abstinence, for example, and I think it's in many cases not a realistic. It's an important outcome long term, but when you take into account that patients try multiple times and the number of times that someone tries is correlated to success, ultimate success, I think some of the recommended outcomes, proposed by that task force and SRNT, I think it make a lot of clinical sense.
So in the brain stimulation studies to date, we've chosen the industry standard and the FDA standard because we wanted to get FDA approval. And then for the NIH NIDA-funded studies, we want to be able to compare across the literature. So we're choosing the outcomes that will allow us to evaluate these new things, comparing to old. But with brain stimulation, we can get at all of those other motivations. So, for example, we know exactly where the anhedonic circuit is in the brain, and I'm doing studies now in post-stroke anhedonia. So the future that I can see, if we can get off the dime of everybody fits into one mold, is where we may be able to use brain stimulation therapies where we actually do ask you individually, what are your primary motivations for smoking, for relapse, and is it just craving, or is it anhedonia, or something else? In the studies, we've gone way out of our way to make sure they're not depressed, because we were worried that people would just say, oh, you're just getting them undepressed, and so we absolutely excluded any depressed smokers, but that's a big group. There are a lot of people out there, and so and I actually have a case series now of patients who are getting TMS at the VA who are smokers, and they came in voluntarily and said, look, I know this thing is also approved for smoking cessation. While I'm getting treated for my depression, should I try to quit smoking? And I said, no, that's going to make your depression hard to treat. And in 3 people, at least, they said, no, I want to try it. And they waited until the 2nd or 3rd week of the TMS, and their depression was lifting, and then they got into it, and they're abstinent. I mean, so I think actually if we begin to individualize treatment, and now with our knowledge of circuits and where those are, and with new tools like brain stimulation, other medications, we really can begin to meet the needs in a better way.
All right, one question. So this is for Drs. George and Villadaga. So Dr. Yermin, doing research that essentially showed that combining an ancillary medication with a well-used first-line treatment may increase efficacy for smoking cessation. Have either of you given any thought to how, in the future, brain stimulation or digital therapeutic treatments may be studied or to be combined with first-line treatments such as NRT or varenicline?
That's for me. Yeah, I mean, I think I always thought that, so speaking of the digital therapeutic that I developed, I think this is it's not necessarily a modular approach, but you can think of in the same way that we're combining NRT, combination NRT with a digital therapeutic. You can boost a digital therapeutic with different approaches. And I think that's definitely a direction I can see in the future. You know, not just the technology, the type of approach Dr. George mentioned, but also, for example, AI or many other approaches that could actually boost. I think, however, that you know, when it comes to developing a medication, you have to think about, you know, how can you get this medication across the brain-blood barrier. And one thing I would emphasize that we cannot forget is that there's a behavioral barrier to adoption of any intervention. And that means that culturally you need to make sure that you understand your patient population, you talk to your patient population, that your intervention is patient-centered, and that you have communication and conversation with the community to understand whether whatever combination of interventions you have is acceptable and has it generates trust. And so that kind of barrier, the cultural barrier, behavioral barrier is important to consider from the get-go, from the beginning, and that's what we did in our work. Because user-centered design, in many ways, you know, it's a mixed methods approach, and then it forces you to have a lot of conversations with your end users and understanding, you know, what could work, what could not work for them. So that's one aspect I would emphasize.
So I'm a scientist and a clinician, and scientifically, when we're asking about these new technologies, we exclude combination therapies. So the early depression trials with TMS, we did the medication free, and then after a while we said, look, it makes a lot more sense if we just add TMS to their underlying medications because they're not working, but withdrawing them is a whole lot of work. And so most TMS now is given as an adjunct to underlying pharmacotherapy. And the same with the smoking cessation studies. We didn't want to do combinations off the bat, but it makes total sense to figure out what combinations of medications or other proven therapies you would add something like brain stimulation on top of. And I can't imagine you wouldn't get augmentation and much better quit rates when you begin to do that. The question is which ones are safe and which ones work. In TMS for depression, we haven't yet figured out a real home run that boosts the antidepressant effect. People are using D-cycloserine trying to get at synaptic plasticity. So if that's actually what we're doing in the brain, we're changing synaptic plasticity, maybe we can give an augmenting agent. And there are 2 trials now that have shown that with TMS for depression. So thinking mechanistically, you can begin to think through exactly which combination therapies would work for the brain stimulation that you do. And I'm excited about the future, and I hope that this group and others will continue to fund that kind of work, because I think that will pay off in the clinic.
Can we get a nice round of applause for this amazing panel and for the moderators? So all this talk of craving has me really wanting to go for lunch, so we have an hour break. We'll be back at 1:35. And also, if you're like me and you suffer from caffeine use disorder, I will say that the coffee shop closes at 2 p.m. So please make sure that you get your coffee before you come back. Thank you, everyone. Good afternoon, people, and welcome back. Thank you again for attending this meeting on advancing smoking cessation. We'll get right into it. Our next session is a particularly exciting one that's focused on regulatory paths forward. And I'm going to just quickly introduce our 2 moderators. We have 2 really excellent, preeminent colleagues from CDER, Dr. Marta Sokolovska and Jeffrey Siegel.
Good afternoon, everyone. Thank you very much for coming back after the lunch and joining us for this very informative discussion, so we hope, and the great presentation so far. The meeting has been very informative, at least for me, and hope for you all too. We've learned a lot regarding the potential for new pharmacological treatments. We've heard about potential new treatments for the device space and digital therapeutics. We also have heard about alternative outcomes. We've heard about clinical practice and how does all these treatments translate into the importance of engaging the patients in the clinical practice and the importance of the different groups that we are trying to address, and to make sure that we do it in a meaningful format so they're really engaged in the treatment. So with that, I think it's really fitting that now we are moving to the next panel of the next session of our meeting, which is focusing on the regulatory pathway. We will have 4 presentations during this session. So I will introduce the 4 presenters, followed by a panel. First, we'll have Dr. Terry Michel presenting on the FDA guidance on nicotine replacement therapies, review of additional outcomes. Then we'll follow with Dr. Devin Riesner, who will present on the clinical outcome assessments from the FDA perspective. He's the Division Director of Division of Clinical Outcome Assessments within CDER. I should have mentioned that Dr. Terry Michel, she's the Director of Office of Non-Prescription Drugs within CDER as well. It will follow by the 3rd presentation on trials integrated with clinical practice, again, very much fitting into what we've been discussing so far. And that presentation will be provided by Dr. Leonard Sachs, from Associate Director in Office of Medical Policy at CDER. And the last presentation, but definitely not least, will be by Dr. David Murray, Associate Director for Prevention and Director of the Office of Disease Prevention within NIH, and this presentation will be on clustering in clinical trials. So with that, please, let's welcome the first presentation, Dr. Terry Michel.
So thank you, Dr. Sokolovska, and good afternoon, everyone.
It's exciting to be part of this conference today and try to find additional ways to really move that needle on getting more people to quit smoking. So as a practicing pulmonologist, I regularly take care of patients who are smoking, and so I see the devastation that smoking can cause kind of firsthand, as well as the challenges that smokers face when they are trying to quit. So as such, I'm really pleased to be able to present and talk about our latest guidance on developing nicotine replacement therapies. Now, while the guidance is specific for NRTs, many of the novel concepts that are covered in the guidance could be easily applied to other smoking cessation therapies. So we issued the final guidance on NRT drug development to help sponsors in their clinical development of NRT drug products, as well as encourage innovation in this space. The guidance includes a variety of novel and expedited development pathways for both prescription and non-prescription drug development. Now, there's also a companion guidance to this that talks about the non-clinical development, including NRTs with a pulmonary route of delivery. So that's a very different kind of guidance, but is the first time that FDA has ever articulated the non-clinical development pathway for an inhalational drug product. So I do have a link for this at the end of the presentation, although I won't be specifically discussing it today. Now, just to make sure that everybody's on the same page when I talk about various development pathways, I'll start off with a brief description of the difference between prescription and non-prescription drugs. So, when do we consider a drug to be prescription? Well, under Section 505B of the FDA Act, a drug is considered prescription if it is not safe to use except under the supervision of a practitioner who is licensed to administer the drug. And this could be either because of toxicity, because of method of use, say you have to give it IV, or collateral measures like monitoring that are necessary for its use. There's actually no definition of a non-prescription drug in the Act, but basically it's sort of the opposite of a prescription drug. So a drug can be non-prescription if it can be used safely and effectively by a consumer without the intervention of a healthcare professional. So while it's not required by the Act, generally non-prescription drugs are available freely over the counter in a pharmacy, in a grocery store. They meet people where they are, and that's what's so useful about these for smoking cessation. We want our products to be very accessible to consumers. So as I previously alluded to, the NRT guidance includes a number of innovations that FDA has not previously articulated in policy. These include new indications, new treatment regimens, various discussion of regulatory pathways, as well as discussion of pediatric and expedited pathways. And finally, one thing that's really unique to this guidance, it's a huge benefit to sponsors, and we hope to consumers and patients as well who may be reading labeling, is the opportunity to make additional labeling claims related to the clinical benefits of smoking cessation. So I'll walk through each of these innovations in turn in the remainder of my presentation. So cessation, or total abstinence from smoking, is of course the gold standard. And that's one that FDA has used successfully for many years to approve all of our currently approved smoking cessation products. It is by definition, as you heard from Dr. Tanhai earlier, a validated surrogate measurement, and that's because it's known to be predictive of various health outcomes like reducing lung disease and heart disease and various kinds of cancers. And of course, that's what we really care about. Most of the smoking cessation therapies approved to date have used an ascertainment period of 12 weeks or longer
because, of course, the longer someone is able to maintain abstinence, the more likely they are to be a permanent quitter and not go back to smoking. Now, however, given the experience that we have with NRTs, in this guidance, we actually shortened that ascertainment period to a minimum of 4 weeks. And we considered that to be kind of the shortest time that has any likelihood of predicting future smoking cessation and, of course, the health benefits that we all care about. In addition, this guidance has something fairly novel in terms of endpoints that we've included a different endpoint, reduction in risk of relapse, as a primary endpoint. This is a new endpoint. We don't currently have any drugs approved for this, but recognizing that relapse is one of the most important issues for people who are trying to quit, we recognize that some NRTs, as well as perhaps some other smoking cessation therapies, may have benefit if given for a longer period or after someone has had a successful quit attempt. So we think this is an exciting new pathway that could be developed. And lastly, for products that have already met one of the primary endpoints for smoking cessation or reduction in risk of relapse, for the first time, we're also allowing sponsors to consider secondary endpoints and achieve additional labeling claims for those endpoints of reduction in urge to smoke, for relief of cue-induced craving in former smokers, and relief of withdrawal symptoms in smokers who are not trying to quit but are required to abstain for one reason or another. We also talk about new treatment regimens, for example, pre-treatment before quit day or reduced to quit. Again, these ideas that while the endpoint is abstinence, there may be multiple different ways to get there. We also consider combination of more than one NRT. And while not specifically outlined in the guidance, sponsors can propose any number of different regimens or any number of different combinations, including combination of NRTs with a different kind of therapy or a different non-NRT drug product. So we encourage sponsors to come in and talk to us if you have an alternative regimen that you'd like to work on. Moving on to other regulatory pathways, because there are a number of already approved NRTs, sponsors can use an abbreviated 505(b)(2) pathway and reliance on the literature. We also talk about a PK bracketing approach based on FDA's previous findings of safety and efficacy, and that's certainly a way to abbreviate NRT development. The guidance also describes the expected study population, which is basically smokers who wish to quit smoking. And then it talks about a variety of ways that you might enrich or define that population. Another new concept in this guidance is a discussion of development pathways for non-prescription development. And I'll talk about that a little bit more in the next few slides, since many people are not familiar with the concept of non-prescription development if you're working primarily in the prescription space. So there are two main NDA pathways for non-prescription drug development. One is prescription to non-prescription switch. Now, this is used when the drug is already approved as a prescription product and the desire is to switch it to non-prescription. If you recall from earlier in the talk, in order to be non-prescription, a drug product has to be able to be used safely and effectively by the consumer without that healthcare professional helping them choose the product or describing how to use the product. So except for aspects that are unique to non-prescription, in this pathway, there's generally no need to repeat the studies that were already done to support approval in the prescription space. Now, even though prescription to non-prescription is the most common way that drugs get to be non-prescription, We also encourage sponsors to come to us with direct to non-prescription development. In fact, there is absolutely no requirement that a drug has to be prescription first before it can be switched to non-prescription. Now, in this case, the non-prescription product has to complete a full development program showing both safety and efficacy, in addition to showing that consumers can use the drug without the intervention of a healthcare professional. So currently, non-prescription drugs have to comply with adequate labeling. And this basically means that they need a drug facts label. And you see an example of this on the right-hand side of the slide. You can see how much information needs to fit in this itty-bitty tiny space on that drug facts label. So for certain products, and I think NRTs are a good example, the limitations of labeling can sometimes present some challenges in adequately communicating all that, and also in communicating some of the behavioral supports that are so critical to using these products. So we encourage the use of all other types of labeling in addition, such as package inserts or websites, or basically whatever you can dream up. So again, we welcome sponsors to come in and talk to us about these alternative labeling proposals. You still need everything on that DFL, but you can use some of these ways to augment your labeling. Now, I've talked about consumers needing to be able to use a product safely and effectively, but how do we know if labeling is adequate? Well, to do this, we use consumer studies. These are studies that include label comprehension studies, which is testing to see if consumers can actually understand the message on the label, self-selection studies, which is determining whether consumers can select to use the product based on their own particular medical condition. Actual use studies. In this case, a consumer will be given the opportunity to purchase the product, and we measure certain parameters related to use of the product. And finally, human factor studies. Human factor studies are used primarily for drug-device combination products, and they test how that consumer interacts to manipulate the device, and such as following the instructions for use appropriately. Another aspect of this guidance is a discussion of pediatric drug development for NRTs. Now, we welcome sponsors to submit a proposed pediatric study request under the Best Pharmaceuticals for Children Act, in which they can get pediatric exclusivity. And the guidance also discusses ways for sponsors to fulfill the requirements under the Pediatric Research Equity Act. This is when you are developing a product for a new active ingredient, a new dosage form, a new route of administration, you have to have a pediatric study plan. Now, to date, we have waived PREA requirements for children under 12 years of age because the studies are highly impractical in this patient population, but the sponsor has to justify that in their plan. And we generally expect that the plan will include an up-to-date estimate of the size of the pediatric smoking population. The guidance also articulates the policy that FDA considers tobacco dependence to be a serious or life-threatening condition. This is, again, the first time that we have put our nickel down on this. So I'll say that one more time. FDA considers tobacco dependence to be a serious or life-threatening condition. And this is important because it opens up the door for consideration of expedited programs or review pathways like fast track and breakthrough and priority review. So products that meet the criteria for one of these pathways have the opportunity to request this as part of their development plan. One thing I will note is that because PK does not necessarily correlate to efficacy, a shorter time to maximum plasma concentration alone is not sufficient for these programs. And that brings us to what I think is the most exciting thing in this whole guidance, which is for NRT products that have demonstrated effectiveness for cessation or reduction in risk of relapse. Sponsors can include labeling for certain benefits of quitting without providing additional data. And while this may seem common sense on face value, that yes, of course, quitting smoking would be predictive of these things, FDA saying like that we will take something like this on faith is without demonstration of a product-specific finding is huge. And in fact, to my knowledge, this is the first time that we have ever done anything like this. So this demonstrates that CEDAR is not just encouraging drug development in this space, but in fact, we are rolling out the red carpet and are very strongly supporting it. So in summary, the NRT Guidance outlines strategies to make NRT drug development more efficient, more streamlined. We clarify appropriate pathways. We explain some simplified efficacy study requirements, outline abbreviated pathways, and encourage sponsors to consider expedited development if they qualify. So I'll end with some references for you and turn it over to Dr. David Reisner, again, who is the Director of the Division of Clinical Outcome Assessment. Thank you.
Thanks, everyone. My name is David Reisner, and I've already been introduced, so we won't dilly-dally. My disclaimer as an individual scientist, and in this segment, we're going to talk a little bit about clinical outcome assessments. I want to ground us in some of the terminology and the methodology that we recommend in guidance for the development of clinical outcome assessments, whether they're a patient-reported outcome, which might be especially appropriate here, in terms of smoking cessation, or another type of COA that you might hear about later in our session. Yes? Thank you. So to start off, we'll talk a little bit about concept of interest. So the concept of interest is what you intend to measure about a meaningful aspect of health, and it helps to articulate this. Sometimes it seems self-evident, but when we review volumes of material, particularly the more abstract they are, for instance, a digital health technology, starting with writing down what you intend to measure is very important. So the concept of interest is the aspect of an individual's experience or clinical, biological, physical, or functional state that the assessment is intended to capture or reflect. And in particularly, you should pick something that you think you can succeed with. So having something that will change within the context of use, which could be length of trial, in this case I've heard for weeks, is important, and having a tool that's sensitive in that same context of use, demonstrated to be sensitive in that same context of use, is helpful. Moving on, another terminology you may hear or is the context of use. So I sort of alluded to that. It has a lot of different components. I won't go through all the detail, but it is presented in guidance. One particularly important aspect of context of use is the target population. So, for example, we would expect evidence that the COA, for instance, a patient-reported outcome, measures the concept of interest in a particular target population. And that's important because these items may not be as relevant or understood when you're moving from one condition to another. So borrowing, say, a question from opioid use disorder should be informed by evidence from a new population. It's also true that the range of severity could differ between populations, could even be subpopulations, right? So for instance, it was mentioned that a secondary endpoint in the context of smoking cessation might be withdrawal symptoms in patients who are temporarily not on nicotine. So it's a different patient population. Maybe it changes the severity of symptoms. So you need to know that your items, that your tools are still sensitive and within the right range. To broaden this out a little, because I've alluded to the COAs that are probably most applicable, but we look at different COA types, right? So patient-reported outcome, PRO, observer-reported outcome, OBSRO, clinician-reported outcome, ClinRO, and performance measures. Those are more or less standardized tasks that are conducted under a certain set of rules. In any case, a COA, broadly writ, is a measure that describes or reflects how a patient feels, functions, or survives. That basically defines treatment benefit in the context of regulatory speak. So I'm going to throw a couple of complex figures at you. I don't expect to cover them all. I will point you towards the end of this deck towards some available guidance that might be informative. You may be doing the exact same things with a different terminology, with a different literature set, and so if you look at our guidances, you'll probably recognize some of the approaches that you're already taking. But we refer to this as the roadmap, and it sort of takes you from where you start to where you end. I want to point out that it's not necessary to develop something from scratch in order to use a tool in the context of regulatory trials. Modifying an instrument would be reasonable, and then supporting that modification with evidence from your particular context of use. The type of patients you intend to study is important. So without going into all the detail, the roadmap suggests you start with understanding the disease or the condition. Seems logical. That you conceptualize the clinical benefits and risks, and that you move forward to picking the clinical outcome assessment type that you might want to use. So again, PRO comes to mind, but there are other possibilities here that have potential in terms of innovation. And then in terms of whether you can find an existing tool to take off the shelf or modify one or develop one, there is a sense in which the way we conceptualize these things is changing, and I'll mention that later again. So as the targets evolve, our understanding of the substance use disorder evolves, you may choose to measure different things. And so in that case, you might still have the potential where you'd want to develop something new as opposed to modify something. In any case, to the extent that you would like us to weigh in, either in terms of advice, say, and during the IND stage of a medical product development, or you would like a particular outcome of a review of a submission like an NDA, supporting your contentions with at least an explanation of your summary from the literature, or if you've generated evidence, to pool that evidence together and argue from your evidence for how you would interpret what your tool measures and how that implies treatment benefit in the context of your program. The second figure is referred to as the conceptual framework. So this is the conceptual framework of the particular instrument. And in the way we've sort of come to summarize it most recently, it has two components: the conceptual model, which is broader than a particular instrument, and then the measurement model, which is actually what you're doing in terms of presenting patients or observers or clinicians with items or individual questions on a questionnaire. So the model is important because you don't choose to measure everything that's out there, but it is very helpful to document what you think the universe is around the patient. So in this example, it's documented that there are symptoms, symptom A, that it exists. So that's come from qualitative research with patients, their caretakers, informed, experienced physicians who treat. But in this case, that's not part of the instrument going forward. This instrument is focused on behaviors, observable behaviors, and the concept there is that these are behaviors associated with a particular symptom, and that they will be collected with the COA type of a OBSRO, usually a caretaker, perhaps a parent, and that OBSRO will be built item by item in the measurement model. So, the conceptual model of the indication is helpful because it gives context to what you choose to measure, because you don't choose to measure everything. And then I wanted to touch on clinically meaningful within patient change. It is where a lot of attention is paid. It seems many times clinical trials read out with a statistically significant finding, and either the clinical meaningfulness of that finding is in question, or you begin to look at certain patient subsets. And as you divide the trial into finer and finer groupings, it becomes harder and harder to conclude whether there's a clinically meaningful within patient change. So we have methodology. There's more than one. I'm going to talk about anchor-based methods a little bit just to illustrate. There are qualitative methods such as exit interviews. Those can be influenced in part by what sort of data you have available, because anchor-based analyses are very quantitative. So if you have 200 patients, very amenable to quantitative analysis. If you're working in the context of rare disease, qualitative interviews and talking to patients about what is meaningful to them in terms of the change they've experienced is probably the more likely pathway. So this is more for documentation than anything else. I'm just going to use an example of a change score from baseline on some hypothetical rating scale and give you some figures to look at and show how we might use them. So this is a ECDF curve. It presents the cumulative percentage of patients reaching increasingly higher criteria. In this case, it's set up with improvement on the left and worsening on the right. So the patient that shows the biggest improvement, often this is a decrease from baseline, are showing up on the left side. And as you move across, more and more patients are getting picked up. The second curve from the right is a curve for all the patients in our hypothetical study who appear to have not changed. And the way we figured that out is we asked them at baseline a very simple patient global impression of severity, none, mild, moderate, severe, for instance. So a patient who reported at baseline they might be moderate on something, such as severity of craving. Then you ask them again at the end of the study. If they're still reporting that they're moderate on severity of craving, that's a no change, right? So that's their shift score is a 0. So that the second curve to the right are all those folks. Now, if people improved, whether it was due to treatment or spontaneously in the placebo arm, they may have shifted, for instance, from a severe to a moderate or a moderate to a mild. Those patients would show up in the second curve from the right. To my eye, those two curves are different. And what we will do is use that difference as a yardstick to determine which patients overall across treatments have improved. It turns out that the median value for people who dropped at least one level on our severity rating from severe to moderate, moderate to mild, mild to none, that median is around 8. So we're going to say that that is about the change we would see in an individual patient that would represent, on average, improvement. We can use that again to evaluate across treatment arms. So let's move to that data presentation. These are the mean differences for your reference. So on average, the two groups are about 8 units apart. This is the representation of our hypothetical case in terms of treatment arms. So it's the same setup as before, only before the groupings were patients who were asserting they changed or not. On this plot, this is a treatment difference. So to my eye, these two curves are separated. They are separated relatively consistently across a wide range where patients who receive drug begin to report improvement sooner and at higher levels than patients who are on placebo. So that's helpful. So there's two things we want to look at. How far away are these two curves? That's where all these red arrows are. And what we can do is take those red arrows and plot them another way. But as I mentioned, they're relatively consistently separated. And keeping in mind, we think that if you can improve by at least 8 points on our change score from baseline, you're probably in that group of people that told us you improved. So if you plot all those red arrows, this is what it looks like. And it's relatively flat in the middle. We can ask ourselves what percentage of patients are reporting improvement in active versus placebo. That's just the separation. So it's running at around 18 percentage points, give or take. So we know that patients who improved by 8 points probably reported an improvement of at least one step, and that leads us to a separation between the two treatment arms of around 18 percentage points. Then we can ask, is 18 percentage points meaningful in terms of a group difference? So that's how we think about meaningful within patient change. So I have just a minute or so, but I wanted to turn this back to today's topic. So areas where clinical outcome assessment may be helpful, essentially has already been presented, because these are, in addition to maybe prevention of relapse, the secondary endpoints that were mentioned. So reduction in urge to smoke and relief of cue-induced craving in former smokers could be a target for clinical outcome assessment. Relief of withdrawal symptoms not associated with a cessation attempt was mentioned. Again, that might involve a different level of severity than you would see in people who have a successful quitting episode. Substitution of substance abuse disorders was one thought I had, having worked on use disorders that are other than nicotine. You might target a particular concept of interest, such as dependency. Now, I wanted to mention, for fair disclosure, the way we think about dependency is changing. This is the citation to DSM-5 and the way that use disorder is characterized most recently. I did find an instrument for nicotine dependence. This seems like an older instrument. If someone wanted to use this, we would have a discussion about whether this reflects our current understanding of the patient experience, whether these items are likely to work well, whether that's a concept of interest is well defined, et cetera. And that's part of the advice we provide to sponsors. But it's usual to begin with a literature review where you would identify what's available and whether you wanted to either select it, to modify it, or consider developing your own instrument. I wanted to point out, considering the sponsorship of the meeting, there are substance use items in the PROMIS item bank. And if you're not familiar with PROMIS, you know, we could provide additional references, but that might be a really reasonable place to start if you're looking for items early in a program and haven't had the opportunity to invest in customized instruments, and you could create your own custom short form from the item bank. And this is just a little background on the development and an appropriate citation. Oh, sorry about the blank. This is the link to the appropriate guidances. These are referred to as the Patient-Focused Drug Development Guidance Series. I talked a little bit about the one selecting, developing, or modifying fit-for-purpose clinical outcome assessments. That's where you would find the two figures I presented. There's an older guidance that's still in effect. It's the 2009 Patient-Reported Outcome Measures Guidance. And I think that's the end of my deck. Thank you very much, and Dr. Leonard Sachs will be presenting next.
Thanks very much, David, and thanks to the rest of the folks for inviting me to join. I'm Leonard Sachs from the Office of Medical Policy. I'm an associate director there, where I lead a lot of the programs on clinical trial innovation. I've taken the liberty of expanding my topic a little bit just to broaden the context so folks can appreciate where point-of-care trials sit. So, So I guess the big picture, the big question is, how can we use modern technology to improve the way we do clinical trials? I think we all recognize that technological advances have given us enormous opportunities to make trials more convenient for patients, more inclusive, and more efficient to conduct. And the particular areas of interest are, first of all, information technology, which supports easy and rapid exchange of data. We can exchange data, we can exchange documents, we can exchange images virtually instantaneously anywhere across the world. The other thing is that technology supports user-friendly remote communication, which may involve videos, may involve audios, and chat. And the third aspect, which I think also gives us new opportunities, are digital health technologies, which allow remote measurement of clinical features in study participants. And I'll talk a little bit more about those and tell you what they are. So as far as technology and new trials go, these are powerful tools, and they provide opportunities to do various things. First of all, they give us an opportunity to integrate clinical research activities into clinical practice environments, which is the one guidance that I'll talk about in a moment. This is platforms where interactive platforms which can exchange data between different users and so on and so forth. The second thing is that they allow us to engage the healthcare community in clinical research, which is not something that's typically being done. Traditional research relies on dedicated study staff. So communication platforms and exchange and IT exchange platforms certainly allow us to engage clinical practice environments, things such as electronic health records and so on. And the other thing that these technologies do is they allow us to obtain information directly from study participants. We've published a couple of guidances to support these opportunities. The guidances include guidance on digital health technologies for remote data acquisition and clinical investigations, which we published in December last year. Another guidance on conducting clinical trials with decentralized elements. The final guidance was just published in September last month. And the third guidance on integrating randomized controlled trials for drug and biological products into routine clinical practice, again, also published last month. The question is whether new trial designs address the challenges for developing drugs in smoking cessation. I'm not an expert on smoking cessation, but I think these are pretty generic issues in drug development. It's finding the right suitable trial populations. It's designing trials that are convenient and align with the participant needs. And it's streaming trial designs to focus on the information that is most critical to evaluate drug efficacy in a particular indication, in this case, smoking cessation. So let's talk a little bit first about trials with decentralized elements. The goal of this initiative and our guidance is to promote bringing the trial to the patient rather than bringing the patient to the trial, and effectively bringing a lot of the trial-related activities to patients' homes. And I think everyone's fairly aware of the advantages there. We're busy people. We don't have time to go to clinical trials and sit in traffic and battle for parking. And some of us have childcare duties and so on. So really to be able to participate in trials without a whole lot of obligations, remote visits and having visits at home are very convenient. Another strategy within the bundle of strategies that we have for decentralizing activities includes direct distribution of investigational products to participants, so they don't have to travel to trial sites to get their drugs. Another important advance there is the use of local healthcare providers and facilities to perform trial-related activities that don't require research-specific expertise. In other words, if you're sitting out in some suburban location, can you go to the local clinic to have your blood pressure measured or your weight measured instead of coming to the central trial site? Electronic informed consent is another useful tool. We published a guidance on this several years ago, and it allows people to sign informed consent from their homes. And finally, as I mentioned, digital health technologies. These are things like smartwatches and pedometers and cardiac monitors and all these things that you can carry around with you, which allow us to obtain information directly from participants. Can trials with decentralized elements be considered to study smoking cessation? Well, trials with decentralized elements generally focus on conditions that can be studied remotely. Smoking cessation seems a good candidate. They focus on investigational products that are safe to study in decentralized settings. And I certainly hope that the products that are envisaged for smoking cessation are safe in that environment. They should be convenient for participants, and they should facilitate the inclusion of large and diverse populations. So I think, I certainly think there's a case to be made for decentralized elements in trials for smoking cessation. Let's move on to integrating trials into clinical practice, otherwise known as point-of-care trials or sometimes known as pragmatic trials. And the goal of these trials is to bring trials to the point of care, the place where patients get their medical attention. The idea is to have trial designs and activities that are streamlined and aligned with clinical practice, that you don't have to do extra procedures for the research. They allow the incorporation of real-world data from electronic or other healthcare records as part of the information needed for the trial. Trial-related activities can be conducted at the point of care and with the participation of local healthcare providers. I'll tell you a little bit more about this. And finally, dedicated trial staff can also participate to perform research-specific activities if needed. So these can be sort of hybrid. I wanted to talk about something which is new, at least it's not something that we've addressed in previous sort of regulatory environment, which is the role of healthcare institutions in the conduct of a clinical trial. And in our guidance, we say that sponsors may engage healthcare institutions, for example, health maintenance organizations, hospitals, and clinical networks. This is a very powerful tool. We've seen it fairly recently. The poster child, in my view, was the recovery trial for COVID-19 in the UK, which used healthcare institutions all across the UK to approve a couple of drugs for COVID-19. And the same environment is obviously potentially of great value in other areas, and I imagine smoking cessation is a good candidate. Healthcare institutions may facilitate rapid enrollment of many patients by improving accessibility and convenience. We mentioned in the guidance that agreements should document the responsibilities of healthcare institutions and their employees and the tasks that they will perform, so it must be clear who does what and what the responsibilities will be of the sponsor. And finally, sponsors should ensure that the institutions and the local healthcare providers are suitably credentialed. And I think that's a critical point. These institutions are performing healthcare functions as in the role of critical care institutions, and generally they are credentialed for looking after people's welfare. Clinical oversight in this environment is very important, as it is in any clinical trial. Clinical investigators are responsible for ensuring that the trial is conducted according to the investigator statement, the investigational plan, and clinical investigators have the responsibility to protect the rights, the safety and welfare of trial participants. And we recommend in the guidance that clinical investigators must review the pertinent trial-related records provided by local healthcare providers and must ensure the accuracy and completeness of data. So clinical investigators have to stay on top of the data that comes in this type of trial. Local healthcare providers can do a number of things in a clinical trial. Working as part of healthcare institutions or individual practices, they may be engaged to perform tasks that really don't require any trial-specific knowledge or trial-specific training, although they may need limited instructions on what needs to be done. And examples here would include things like performing a routine medical procedure, maybe drawing blood or taking an X-ray at specified time points as stipulated in the protocol. Local healthcare providers can collect routine clinical data for the trial, for example, vital signs, and put them in a template. Local healthcare providers can presumably follow prompts in the EHR to document specific clinical events, for example, deaths, myocardial infarctions, strokes, or seizures. And procedures or processes that contribute directly and significantly to the trial data and do require study-specific training or detailed knowledge of the protocol should be left to trial personnel who've had the proper training. The scope of point-of-care trials, in our guidance, we point out that these are probably most suited to drugs that are already approved, for example, for new indications, new populations, or new routes of administration. But it may be possible to study unapproved drugs in these point-of-care trials, particularly those that have well-understood safety profiles in clinical practice environments. For example, those that are members of existing classes, those where we already know a fair amount about the safety from prior work. And it's important that the sponsor ensures that the source records for or certified copies of source records are available for these to support clinical trial data and can be reviewed by FDA if needed. We incorporate in our guidance a couple of quality design considerations for point-of-care trials, appropriate flexibility in trial protocols so that we can align them with clinical practice, Eligibility criteria should generally be designed to be minimal and straightforward. Informed consent documents must be obtained, and they can be embedded in the electronic health record. We emphasize the importance of randomization and blinding. We also talk about flags in electronic health records that may alert healthcare providers to comorbidities or to concomitant medications that are not allowed by the protocol. And we also recommend the real-time monitoring of EHRs and or calls periodically to study participants to make sure that we capture adverse events, which is not that different from the way they're captured in traditional clinical trials conducted on an outpatient basis. So what about point-of-care trials? Are these suitable for studying smoking cessation? Well, patients seeking assistance with smoking cessation may present to primary care providers. It's definitely been my experience as a practitioner at the VA hospitals, where people come either because they're wanting to quit or because they've developed pulmonary populations and we tell them that they need to quit. So they certainly are likely to approach primary care providers. We can find the populations. Clinical networks can recruit large numbers of patients. The evaluations for smoking cessation can presumably be fairly easily aligned with clinical practice. And study-related activities don't require specific research-related expertise. I just want to finish off with two very quick slides on digital health technologies. The goal of digital health technologies is to obtain measurements of clinical features directly from patients. And as I mentioned earlier, these include wearables, things like smartwatches and Fitbits and smartphones and so on, interactive applications, instruments placed in the patient's environment that can measure clinical features of interest in a clinical trial. And these digital health technologies may allow us to continuously measure clinical features. They may allow qualitative, objective measurements of clinical features, and they also may allow us to capture infrequent events, which probably don't particularly apply to smoking cessation, but things like seizures, falls, arrhythmias, and so on. Is there a role for digital health technologies in smoking cessation? I'll leave that up to the audience to think about. Are there wearables that can record smoking activities? Probably are. I'm not aware of them. Certainly, digital health technologies provide electronic patient-recorded outcome tools on smartphones and so on, and these can obviously be of value. That brings me to my conclusions, which are smoking and the treatments for smoking cessation appear pretty well suited to study in point-of-care trials or decentralized clinical trials. These sorts of trials may allow access to large, diverse patient populations. And I leave us with a question about whether DHTs can serve as innovative tools to monitor the efficacy of treatment for smoking cessation. Thanks very much. Hand over to David.
So we've spent the day talking about development, evaluation of new drugs, devices, products that could improve smoking cessation, help people quit. I want to turn our attention during this last talk to an issue in trial design that is not well understood. And I'll start with two key questions for trial design. So the first one, how are participants assigned to study arms? Most investigators and clinical trialists have come to understand that it really matters whether we're assigning individuals or whether we're just assigning groups or clusters. That question is largely understood, and people pay good attention to it. The second question, how is the intervention delivered to participants? I'm here to tell you that most investigators pay no attention to that question as regards the design of the study and are unfamiliar with the implications of decisions made on how the intervention is delivered to participants, implications for data analysis, for sample size estimation. So I'm going to particularly focus on the second question, though I will speak a little bit about the first question as well. Let's take the common situation, certainly the common situation for FDA, where drugs, devices, product trials are very common, and most of these studies randomize individuals to study arms. so long as there is no interaction among participants after randomization, they're not talking to one another, they're not meeting together in small groups, they're not interacting with the same counselor or therapist, the issues that I'm going to talk about later don't apply, and we can design and analyze these studies as RCTs using the methods that you all know well. Other trials randomize groups or clusters to study arms. Maybe it's a pragmatic trial and we've randomized physicians' clinics or physicians themselves, and then we deliver the intervention to participants or patients within those clinics or patients of those physicians and monitor them over time to look at the effect of the intervention. These trials often use existing groups or clusters. We have to worry about interclass correlation that exists before the study ever starts and certainly exists during the study, and we call these group or cluster randomized trials, and most investigators are now acquainted with these. There are two varieties. We used to talk only about the first one, the parallel group randomized trial, separate but parallel arms, no crossover. Over the last 15 years, the step wedge trial has become very popular. It's an alternative to a parallel group randomized trial. The perceived advantage is that everybody gets the intervention before the study's over. I would argue that's a false advantage. Step wedge designs face a number of risks that parallel designs don't face, so I always recommend the parallel approach if it's at all possible. The impact of clustering in a parallel group randomized trial. Groups or clusters are nested within study arms. Those factors have to be modeled as random effects in your data analysis. And the big 2 points, the variance of any group-level statistic is going to be larger, and the degrees of freedom available to estimate group-level components of variance are going to be limited. If you ignore those points, you're going to have an inflated type 1 error rate. Nobody wants that. Most people now understand this, and so they plan these trials accordingly to accommodate the extra variation and the limited degrees of freedom. The step wedge design, as I said, is more complicated. Everybody starts in the intervention arm. Groups transfer over to the, start in the control arm. Groups transfer over to the intervention arm on a pre-specified schedule in a random order. And there are a number of biases that step wedge designs face that the parallel designs don't face. The chief among those is that external events can completely disrupt a step wedge design, because as the study goes on, I have fewer and fewer comparison sites, and by the end of the trial, I have none. And if something happens in the external world that could affect my outcome, especially late in the trial, I could be in a lot of trouble if I've used a stepped wedge. It doesn't happen in the parallel design because we have comparison sites throughout. The other issue that's very important in step wedge designs, and it's only become known to us in the last 2 years, is that the standard methods for evaluation of step wedge designs assume that the intervention effect has a rapid onset and is persistent, does not change over time. If you use those standard methods to evaluate a step wedge trial where the intervention effect varies over time, maybe it gradually develops, peaks, and then declines, you can have wildly inaccurate point estimates and standard errors, again, with the standard methods for step wedge trials. The issues that I want to talk about most begin here. So we randomize individuals to study arms. Starts just like a regular RCT. The catch is that the methods that are used to deliver the intervention jeopardize the independence of the observations on my patients or on my participants. There are two common ways that this can happen. The first is that there's some group-formatted component that's part of the intervention. Let's say we randomize people to smoking cessation intervention or usual care. In the smoking cessation intervention, participants get together on Tuesday night, or others get together on Thursday afternoon, others get together on Saturday morning to get part of their intervention in a group format, often a small class to teach skills to help them quit smoking. That's an example of a group-formatted component. Those sessions could be in person, they could be virtual, doesn't matter. The participants are interacting with one another within the same arm post-randomization, and that's what jeopardizes the independence of the observations. The other intervention delivery technique, which is actually even more common, which can jeopardize the integrity of the individual randomization, is the use of shared intervention agents. If you've been listening today, you've heard repeated references to counselors or other behavioral interventionists who are delivering some part of the intervention. Could be a coach, could be a counselor, could be a therapist, could be the instructor of the small groups that we were just talking about. These are all examples of what we call shared intervention agents. They are people delivering parts of the intervention who interact with more than one participant in the same arm of the trial. It's interacting with more than one that makes them shared. They're delivering part of the intervention, so they're intervention agents. We need to pay attention to those kinds of activities in the design of the trial, certainly in the sample size calculation, and absolutely in the data analysis. And I can tell you that most studies that include these components ignore those issues altogether. The result is an inflated type 1 error rate. Most investigators are unaware that that's even a part of what they're publishing. We call these trials individually randomized group treatment trials. They're also sometimes called partially clustered designs. Now, we looked at alternative analytic models for these kinds of trials across a variety of conditions that are seen, and I think we skipped a slide. There we go. The impact of clustering on the analysis for these individually randomized group treatment trials. Participants in the same arm at the beginning of the study are independent. They've never met, no interaction. They haven't dealt with a shared intervention agent. Over the course of the study, their observations become correlated as a result of the group formatted components or interaction with the shared intervention agents. The impact of that correlation depends on the nesting structure and the membership structure. I'm not going to go into detail on either of those. I just want to draw your attention to the fact that it matters whether you have intervention agents or small groups in both arms, just in one arm, whether the intervention agents are working in both arms or just in one arm. These are the kinds of nuances that make a big difference. And John Moyer published a paper earlier this year looking at a variety of analytic models for these trials, and the key points are summarized here. If you've got small groups or shared intervention agents just in one arm, and they're not operating at all, for example, in the control arm, the interclass correlation has an effect that's quite similar to group or cluster randomized trials. You really have to pay attention to it. It can hurt power, can certainly increase the type 1 error rate. If the shared intervention agents are working in both arms and dealing approximately with the same number of intervention participants as control participants, that's the magic solution that we look for with this kind of trial, because it turns out that the effect of the ICC is quite small, really negligible. So if it's at all possible to balance your shared intervention agents across arms, that's desirable. Either way, it's very important to document the interactions that occur among participants in the trial, between participants and shared intervention agents. If you don't keep track of those things, you can't deal with it effectively in the analysis. We always recommend a power analysis with realistic and data-based estimates. Much more difficult in this kind of trial than in the standard RCT, because you often don't have an estimate of the interclass correlation and have to figure out how to get one. How do you choose among these designs? Is there a strong rationale for randomizing groups rather than individuals to study conditions? Hopefully the answer is no. And then you can consider the next question. Are you going to give the participants some of their intervention in a group format or through shared intervention agents? If the answer is no, good, you've got an RCT. That's the most efficient and most powerful approach. Highly recommend it. However, if you're planning to deliver some of your intervention in a group format or through shared intervention agents, then you've got an IRGT, and it's much more complicated, and you need to attend to the issues that I've just been talking about. If there is strong rationale for randomizing groups rather than individuals, and usually the reason, the rationale is, I'm really worried about contamination. If I don't have everybody in the clinic getting the same intervention, they'll talk to one another and there'll be contamination and it'll spoil the study. So if there is a strong rationale for group randomization, then we consider a follow-up question. Is there a strong rationale for rolling the intervention out in all the groups before the end of the trial? If the answer is no, that's my preferred answer, parallel group randomized trial. If the answer is yes, you've got a step wedge design. So these are three questions that will lead you to one of the four designs. As I said, I strongly prefer the RCT, but I spend most of my time working on the others. Jerry Cornfield published this in a paper in 1978, randomization by cluster accompanied by an analysis appropriate to randomization by individual is an exercise in self-deception. I love the understatement. You've all been warned, so don't do that. We need all of these designs. If we can use an RCT, it's the best comparative design available. The IRGT is best used when you've got a need to deliver some of the intervention through shared intervention agents or in a group format, and often it's logistics that drives that. It's far more efficient to deliver the intervention in groups than it is one at a time. And if you're delivering it by people, one at a time, you can't have a separate intervention agent for each participant in the trial, so you have shared intervention agents. The group randomized trial is the best comparative design whenever you want to evaluate an intervention that manipulates the social or physical environment or can't be delivered to individuals without substantial risk of contamination. And then the step wedge design, my least favorite on the list, only if I can't avoid it, is my recommendation. I want to close with some resources. The one at the bottom is perhaps the most important. Anyone who's considering design of a trial or thinking about analysis of data from a trial where you've delivered some of the intervention using group formatted components or through shared intervention agents, or where you've randomized groups or clusters to start with, the Research Methods Resources website, which is an NIH website, provides material, certainly does for group randomized trials and IRGTs, but it also covers step wedge designs. There's a sample size calculator for all three of those, not just for GRTs. Lots of background, lots of key references, so it's an important resource for people that are dealing with these trials. These methods are most common when there's a behavioral intervention involved. So if you're doing strictly a drug trial and you're not going to have coaching or counseling or anything else like that as part of it, you've probably got an RCT. But as soon as you weave in the behavioral intervention components, you often are dealing with one of these other designs. Thank you.
Good afternoon, everyone. My name is Jeffrey Siegel. I'm Office Director for the Office of Drug Evaluation Sciences in the Office of New Drugs in CDER at the FDA. I'm co-moderating this session with Dr. Sokolovsky, and I'm going to be leading the panel discussion. For our panel discussion, we've asked two individuals to join the four speakers that you just heard from. The first individual is Dr. Megan Piper, who you met earlier, who spoke in the previous session. Dr. Piper is a professor in the Division of General Internal Medicine within the Department of Medicine. She's a UW Chair for Tobacco Research and Intervention Research Professor and a co-director of research at the University of Wisconsin Center for Tobacco Research and Intervention. The other individual who's joining the panel is Dr. Nancy Rigatti. Dr. Rigatti is a professor of medicine at Harvard Medical School and associate chief for academic advancement in the Division of General Internal Medicine at Mass General Hospital. I'd like to ask each of the new individuals joining to present for perhaps 5 minutes on our general topic of the regulatory path forward. And could you begin, Dr. Piper?
Happy to. Can folks hear me? You're right on top of this, okay. So I think this issue they were fantastic presentations because I think what they each did is they got into a level of detail that sometimes when we're thinking clinically or we're thinking about how best to make it work, we don't think about the intricacies of study design, the importance of making sure that the outcomes are patient, relevant to the patients as well as relevant to the regulators, thinking about how we're going to capture the data. So one suggestion was looking at EHR data. Anybody tried to look at EHR data? It's a challenge, but we're doing it more and more, and I feel like there's work to be done with clinicians and health systems to see what kind of work can be done to ensure the reliability of EHR data in a way that perhaps we don't have right now, whether that's training a health system or a clinic setting before you go in to get quality baseline data and making sure you get the appropriate outcome data. But I think that's one source of data collection that would be fantastic to have, and it's been utilized in lots of smoking-related research. But again, it needs some work and it needs some thinking about how we're going to collect those data. Another important thing to think about in terms of the patient-reported outcomes, making sure you're picking validated measures. And there was a mention of the PROMIS toolkit. And I don't think I was clear about this earlier, but the Phoenix toolkit is something that was developed to have clear phenotypes across lots of different studies, and so that website has a lot of great, very well-validated outcome measures, patient-reported outcome measures, so that's another place that folks can look for those. And I think one of the other issues that comes into play, again, when we're thinking about methodology and being careful with how we draw our conclusions, comes from this notion of studying different groups. We've had this discussion earlier this morning about the importance of understanding and making sure that interventions work well for lots of different people, and whether that's with respect to genetics, whether that's with respect to, some people's social and environmental exposure to different issues, whether that's with respect to dependence and neuroadaptation that they may have. The challenge becomes, how do we best actually assess whether or not there are group differences? It's hard enough to to recruit enough people and to have sufficient power to just compare two different treatments, right? That's a challenging task in and of itself. When you're trying to look at subgroups, you're going to have to use moderation analysis if you're using traditional statistics. That means you have to make sure that you are adequately powered. How many times do we see moderation analyses or subgroup analyses where their conclusion is there's not a statistically significant difference? It happens all the time, and then we conclude that the treatment works for everybody. But that's an illogical conclusion. If you didn't have enough power to detect the fact that a 10 or 15 percentage point difference was statistically significant, you're missing the fact that you may have something that was clinically significant there. And so I think that's another one of those key methodologic issues that we're challenged by. I've talked to colleagues about possibly using Bayesian statistics. Does that make more sense to help us make clinical decisions, perhaps not statistical conclusions, but again, what can we do with the data that we have so we're not throwing, so to speak, the baby out with the bathwater, but we're getting some sense that there is effectiveness in a variety of different populations, especially priority populations that have tobacco-related health disparities, that are underserved, that are understudied. So I think one other methodologic issue I will raise, and that is the challenge, and I don't have an answer, so if anyone on the panel has an answer, that would be fantastic. But the challenge between balancing the need to understand mechanism and using some of the EMA research strategies I spoke about earlier to understand how cravings and other key mechanisms change over time, how do we balance that with pragmatic trials, where we're trying not to interfere overly or overly complicate or drive withdrawal from study because it's too onerous to participate? How do we balance those two needs to understand what works in the real world so that we can minimize sort of effect attrition that happens with implementation relevant or compared to trying to truly understand what a treatment is doing by examining mechanism. So I struggle with that constantly, and I would love any feedback from the panel members on that. So thank you.
Thanks very much, Dr. Piper. Dr. Ragati?
Hello, can you hear me? So I'm a bit of an outlier on the panel. I'm sort of the clinician who was asked to come and provide my perspectives, both as a clinician and as a representative of the broader healthcare system where these things are treated. And basically what we do is we do the mop-up work, which is to say that for decades what we do is take care of the consequences of people's tobacco use, and they still kill 480,000 people a year and are a key driver of health disparities. Over the last few decades, we've actually done a better job because we've slowly begun to move upstream and address the tobacco use itself and try to help prevent the disorders and the deaths by doing that. But and we're trying, but frankly, we need help. I think my message here is to you all is that we need help. We need new options for treating tobacco use and dependence. It's a critical need. Think about our context. I'm a primary care physician. When I deal with diabetes or obesity, it feels like there's a new drug every month. When I look at tobacco, we haven't had a new drug for 20 years. Why is it taking so long is the question that we think about. We really need more options for people who are using tobacco products because it's also led to what I think people have called a clinical inertia around tobacco in the healthcare delivery system and among clinicians and colleagues. We're just kind of tired of it. There's nothing new. There's nothing to get excited about. And therefore, also what we had heard earlier from Dr. Leone was that our healthcare systems are not supporting the kind of interventions that we need to do as well. So we really do need some new products. And so I want to thank the FDA and NIH for having put together this day, of which I seem to be the last speaker, of putting this together to bring new ideas to the front. I think this is well-timed and exciting, and have learned a lot by listening, and I won't summarize them all for you. But I wanted to highlight a few and pose a few questions. I think what I'm really asking for in the regulatory panel is more flexibility from CDER around efficacy and safety outcomes. We've heard a lot about great alternative outcomes and decentralized trial designs and different kinds of technologies, and a lot of those are used in NIH-funded trials, but whether the people who are developing new medications will use them or new tools are going to be using them when they come to the FDA is the question. The question is, will the FDA, when making decisions, be willing to be more flexible than they have been? Because what I heard was, I really liked the fact that there was the new guidance on nicotine replacement therapies and that it applies. I was glad to hear that it applies to other things. But I wonder, is that going to, it still requires abstinence, that in order to get a drug with an indication, it has to have an abstinence indication, even though there are these secondary outcomes that are really useful. And I think that having those secondary outcomes in the approval could be very helpful at getting doctors and patients something you can market and something you could talk to patients about. Well, you're not ready to quit, therefore you could try this. Or you've relapsed a lot, maybe you could try that. So I think that would help with the clinical inertia. But the question is, do you require every drug to show abstinence, especially if we're thinking about harm reduction as well? And then in terms of safety, again, I think we need more flexibility to acknowledge that the comparison here is not healthy people necessarily. It's people who are smokers and who have a high risk of having severe bad effects in the short term. And so while we have to make sure that our drugs are reasonably safe, we need to accept the fact that our comparison is with a group that's actually likely to have a lot of bad outcomes. And I think that maybe we need to reset that somehow. And I guess at the to end up, I wanted to say a few words about a drug that's trying to get approved called Cytosine, which some of you may have heard about. Let me just say that I have been working as an in my academic role with the company that is trying to get this drug approved by the FDA. It's in the process, and I'm not going to speak about those details. But what I want to do is just sort of say, explain to you why this sometimes has been a frustrating experience, because cytosine is a natural product with a long history as a generic product in Eastern Europe. It has a mechanism of action that's well known. It's very similar to varenicline, which is our best single agent otherwise for smoking cessation. And in the last decade, there have been several new clinical trials done with sort of good quality trials in other countries that have shown efficacy and safety. And so the question has been for a number of us, and it's not just me, a number of us in the academic world who care about the cutting down the number of deaths, is that, how can we get this licensed in the United States? How can we bring this to the U.S.? And it's been a challenge. I'm glad that the company Achieve Life Sciences has been trying to do that, and me and my colleagues have been trying to help them along. But what they did have to do, when I think about it, is the guidance says you need 2 U.S.-based trials. Now, maybe we could be more flexible and say, gee, we had a good one that was done in Poland, or we had a good one that was done somewhere else. Maybe you could have one trial in the U.S. That would have accelerated the process. And then there's the issue of safety, because when the company went back and said that the drug had fulfilled the criteria for efficacy, their response was that there was a requirement or request for additional safety data with long-term exposure. And that makes sense, but this is a drug that's been used for many years in other countries, and a safety signal hadn't appeared. And so it seemed to me that, maybe there's a different way of thinking about the cost of the delay of not having a drug available to U.S. smokers. And I worked with some modeling colleagues of mine to make a little analysis to sort of say what's the cost of a year's delay in having cytosine become available. And, we came up with an estimate of 71, with conservative assumptions, we come up with an estimate of 71,000 people who would have quit if we had had the drug available a year earlier, and that could have led to an increase in over 500,000 life years gained across the population, and that the delay costs 10,000 life years each year does that. So the alternative would have been, could this have been a post-marketing requirement as opposed to a pre-marketing requirement? Now, these decisions have all been made. It's water under the bridge. I'm not suggesting that things change. It's going to move forward the way it's going to move forward. But I think that as new drugs come forward, it would be useful for us to think about, are there ways to be a little bit more flexible in some of the ways using alternative outcomes, alternative measures, the way that you've been describing, for it to move forward, to help us get drugs to the population faster. Thank you.
Thank you very much, Dr. Rigatti. We can't discuss particular products in development, but thank you very much for sharing those concerns and issues. Linda, Dr. Rigatti brought up novel ways to develop new medical products, and you talked about remote data collection. And this morning, actually, we heard about an entire trial that was done with data collected entirely remotely, I think, for a device. Can you talk about instances where we've accepted data from remote trials and what the landscape looks like now with the current guidance?
Sure, Jeff. So I think most trials are a hybrid of remote and site-based data for good reasons. I mean, there are certain things that you can't do remotely in many areas of drug development. But having said that, I mean, there have been trials which have been. You can't hear. Sorry. There have been a couple of trials which have been completely remote. I mean, we always refer to one which was done, I think, in 2014. It was called the remote trial. It was a trial for a drug for overactive bladder. And I think at that stage, perhaps our electronic systems weren't as sophisticated as they were now. They recruited an enormous number, or at least they engaged an enormous number of patients over the internet. But in the end, through all the procedures and so on that had to be done without in-person engagement, they were left with really a very tiny population and barely were able to discern the efficacy of the drug. So I think we've gone beyond that. Now communication strategies are much more advanced. I think telehealth has become pretty much a standard procedure in many clinical environments, so people are comfortable with that. So I think we're likely to see more either completely or largely remotely conducted trials. And I think in the area of smoking cessation, there's an enormous opportunity there because everyone appreciates the difficulty of getting to a trial site. I think we're all busy. We all have other things. There's traffic and there's parking. And so I think there's a lot of motivation to do that. I don't know if that answers the question at all, Jeff.
Thanks very much. We only have a few minutes left, and I'd like to ask a follow-up question for Dr. Piper. You talked in your talk this morning about 6 different areas that were important to understand to see the underlying features that may promote cessation of smoking. Can you talk about how those can be implemented in clinical trials to perhaps subset patients or stratify clinical trials based on different subcategories and understanding the different mechanisms that different drugs might work under?
Sure. I think one of the most important things, and somebody talked about this earlier, just in terms of the development of assessments, right? There's a conceptual model and a measurement model. And I think that same approach works very well when you're conducting clinical trials. So what is your conceptual model? What are the mechanisms or features that you expect to change when people engage in your treatment or use your treatment? And then from there, thinking about what are those constructs, what's the timeline upon which I expect these to have an impact? You know, should craving be suppressed early on? Do I expect to see a reduction in cessation fatigue, but that doesn't really emerge until 2 weeks out? So with a conceptual model that has hypothesized mechanisms and hypothesized timelines, then you can use that to identify now what are the assessments that I would need to use to address each of these. Do I need something that is a comprehensive, fully validated measure? Because let's be honest, those tend to be long. And so those might be something that you do at baseline and then you do at whatever time points you deem might be critical. So perhaps you do those at baseline, you do those at 2 weeks and 4 weeks, 8 weeks and 12 weeks. Or again, making sure your assessments are centered around the time frame in which you expect to see those differences. So then using your measurement model, again, you have to balance the notion of, as much as we'd love participants to answer 200 questions from 7 fully validated measures, we can't. They'll refuse. I've tried and they will refuse. And so I think it becomes this balance of how much of my conceptual model am I actually going to be able to assess in a way that's sufficient for the regulators? And then what can I get that's perhaps not sufficient for regulators, but boy, would be very impactful for people creating guidelines, people who are making clinical decisions, and using that, that approach for what the data can actually do to drive this intervention into clinical practice and utilization. Did I answer that?
You did. Thank you very much. I have so many more questions I'd like to ask, but unfortunately we're out of time. So I'd just like to thank the speakers and the whole panel for this great discussion. I hope that the concepts that we discussed this afternoon will help developers of new treatments for smoking cessation bring their treatments forward in a more efficient and effective way. Thank you very much for your attention.
Thank you to all the speakers for a great session. Now we're going to take a 10-minute break, and then when we return, we'll have time for public comment. Everyone who is assigned for public comment, we have chairs that have your name on it, so if you can just be there maybe 5 minutes before the break ends, we're going to look forward to that, and that will be followed by concluding remarks by Drs. Compton and King. Thank you, everyone. All right. So it's great to see all these discussions ongoing. Obviously, this is a topic that is near and dear to many people's hearts. Next, we're going to move to the public comment period of this meeting. So just some housekeeping issues. The folks who are providing public comments, they're sitting right there. Each of you has 4 minutes each to speak. And what you'll see in the front is my colleague will have that folder right there, which is a yellow folder. Once that is up, that means you have 1 minute left. And when you have the red folder up, that means your time is over. So please keep your comments within the time limit, but we are really, really looking forward to your comments. And we'll just come up right here in order. So come right up.
All right. Hey, y'all. Can you hear me all right? Cool. Hi there. My name is Brendan Wong. I'm the founder and CEO of Capnos with the mission to help 1 million people quit vaping by 2027. I was 14 years old when I first began vaping. I was addicted for over 5 years. Throughout this time, I've come to recognize that the high usage of nicotine has actually evolved into a craving for the behavioral dependence rather than just physiological. I struggled because there was missing tools that were available in the marketplace that youth like myself could access. NRTs were out of my age category, and other solutions were just too expensive and inaccessible. I then became obsessed with finding a solution to address this disparity. I became obsessed with surveying people, asking thousands of people who vape to discover that 2 out of 3 have indicated that the root issue is an oral fixation. From then on, in 2019, I invented the Capnos Zero. It's a flavored pressurized air inhaler that works like this. Inside, there's a silicone valve that pops open that creates a throat hit sensation, but there's 0 smoke, 0 nicotine, and 0 charge. It's flavored with optional natural extracts that are soaked into these cotton wicks that are then perceived through the olfactory system or the sense of smell. This addresses the tactile, auditory, and aromatic sensory dependencies that people are looking for with innovative products to help address behavioral cravings. We then released the Capnos Ligura and the Breezy, which are premium versions. These feature a silicone bite cap, as well as adjustable pressure strength and fidgeting features too. We've priced our value line of products at $24.99 and our premium line at $74.99 so that our products are, one, they're available to last a lifetime to reduce disposable waste, and our products are neither a tobacco product, medical product, food, or drug product, but we do aim to earn cessation messaging eventually. Since launching in January of 2022, we've been able to ship over 70,000 units of our devices to 70 countries and growing. We have a global team. We're also a minority-owned business and registered LGBTQ-owned business as well. In a recent July survey with 379 respondents to folks in the Capnos mailing list, we've been able to discover that of those using our devices, 21% reported a 5 out of 5 rating, saying highly effective in achieving their goals for behavior elimination, reduction, or replacement. We've been able to help pregnant mothers, patients awaiting clinical or medical procedures, and people who have suffered with smoking or vaping addiction for decades long achieve their goals by using Capnos devices. Our goals are inextricably linked with many of those of you here today. So with that, I ask you all to join our efforts. We've recently partnered with colleagues in academic medical centers to evaluate CAPNOS devices in a human laboratory setting. We currently have a grant application currently under review. We aim to expand access through retail adoption, work with health insurance payers and other regulators to achieve mutual harm reduction goals. I ask you to join us by inviting me to speak with your groups, share my story, and to also participate with us in pilot studies as well. It's helpful to get buy-in from folks like myself, so that way representation fosters innovation. I've shared some comments on the public registry, so feel free to reach out there or contact me at hello@mycapnos.com. With that, I ask you all to join our efforts, and together we can put a cap on vaping and smoking. Thank you.
Hello, my name is Dr. Jacobs, and I'm from Achieve Life Sciences. Each year in the U.S., over $300 billion are spent in smoking-related illnesses, in medical care costs, and lost in productivity. As you've heard many times, 480,000 Americans end up dying as a result of their smoking-related illnesses. Cytosine has been a successful smoking cessation treatment for decades in Eastern Europe. However, it is considered a new chemical entity by FDA because it is a novel drug that has not been approved by FDA in any other application. Although the efficacy and safety profile has been well established over the decades, cytosine treatment involves a complex 25-day treatment schedule, and there has been no significant improvements in this older Eastern European product until the efforts of Achieve Life Sciences. Even prior to the IND submission in 2017, Achieve Life Sciences has been redeveloping this older product into an improved novel treatment for smoking cessation. This has been accomplished by revising the formulation, increasing the tablet strength, simplifying the administration schedule, and increasing the treatment duration. This decade-long process of redesigning cytosine, now called cytisinicline, has included NIH support from the NCCIH and NCI for conducting additional non-clinical toxicology studies required for the IND submission, as well as initiating the phase 3 clinical trials. To date, Achieve shareholders have invested over $100 million in the clinical development of cytisinicline. Even though we have demonstrated robust efficacy results with the 2 phase 3 trials already completed, we remain delayed in submitting for market approval due to FDA wanting more safety exposure data. Meanwhile, the less effective form of cytosine has gained expanded approvals in Canada, the UK, Ireland, and many select Western European countries. It has been over 13 years since initial discussions began with FDA regarding cytisinicline's potential to assist Americans in overcoming their dependence on combustible cigarettes. During this journey, Achieve Life Sciences has overcome other significant delays, primarily due to the rigorous non-clinical and clinical trial requirements for getting a new chemical entity approved as a new product to treat nicotine dependence. Frustratingly, the approval process of cytisinicline as a smoking cessation treatment appears to be far more stringent than for other products that perpetuate nicotine addiction. We urge FDA to address these discrepancies in the approval process, which appears to be allowing recreational nicotine products to enter the market with comparable ease, while seemingly hindering approval for crucial treatments aimed at helping individuals overcome their nicotine dependence. In the past decade alone, FDA has authorized the marketing of over 40 new tobacco products in the U.S., yet there have been no new FDA-approved prescription treatments for smoking cessation in nearly 2 decades. It is crucial that the discrepancies in the approval process for recreational nicotine products versus potential life-saving cessation treatments are rectified. I'd like to thank you for speaking on this issue.
Hello, everyone, and thank you for the opportunity to present the view of the American Heart Association. I'm Jeffrey Willett, National Advocacy Vice President for Integrated Strategy at AHA. Previously, I directed multi-million-dollar statewide smoking cessation programs in Ohio and New York. So I've spent quite a bit of time thinking about this issue and, in a very applied population-based context. So first, we want to thank FDA and NIH for holding this important meeting and for the perspectives shared last week in the article published in the Annals of Internal Medicine. And to respond to the 4 discussion questions that we were given, first, we share your focus on diversity in clinical trials and believe this is particularly important in smoking cessation, where patterns of use are increasingly concentrated in underserved communities. In 2022, AHA, with support from Pfizer, Gates Ventures, and Care Access, launched a $20 million 4-year strategically focused research network titled The Science of Diversity in Clinical Trials. This effort is funding researchers across 12 universities and health systems to identify best practices that increase clinical trial enrollment from traditionally underrepresented populations. We believe this research collaboration is yielding important insights that can inform cessation trial design and are happy to share those. Regarding your question about a patient-focused drug development meeting, we believe FDA should hold a PFDD meeting to hear directly from current and former smokers. We know people who smoke want to quit, and while each year more than half try, fewer than 1 in 10 succeed. So as a result, millions of Americans can share with you the barriers and challenges with quitting. People who have experienced the smoking cessation journey can share their personal health goals and intrinsic motivations to quit, as well as their views on acceptable risk for therapeutic products when the alternative is a product that regularly kills its users. On average, smokers make 7 quit attempts before they're able to quit for good. Feedback from current and former smokers is critical to identifying potential efficacy endpoints that may not entail complete abstinence, but would substantially improve their health or substantially increase their probability of eventual cessation. And with regard to questions about novel targets and challenges to drug development, we certainly applaud your exploration of novel targets, and we would add that we also believe the following changes to smoking cessation drug product reviews are necessary. First, due to the tremendous risks with continued cigarette use, FDA should adjust its risk-benefit analysis for new smoking cessation therapies. Using continued smoking as the reference point, FDA should carefully assess how its existing considerations for addiction potential and toxicity may constrain the approval of products that would have tremendous benefit to both individual and public health. Second, a key challenge for researchers and drug developers is the time required for new product evaluation. FDA should fully utilize its expedited programs and other mechanisms to facilitate and incentivize smoking cessation drug reviews. And third, we already know a great deal about nicotine, particularly that FDA-authorized NRT products pose minimal risk and can help smokers quit. That said, the existing array of NRT have not proven particularly popular or sufficiently effective. With many competing forms of commercial nicotine delivery available to sustain addiction and undermine cessation, FDA must emphasize therapeutics approaches that smokers will actually use. And finally, there's little economic incentive for drug manufacturers to develop therapeutic products when new and increasingly popular commercial tobacco products can be so easily introduced to the market regularly without FDA pre-market authorization. So it's in the public's interest to ensure that efficacious new therapeutic products are identified and introduced to the market quickly, that all unauthorized commercial tobacco products are removed from the market, and tobacco product standards are established to more rapidly reduce combustible cigarette use. This is an imperative that must be embraced across FDA. So thank you for the opportunity to share the views of the American Heart Association.
Good afternoon. Thank you for holding this meeting, and thank you for the opportunity to give these brief remarks today. My name is Stacy Younger-Gagosian, and I'm the Senior VP of Public Policy at Truth Initiative. Truth Initiative is a national nonprofit public health organization committed to a future where commercial tobacco and nicotine addiction are a thing of the past. Our mission is to prevent youth and young adult nicotine addiction and empower quitting for all. As has been pointed out, there are still 30 million adults who smoke and millions of youth and young adults who use other tobacco products, such as e-cigarettes, all of whom are clamoring for help to quit their addiction to nicotine. This meeting is an excellent step in the right direction in exploring the research needs for improving cessation treatments and delivery options. However, much more needs to be done. In my short time today, I will highlight 5 areas that need attention. We will submit written comments that expand on each of these areas. First, current cessation treatments are insufficient, and much more needs to be done to improve pathways to market for cessation interventions. There has not been a new cessation treatment approved for tobacco in decades. While it has been exciting today to learn of some new research on novel tobacco cessation treatments in the works, much more needs to be done. Truth Initiative has joined other public health groups in making recommendations to FDA and CDER on things that could be done to facilitate the process for bringing safe and effective tobacco cessation products to market. And we will submit these specific steps in our written comments, some of which were highlighted by the previous speaker. Second, we need to ensure that research and treatments are not only for smoking cessation, but for all nicotine products. While smoking cessation is critical to address, we cannot only focus on combustible cessation. It is important to remember that nicotine is the reason that people smoke, vape, dip, and use other nicotine products. While cigarette smoking and the combustion of tobacco is the cause of most of the smoking-related disease, nicotine addiction itself is also a health harm. Furthermore, youth and young adults are using e-cigarettes and other nicotine products more than they are smoking. We know from our experience in working with young people that they are desperate for proven methods to help them quit vaping. While a stepwise process may be helpful for some smokers, meaning moving smokers completely to less harmful products, may be a step in the quit journey. It should not be viewed as the final step. The goal should be to quit all nicotine products altogether. FDA, NIH, and other agencies should ensure that cessation treatments can apply to all nicotine products. 3, we need a whole government approach to cessation. Today's meeting is a good start, but it really only includes 2 federal agencies. FDA and NIH are very important agencies to tobacco cessation, and we are very glad for the work they do. But cessation needs to be broadened to the entire government. We need to see research and action from a much broader group of federal agencies, from CMS to the VA and DOD, in addition to NIH and FDA. For example, CMS could hold demonstration programs to explore how best to ensure participants have access to counseling and treatments. They could also explore changing who can be reimbursed for tobacco cessation treatment and counseling. The VA and DOD could be conducting research on treatments that work for their population specifically, and how best to make those treatments easily accessible. That leads to my next point, which is we need to ensure that populations disproportionately affected by tobacco and nicotine products receive treatments that work best for them. This would include youth, those with mental health issues, those serving in the military, and importantly, LGBTQ+ and minority populations that use tobacco at disproportionate rates. This was discussed importantly at a panel earlier today, and more needs to be done. Finally, we need to expand coverage of all cessation treatment and ensure that effective technology-based interventions are covered and available for all who need them. In the interest of time, I will leave that alone, but thank you again for holding this meeting and allowing public comment. We hope that the next steps will, that more steps will be taken soon to address shortcomings in tobacco cessation and ensure that all those who are addicted to nicotine can overcome their addiction and live their healthiest lives. Thank you.
Hello, my name is Anna Giulio. I'm the Senior Director of Nationwide Tobacco Cessation and Health Policy at the American Lung Association. Thank you for the opportunity to speak and representing the Lung Association. The American Lung Association believes that anyone who uses and is addicted to tobacco products can quit, not just switch to another tobacco product. Our mission is to save lives by improving lung health and preventing lung disease. And one of our 4 strategic imperatives is to create a tobacco-free future. Helping people quit their addiction to tobacco is both integral to our mission and is of the utmost national importance. Almost 500,000 people die each year from a tobacco-related disease, and over 16 million people live with one of those diseases in our country today. And while new data shows a decline in tobacco use among youth, 10.1% of high school students, or 1.58 million high school students, use tobacco products. These numbers show an urgent need for more FDA-approved tobacco treatment for adults and efficacious FDA-approved cessation treatment for kids. Data shows that approximately 2/3 of adults who smoke want to quit. This holds true across demographics. Unfortunately, these data also show that people who smoke did not frequently use medication in their quit attempt and ultimately were not successful in quitting. While the desire to quit is consistent among adults in the United States, despite differences in things like socioeconomic status, education, and race, the burden of tobacco use is not even. For example, lower-income people in the United States use tobacco at a significantly higher rate than their wealthier counterparts. FDA and NIH need to understand the disparities that exist related to tobacco use and ensure that any action taken reduces these disparities and does not exacerbate them. The Lung Association urges the FDA to take the following steps. First, the FDA can consider new indications for cessation products and can consider labeling changes for existing cessation products. These can include combination use, longer-term use of existing cessation products, and pre-quit use of cessation products as part of a reduce-to-quit regimen. FDA should take affirmative steps to explore alternative to long-term clinical trials for promising new cessation products or for new indications for existing cessation products where sufficient evidence is available to meet both safety and efficacy. Third, FDA needs to establish criteria for accelerated pathways for approving promising cessation drugs. This is especially important given the fast-changing landscape of commercial nicotine products, including e-cigarettes and pouches. And fourth, FDA should consider modifying its organizational structure by moving cessation products from CDER's Division of Anesthesiology, Anesthesia, and Addiction to another office, such as one that has experience with evaluating treatments caused by tobacco use, such as the Office of Hematology and Oncology Products. But that's not all. NIH is also here, and NIH has an important role to play as well. The Lung Association, among other things, encourages NIH to prioritize research on effective cessation treatment for youth. Tobacco use is unsafe for kids, and unfortunately, many are currently addicted to tobacco. NIH has an incredibly important role to play in this space. The dangers of not doing anything is millions of people in the United States, including kids, will continue to use tobacco products and suffer the death and disease they cause. Action from today's meeting by FDA and NIH could help realize the promises of the President's cancer moonshot and address the epidemic of tobacco that our nation faces. Thank you so much, and there's going to be more in our written comments.
So I can see you and you can see me. Hi, everyone. My name is Meg Reardon. I'm Vice President for Research at the Campaign for Tobacco-Free Kids. I want to thank FDA and NIH for hosting this important meeting and for the opportunity to participate. It's terrific to see NIH and FDA collaborating in this way. It's well past time for significant review of the content and manner in which CEDAR addresses the entire issue of tobacco cessation and its role in curtailing the number of Americans who die from tobacco use. The public health community has repeatedly called on CEDAR to review its approach to smoking cessation products and to support the development of effective cessation products more aggressively. The need for review and action is as compelling as ever. More than 28 million Americans still smoke, and large disparities still exist in terms of who smokes and who suffers. Far too few smokers are able to quit successfully. The large majority of smokers want to quit, but current medications are not giving them the help that they need. The need for innovation in the use of existing approved therapies and in the development of new therapies is clear. The fact that no new cessation products have been approved in the last 20 years underscores the need for FDA to make change. Right now, we are in an illogical situation where manufacturers who want to market products to help smokers quit and promote these products to smokers face a regulatory system that has discouraged innovation. Approval for cessation drugs has been too costly, too time-consuming, and restrictive. By contrast, manufacturers who market a wide variety of e-cigarettes and other nicotine products to as many consumers as possible without regard for whether these products help smokers quit have flooded the market with their products. Virtually all of them are illegal and unauthorized tobacco products. FDA must take strong steps to complete pre-market review of e-cigarettes and enforce the Tobacco Control Act against products on the market without authorization from CTP. It's critical that CEDAR and CTP work together to change the incentive structure to enable the right products and only the right products reach the market. Those are the products that actually help smokers quit without threatening to addict a new generation to nicotine. Public health advocacy groups and leading smoking cessation experts have long advocated various concrete steps that CEDAR could take to create a regulatory environment more conducive to innovative smoking cessation products. I'll highlight just a few in the interest of time, and we'll submit longer comments to the public record. But first, in considering the risks versus benefits of new cessation drugs, FDA must compare the risk posed by the cessation therapy compared to the risk posed by continued smoking. CEDAR should explore alternatives to long-term clinical trials for promising new cessation products without compromising the safe and effective drug standard. CEDAR should proactively reach out to companies interested in developing smoking cessation products to discuss existing barriers to innovation and to discuss creative ideas for incentivizing development of new cessation products. FDA should encourage the development of cessation therapies in pediatric populations. There's a significant gap in the availability of cessation products with labeling for use in pediatric populations. While yes, smoking has gone down dramatically among youth, which is a great thing, we still see higher use among some subpopulations of youth. FDA needs to do more to encourage the development of drugs, cessation products,
that are effective for and available for these populations. There's also a huge problem, of course, of youth addicted to e-cigarettes. Certainly more needs to be done to help youth addicted to nicotine. In conclusion, I'll say that FDA has a critically important opportunity to dramatically reduce tobacco-related disease and death. But this will only happen if there is coordination across the agency. Both CTP and CEDAR need to work together. They need to make smoking cessation a priority. Smoking cessation, the priority being driving down smoking rates and making sure that every smoker is provided the best possible assistance with quitting. Thank you again for holding this meeting, and I appreciate the opportunity to speak on this important issue. Thank you.
Hello, my name is Ryan Corey. I'm CEO of CTT Pharmaceuticals. I want to thank the FDA and NIH for putting this together. The World Health Organization states the tobacco epidemic is one of the biggest public health threats the world has ever faced, killing over 8 million people a year around the world. In the United States, more than 480,000 Americans will die this year, or 1,300 Americans per day. And more than 16 million Americans are living with diseases caused by smoking. Before I speak about our technology, I want to share with you how smoking has affected my life. I lost my grandfather, grandmother, and uncle to smoking. My grandparents both had COPD, and my grandmother never smoked. My uncle developed bladder cancer from smoking, and towards the end of his life, he would call cigarettes cancer sticks. I was a third-generation smoker and smoked a pack a day for almost 7 years. I started smoking at 18 years old because I looked up to someone that smoked. I developed foot problems from smoking that has required multiple surgeries. What I discovered as I got older is that smoking can cause tendon issues. I became CEO of CTT Pharmaceuticals, a company who has the ability to put a small amount of nicotine on a dissolvable strip, also known as microdosing. Our patented technology, which incorporates micelle delivery, was invented by Dr. Pankaj Modi. Dr. Modi, who is the founder of our company, also invented an insulin spray that was proven to be as effective as an insulin injection and was later bought by Johnson & Johnson. Like most small companies, a lack of funding has prevented us from making our technology available to smokers. This is why I'm grateful for the NIH giving opportunities to a company like ours by obtaining grants for smoking cessation technology. And because of these grants, CTT Pharma partnered with Johns Hopkins University this year to achieve a grant for human trials utilizing our technology. We believe using our micelle delivery allows and offers for a faster dissolving, better absorption product, while giving consumers looking to quit potentially 50 to 75% less nicotine per serving compared to Nicorette gum. Nicorette gum has helped many smokers quit, including myself. However, there's a segment of Nicorette users that use this product as a daily use replacement to smoking and vaping, which now goes into a category of other nicotine products that stay in the mouth for excessive amounts of time, which comes with its own set of problems. The goal for an oral nicotine cessation product, and frankly all nicotine products, should be to get the nicotine in and out of the mouth as quickly as possible. I believe that our technology would meet this criteria. I'm hopeful that by developing innovative products like ours, that could lead to new laws that would change the landscape of the tobacco industry, as smoking is self-harm to one's body. And again, I want to thank everybody at the NIH and the FDA for putting this together. Thank you.
Good afternoon. My name is Brian Quigley, and I'm the CEO of Kenovia Incorporated,
a 5-employee drug development company based in Richmond, Virginia. We are developing a non-flavored prescription inhaled smoking cessation therapy and are advancing this combination product through the FDA and the UK's MHRA. Recently, Cunovia received IND clearance from FDA to begin U.S. human clinical studies. I would first like to thank FDA and NIH for creating this forum. I'm encouraged by the opportunities very recently outlined by FDA and NIH for pursuing innovation in smoking cessation. A modernization of the approach to advancing new smoking cessation medicine is overdue, and I'm hopeful these recent views will allow us to improve outcomes for smokers who want to quit smoking. Today I will discuss 2 topics. First, I will discuss inhaled nicotine replacement therapy as a novel medicinal treatment option, and secondly, I will share opportunities to address challenges in advancing novel therapeutic candidates. Inhaled delivery of nicotine as a safe and more effective NRT medicine is not only possible, but is also needed to be part of the toolkit available to patients and practitioners. A cigarette starts to deliver nicotine in seconds and achieves a peak spike in minutes. Existing NRTs that deliver nicotine buccally or transdermally can take an hour or more to deliver the drug and never deliver enough of the drug to provide relief from cravings and withdrawals. We believe an NRT that safely delivers pharmaceutical-grade nicotine using the pulmonary route of administration can address the massive shortcomings of the existing products. Yes, nicotine can be addictive, but its safe use as a pharmacotherapy has been well documented for 40 years. Yes, an NRT delivering nicotine via inhalation raises new safety questions compared to existing NRTs. However, it is important to balance the safety analysis of novel treatments against the status quo of 28 million smokers continuing to smoke. This leads me to the challenges drug developers face today. Identifying and developing novel therapeutics requires risk-taking and investment by the sponsor, but it also requires a regulator who brings a genuine public health approach to solving the problem, one that reflects the urgency of addressing the unmet need for patients and the ongoing harm caused by perpetuating the status quo. I am hopeful today is the beginning of a new path forward where FDA has genuinely evolved from a history where conservatism regarding real issues such as abuse liability, inhalation, and nicotine safety were held out of balance with the reality of the ongoing toll of combustible cigarette use. We have seen FDA take bold steps to promote innovation in other areas of treatment. One example was the October 2023 guidance for treatment of stimulant use disorder. In this guidance, FDA proposed using change in pattern of stimulant use as an appropriate endpoint to promote innovation. However, for NRT drug development, we continue to point to abstinence after treatment as the primary endpoint of efficacy. Yes, abstinence is our common goal, but abstinence in the clinic does not reflect the hard reality smokers face when attempting to quit in the real world. While I'm encouraged to see that last week, for the first time, FDA pointed to other possible endpoints for smoking cessation, we must continue to engage to identify the most appropriate application-specific endpoints to change the status quo. Today, I'll propose 3 ways the agency can engage with sponsors to make this change happen. First, we ask the agency to evaluate clinically meaningful changes in patterns of cigarette use as endpoints, acknowledging these can lead to achieving abstinence over time. Second, the agency should lift the restriction that limits the use of identified secondary endpoints to only the decades-old approved medicines, so all novel candidates can demonstrate effectiveness in supporting individuals seeking to quit in innovative ways, to use FDA's own words. FDA should also evaluate all the tools to monitor long-term efficacy in the real world rather than relying on long-term monitoring in the clinic, because this can include guidance on the use of phase 4 surveillance and improve efficiency of phase 3 trials. Today's meeting represents an important step in changing the status quo, but this discussion is urgently needed because without change in the U.S. regulatory process, the fact is the novel NRT being advanced by our company will be approved in the United Kingdom years before it is approved in the U.S. We are proud to be engaged with FDA in this process and are committed to working with you to making change happen. Thank you.
Good afternoon. I'm Miriam O'Day, and I'm Senior Vice President for the American Association for Respiratory Care. AARC was founded in 1947 and today is the leading professional association for respiratory care in the world. At AARC, we focus on professional excellence, advancing the science and practice of respiratory care, and advocacy for patients and their families, the public, the profession, and the respiratory therapists. Supporting more than 42,000 members worldwide, the AARC is a not-for-profit professional association headquartered in Dallas, Texas. Thank you for the opportunity to discuss the critical role respiratory therapists can play in supporting the FDA's tobacco control efforts, particularly in smoking cessation research. RTs are on the front line of patient care, treating individuals with respiratory diseases often caused or worsened by smoking. This gives them valuable insight into smoking cessation, clinical trials, and tobacco control strategies. To increase enrollment of underrepresented populations in clinical trials, such as racial minorities, LGBTQ+ individuals, and rural residents, RTs can be a valuable resource. RTs are well positioned due to the trusted relationship they hold within these communities. RTs build trust with all patients through direct care, allowing them to act as a bridge between researchers and potential study participants. RTs also can play a role in promoting patient-focused drug development meetings. They can encourage patients to share their experience with smoking cessation and provide professional insights into treatment challenges, adherence issues, and possible new trial endpoints, such as improvements in lung function and quality of life. When it comes to developing smoking cessation products, RTs offer unique perspectives on behavioral and psychological factors, including stress reduction and habit replacement strategies. RTs also recognize the potential of integrating digital tools, with many utilizing mobile apps, artificial intelligence, and telehealth platforms to provide real-time support and improve patient outcomes. One of the main challenges in identifying new targets for smoking cessation is recognizing the need for personalized approaches. RTs understand the important compliance and adherence issues patients face, whether due to side effects, complex regimes, or lack of immediate results. Their insights can also help simplify therapies and improve education. RTs can also advocate for therapies that address both nicotine addiction and comorbid conditions like COPD and cardiovascular disease. In addition, RTs regularly observe the unmet needs in current cessation options, such as insufficient support for dual users of tobacco and electronic nicotine products. Many of our patients have conditions such as COPD or cardiovascular disease, which can complicate smoking cessation, and RTs can be critical advocates for therapies that address both nicotine addiction and these coexisting conditions. RTs are essential in advancing tobacco control efforts. Not only is the product or intervention important, but the individuals who are involved in coaching and supporting and educating those patients. Their ability to engage diverse populations, offer patient-centered insights, and identify therapeutic targets, which make them invaluable partners in shaping more effective, patient-focused smoking cessation strategies. The AARC is here today to encourage the FDA, NIH, and sponsors to think of the role that RTs can have in helping to solve this problem. We have submitted a full comments for the record that go into additional detail. Thank you for including us in this meeting today.
Good afternoon. I'm Robin Gougelet, and I'm the Vice President of U.S. Regulatory Affairs at Juul Labs. On behalf of the company, I appreciate the opportunity to address the important question of how to spur meaningful innovation to reduce death and disease caused by combustible use. Ending smoking requires an all-of-the-above approach. This includes increasing access to proven pharmacotherapies, new medicinal options, behavioral support, and the access to products that deliver nicotine without smoke regulated by CTP. This includes electronic nicotine delivery systems, or ENDS, more commonly known as e-cigarettes, like the Juul system. At Juul Labs, our mission is to transition the world's billion adults who smoke away from combustible cigarettes, eliminate their use, and combat underage usage of our products. And we believe that our products are benefiting public health. Over 2 million adults who had smoked switched using Juul. This includes many individuals who had no intention to quit, who ended up switching completely, also known as accidental switching. The premise is simple. As adults who smoke try e-cigarettes, they began substituting vaping for smoking if they find them to be satisfying alternatives. Over time, we see increased abstinence. The success that we've seen with Juul products in the real world is likely due to the superior nicotine delivery compared to NRTs, coupled with similar rituals to smoking. It may also be due to a different mindset than smoking cessation. With NRTs and other pharmacotherapies, people who smoke must be committed to quit completely, abruptly, immediately, and forever. It requires them admitting they need medical help, and many people who smoke do not see themselves as sick. Any slip, a cigarette while out with friends or during a stressful time, is seen as a failure, and over 90% of these lapses lead to relapse. With Juul, there does not need to be a defined goal. This appears to be a promising intervention occurring in the commercial nicotine space for adults who smoke who would not otherwise quit using nicotine. Last week's commentary of internal medicine by senior leaders at FDA and NIH highlights the public health potential of e-cigarettes. The authors note many adults who smoke have used e-cigarettes to quit smoking. We also appreciate their overview of the requirements of the clinical development program for medicinal inhaled nicotine product. From our perspective, CDER created barriers to prevent us from pursuing a medicinal product. The substantial preclinical assessments required for inhaled nicotine products before they can even be subjected to clinical trials are particularly cumbersome. Preclinical burdens for novel compounds are understandable and appropriate in a new drug context, but this situation is admittedly unusual. E-cigarettes have been used by millions of adults who smoke and have already demonstrated superior effectiveness for smoking cessation. The Cochrane Review did not find evidence of substantial harms, though more data are needed to assess longer-term health risks. Although this situation is unique, it's important to consider the urgent need to appeal to the 28 million Americans who still smoke. As we contemplate the public health burden of combustible cigarette use and the wealth of real-world evidence on e-cigarettes, we urge CDER to be more flexible in its requirements. It's important to note, as a potential sponsor of such a program, we must consider risks, including delays to the development of cytosine, the lack of approval for a nicotine mouth spray already approved in dozens of countries. Given the significant resources we would need to invest in this program, the systemic risks are sobering. Were FDA to revise these preclinical requirements or even consider shifting them to later in the development program, JLI would be in a much stronger position to allocate the resources necessary to pursue such a program. Absent substantial changes to these requirements, a smoking cessation indication through CDER is not tenable as compared to other steps we are taking to reduce the burden of smoking.
Good afternoon. My name is Catherine Olcani, and I'd like to begin by expressing my gratitude to the FDA and NIH for hosting this important meeting on advancing smoking cessation. One of the key topics in today's meeting is the importance of bringing new perspectives into product and drug development. As both a youth advocate and someone who personally struggled with nicotine addiction from a young age, I want to emphasize a few critical areas. We are facing a generational shift with the rise of e-cigarettes and nicotine products targeting young people. For millions of youth, their first exposure to nicotine comes not from traditional cigarettes, but from newer, more insidious products marketed to appeal to them. A 2023 study by Imperial College London found that among youth who exclusively used e-cigarettes, 14.3% transitioned to cigarette smoking within a year, a number that rose to 25% just after 3 years. This alarming trend shows that e-cigarette users were more likely to transition to cigarette smoking compared to those who had never used nicotine products, highlighting the increased risk associated with starting e-cigarettes. The study also found that this transition was most common among youths aged 14 to 17, a group more prone to experimenting with e-cigarettes and subsequently moving to cigarette use, creating a new generation of smokers. Current cessation products such as gums, patches, and lozenges often carry stigmas due to poor user experiences. Reported factors such as unpleasant taste, cumbersome techniques, and lack of discretion deter users from completing the treatment course, resulting in lower quitting success rates, especially among younger generations who view them as outdated and ineffective. We need to rethink how we approach cessation for these groups. One of the biggest challenges for researchers and developers of smoking cessation therapies is navigating the complex and lengthy regulatory approval process in the United States. From a business perspective, the high cost of bringing a new cessation product to market, historically amounting to hundreds of millions, combined with regulatory uncertainty, offering no clear timeline or assurance of approval, discourages investment. A 2017 British Medical Journal study found that countries with easier regulatory pathways for approving new therapies tend to see quicker adoption of novel solutions and better health outcomes, while the slower U.S. process limits the availability of effective therapies and contributes to lower quit rates. The challenge is not just in identifying novel therapeutic targets, but in ensuring these innovations can efficiently navigate the regulatory framework to meet urgent public health needs. Streamlining regulatory pathways would accelerate the availability of effective products, reduce smoking rates more quickly, and help the U.S. keep pace with global advancements in cessation therapy. More than 11,500 vaping products flood U.S. store shelves, yet there are few CTP-approved vapor products. We cannot afford to delay in addressing this disparity, as it should not be easier to access harmful nicotine products than life-saving cessation tools. In conclusion, the challenge before us is clear. We must reduce smoking rates in this country through regulatory reform, and we must do it with urgency to protect future generations. On the same token, I urge the FDA to accelerate its regulatory pathways for new cessation products so that more companies are encouraged to utilize the CEDAR pathways to have products marketed as smoking cessation products. The regulatory process through CTP or CEDAR does not fully reflect the urgency of public health crisis we face. We must balance rigorous safety standards with the need to get effective products to market faster, which will be critical for reducing smoking rates faster and saving lives. Thank you for the opportunity to speak today, and I look forward to the continued collaboration between the FDA, NIH, and all stakeholders as we work toward advancing smoking cessation and improving the health of all Americans.
Thank you to everyone who offered public comments, and thank you to all the folks who are here in person and on Zoom. I do want to reassure you that this meeting is, I think, a reflection of the fact that this is an area that FDA is taking incredibly seriously, and I really feel that this meeting has been really, really important and really, really informative, and I hope that we will continue to make progress in this space. Now, to offer concluding remarks, I would invite Dr. Wilson Compton and Dr. Brian King to come up to the stage and wrap this up.
Well, thank you very much. It's certainly been a wonderful day to learn so much from one another about how to both take advantage of some of the new targets that we highlighted in our NIH presentations, as well as new methods, new statistical approaches, as well as potential changes to the regulatory framework. It's been a very exciting day for me to learn so much about these areas. And even as somebody who knows this area reasonably well, I've learned a lot today. So I really appreciate the opportunity to learn from one another. Really want to take a moment to thank the NIH and FDA committee members that really helped organize the day and brought together such a diverse group of perspectives from two different agencies that have a different role, but by working together, we think we can improve the health of the population. And after all, that is all of our goals in the public health services across government. Please join me in thanking the committee, if you would. Also want to thank those who spoke in the public comment period for some really important and challenging ideas. We take your thoughts very seriously, and we'll weigh them carefully as we try to figure out how best we can move forward after this meeting. Our purpose of having this meeting is not just to have it and then go on about our business, but to see that this becomes a challenge for us to make sure that we are developing new products, whether those are medications, devices, or digital therapeutics, in the most efficient and effective manner. And that takes all of us working together, and we appreciate a number of the new ideas that we heard today. I just want to say a couple things that I've reflected on during the day. One, I've already been approached by people with some new ideas and new grants they want to submit to NIH, so to my mind, that's a success already, because that is one of the ways that I'm going to measure this meeting is, what do you all send to us in the next months, I hope months and not years, but at least in the next months, regarding new possible approaches to addressing smoking cessation? That's our number one goal. I think the other way that I took away from this, and I didn't expect it, but one of my colleagues reflected to me that when we looked at some of the presentations, we actually saw lots of NIH grants mentioned. And it reminded us that there are different pathways to going from sort of the academic or research perspective into submitting something to our colleagues at CDER or CDRH or CTP for consideration. And that our colleagues are already using those variety of pathways, and I look forward to partnering with many of you all in this room and online to find new approaches to bring your best ideas to market. That will be our goal, is to make sure that they can reach as many people as possible. I hope you were inspired as I was by the variety of presentations, and please reach out to those of us at NIH with your best ideas. You can easily find me online. If I'm not the right person, I will share it with my colleagues at NCI, with my colleagues at Heart, Lung, and Blood, with National Institute of Minority Health and Health Disparities, and the other partners that you saw represented here, and many others that were not here today, but represent the 27 different centers and institutes at NIH that join together to help address this public health concern. And with that, let me turn it over to Dr. King for the final words.
Hey, thank you very much. First, I just want to extend my appreciation for everyone who attended the session today, whether it was in person or via the electronic ether. You could have been anywhere in the world today, but you chose to be here, and I think that says a lot. So many thanks to those who attended, those who provided their oral comments, and also importantly the folks who will be submitting or have already submitted comments to the written docket. We were very intentional in ensuring that that would extend a little bit past the meeting in case something percolated in your mind today that you'll want to continue to submit. We definitely welcome that and appreciate those taking the opportunity to do so. As Dr. Compton noted, I also want to thank all the folks who are involved very heavily to make today's session a reality. I can say as someone who has been in the federal government for many years that these things don't materialize out of thin air. There is a lot of work that goes into them, and particularly one across multiple agencies that involves multiple institutes and multiple centers. So I'm really thrilled that we were able to make this happen, and I want to give a special shout out to our colleagues from Center for Tobacco Products Office of Regulation, also our Office of Health Communications and Education, who spearheaded a lot of the logistics today, including Dr. Priscilla Callahan-Lyon, and also Tracy Galloway and Denise Miller as well from CTP, who have done a formidable job making sure everything went seamlessly today. Thank you. And also want to thank Dr. Hyder-Warwick for his stellar moderating skills that kept us rolling and on time. We're even a tad bit ahead of schedule, which I think is awesome. And then also want to acknowledge all of our colleagues from both CDER, but also CDRH here at FDA, but also Office of the Commissioner, and also our colleagues at NIH. I'm thrilled that we were able to pull together this new working group, and this was one of our first tangibles from that group. And I'm really hopeful after this, and also the commentary that we just published earlier this week, that it's very clear that we do have a vision, we do have an interest in making momentum in this area, and we look forward to continued opportunity in that front. Now, I realize I am the last person today between the end of this session and whatever you've got going next, whether that's home, the airport, I don't know, a highball and some appetizers. So I'll be very brief, but in my last remarks, I just want to reinforce that today's discourse is an important part to what I hope is something that's going to continue thereafter. One of the things I absolutely loathe is, a bureaucratic shelf piece session, folks. And so we've had a lot of great dialogue. We've brought together a lot of folks, but we've got to continue that, and it's going to take a village. And it's not just federal agencies. That's going to involve non-government and government agencies, also those across the research, clinical, and public health spheres, and even the general public, and we've all got to rise to the occasion. I've said this publicly in the past. It's very easy to complain and point fingers, but we've got to rise above that and find solutions. And so I'm very hopeful that the dialogue we have today is a really important pivot point to that juncture to continue some substantive focus moving forward. And I'll close, and we've been hearing this all day, but I'll reiterate that tobacco smoking is the leading cause of preventable disease and death in this country. 30 million American adults continue to smoke, 500,000 deaths a year, millions more are living with smoking-related disease, and $600 billion with a B in terms of direct and indirect health care costs. And so this is a monumental issue at hand, and again, that's going to require a monumental approach. And so I'm very heartened by today's discussion, but I look forward to continue momentum moving forward. We certainly can and should do more, and that's going to require examining what we've done well, identifying areas where we can do better, and importantly, continuing the I word, innovation, folks. It's been said that innovation is the ability to see changes in opportunity, not a threat. I completely agree with that. And every once in a while, a new technology, an old problem, and a big idea can turn into an innovation. So let's make it happen, folks. Appreciate the time, and hope you have safe travels wherever your next destination is. Thanks for joining us today.